Welcome to biology for the MCAT exam. MCAT biology is very different from the other three science subjects, more specifically general chemistry, organic chemistry, and physics. And I'm speaking about your preparation. You see, for the other three science subjects, there are very clear equations which must be memorized and their usage must be understood. And in the case of organic chemistry, there are many mechanisms of reactions that must be understood and very few that actually must be memorized. In biology, however, there is a huge amount of material, yet the MCAT exam has questions designed such that even if you knew only a small percentage of the biology needed for the MCAT exam, you can still excel at this section. That's very different from the other sections. When it comes to physics, if you don't know F equals ma or the basic equations and how to use it, then there's no chance on the actual exam. Whereas for biology, there is a basic amount of material that you need to understand. But then after that, for the exam itself, many questions are based on material that you will be reading in the passages. In other words, they will present material to you in the passages, and most of the questions you will be able to find the responses in the passages based on, for example, experiments or graphs or tables presented in the passage. That is very different for this particular section of the MCAT exam. What this means is that, yes, it is important to review all the information for biology by reading the chapters in the gold standard and by going through this series of videos. However, even after you've completed that basic biology science review, you've only completed 25% approximately, in my point of view, of your preparation for this section of the MCAT exam. The rest of your preparation is going to be dependent upon doing questions, specifically doing all the questions which are the practice items published by the AAMC and doing all the exams published by the AAMC and all the gold standard exams. That will be the most critical part of your review or your preparation for this section of the MCAT exam. Having said that, let's begin our review of biology. The functional unit of biological systems is the cell. The cell has many different components. The cell has fluid which bathes the interior of the cell. That is called the cytosol. In fact, the prefix cyto refers to cell and sol refers to solution. So the cytosol is the fluid that bathes the interior of the cell. There's also the cytoskeleton, which provides shape or form to the cell and also is involved in movement for the cell. Then there is the cytoplasm, which is the rest of the stuff inside of the cell, including the organelles. Surrounding the organelles and surrounding the cell is the plasma membrane. We're going to look at the plasma membrane first. So, if we were to examine the plasma membrane which surrounds the cell and also surrounds the organelles inside the cell, we would find that they are composed of phospholipids. This molecule is called a phospholipid because it has a phosphorus head, and we know that phosphorus, or the phosphate anion, is charged. And so if it's charged, that means it can associate with water molecules because of the delta negative and delta positive charges, which will create a force of attraction. Therefore, the phosphorus head will be hydrophilic. Hydrophilic literally meaning water-loving. Then there are the hydrophobic tails, phobic meaning afraid of. So afraid of water tails, which are composed of lipids. Lipids are fats, and of course, as you know, if you put oil in water, the oil does not dissolve in the water. They remain immiscible. They do not dissolve one into the other. The oil avoids the water. So here we have the hydrophobic tails, the hydrophilic heads. These phospholipids arrange themselves in a regular array in the plasma membrane. They arrange themselves such that the hydrophobic tails stay at the interior part of the membrane, and the hydrophilic heads are on the outer parts of the membrane. So it creates a bilipid layer, 2 layers of the lipid, and that is one plasma membrane. So every plasma membrane that you see are surrounding the cell or inside of the cell, they have a bilipid layer with the heads facing outwards so that it can associate with the fluid that is either around the cell or the cytosol itself. Embedded in the plasma membrane are proteins. You can see a large protein here which is within the membrane. It spans the membrane, and therefore this protein is called either an intrinsic protein because it's in the membrane, or an integral protein. There are also proteins that are associated with the outer part of the membrane. So these are called peripheral proteins, also called extrinsic proteins. So the arrangement of the phospholipids and the proteins creates a model which is called the fluid mosaic model. The reason it is called the fluid mosaic model is because the phospholipid portion of the plasma membrane is fluid, meaning that it moves about, it diffuses all over the different parts of the plasma membrane freely. It's not held in any particular position, so it is fluid. The mosaic part of the plasma membrane are the proteins. The proteins are sort of spread out in the plasma membrane, and they have limited mobility. In fact, you can imagine the proteins in the membrane being somewhat like in raisin bread, the raisins in raisin bread. If you imagine a slice of raisin bread, if you examined it carefully, you would see that there are some raisins that span the slice of bread. There are some raisins that are just at the top, some that are just at the bottom. And that sort of mosaic of raisins in your raisin bread would be a reasonable analogy to the proteins which are in the plasma membrane. Now, if you were a molecule or an atom and you wanted to cross the plasma membrane, you would find that the plasma membrane is semi-permeable, meaning that it is not available for being crossed by just anything. If anything can cross this plasma membrane, then we would say that the plasma membrane was permeable. But the plasma membrane prevents or impedes certain things from crossing, so we say that it is semi-permeable. If you were something that was small and you had no charge on you, then it would be relatively easy for you to cross the plasma membrane, because as a molecule you would be able to go through the system, and if you're not charged, you wouldn't have a problem with the lipids in between. If you were a lipid yourself, then dissolving into the plasma membrane to cross would be relatively easy. Like, for example, a steroid, which there are many steroid hormones that have to dissolve in the plasma membrane in order to get across. However, if you were highly charged, or if you were a very big molecule, then crossing the plasma membrane would be quite difficult. And often you would have to rely on something that spans the membrane. For example, a protein that spans the membrane, like receptors in the plasma membrane, to help you get across the plasma membrane. Now, there are different ways to cross the plasma membrane in general. You can cross the plasma membrane either by diffusion or by facilitated transport. Now, there are a couple of graphs that are very important for you to know about diffusion or facilitated transport, because it comes up often on the MCAT exam. Take, for example, the following. If we had a certain substance outside the plasma membrane, and we were calculating what its concentration was within the plasma membrane, Then we continued to increase its concentration outside the plasma membrane, and then we measured the rate at which it was crossing the plasma membrane. Well, then we would get something like this. So what we have here along the y-axis is the rate to cross the plasma membrane. Here along the x-axis we have the gradient. For example, we could imagine that we have a certain concentration inside the plasma membrane, and then we start increasing the concentration of our substance outside the plasma membrane, meaning we're increasing the gradient, the difference between the concentration outside and the concentration inside the plasma membrane. So this is increasing the gradient. So one would imagine that if you increased the concentration outside and the concentration inside was very low, that things would just start moving inside just by simple diffusion, meaning that it's crossing the plasma membrane in a sense that demonstrates that the difference in concentration, the concentration gradient, suggests that it will move from a position of high concentration outside the membrane to a low concentration inside the membrane. It would move to that position until the concentration outside the membrane and inside the membrane were the same. And so, if we were to imagine that something was moving across the membrane by simple diffusion, this is what we would expect. We would expect that as we continue to increase the gradient and continue to increase the amount of the substance outside the plasma membrane, if it's just moving across the membrane by diffusion, then the rate at which it's crossing the membrane would continue to increase as we increase the concentration of the substance outside the plasma membrane. However, if we were to see this, then we have a different story, because we would notice that, okay, we're increasing the gradient, we're increasing the concentration, but then it gets to a point such that the rate to cross the plasma membrane has reached a peak. It doesn't go beyond that amount. Now, if we were talking about simple diffusion, and that we have a whole cell like this, and we're increasing the concentration of something outside of the cell, one would expect things just to start moving across more and more and more as we increase the concentration outside. If it got to a point where it peaked at some level, it would mean that there is a limiting factor within the plasma membrane. There is something that's actually assisting the chemical or molecule to cross the plasma membrane, and that once you started increasing the concentration outside the plasma membrane so much, it got to the level that whatever was helping to cross the plasma membrane became saturated, meaning it was working overtime. It was moving as fast as it possibly could move, so that even if you start putting more concentration of your substance outside the plasma membrane, nothing more could happen because it's already moving at maximum capacity. That is called saturation kinetics. So the movement is saturated. It is at its maximum capability. And so when you see that the curve flattens out like this, that suggests that there is saturation kinetics. It suggests that there are a limiting factor in the plasma membrane. And now knowing about the fluid mosaic model, that suggests in turn that there are proteins in the plasma membrane or receptors in the plasma membrane that help to take things from higher concentration down to lower concentration, etc. So this would be the limiting factor. And the idea that there are proteins or substances in the membrane that will assist molecules to go from high to low concentration would be called facilitated transport. In other words, it is helping to transport the molecules from one side to another. The opposite of facilitated transport is active transport. Active transport, as its name suggests, takes molecules or ions from an area of a low concentration up to an area of high concentration. Therefore, active transport transports species against their concentration or electrical gradient. So it goes against the gradient. And because it moves against the gradient, it requires energy. And energy is in the form of ATP. So we have two ways to cross the membrane using some sort of carrier. Therefore, these two ways, facilitated transport and active transport, are both called carrier-mediated transport systems. And they would use some sort of protein in the plasma membrane to get across. And so they would both exhibit some sort of saturation kinetics. Facilitated transport, because it goes along the concentration gradient, does not require energy. Active transport does require energy as it goes against the concentration gradient. The most famous receptor or protein in the plasma membrane that uses active transport is the sodium-potassium ATPase. Now, the sodium-potassium ATPase, or pump, is a receptor that we would look in greater detail when we come to neurons, because it's particularly important for neurons. Nonetheless, there is another way in which molecules or species can cross the plasma membrane, and that is they can enter the cell or they can exit the cell by a process called either endocytosis or exocytosis. And cytosis sort of stands for cell eating. So endocytosis is when the cell will bring something into the cell, and it does so by having a molecule or something that lands onto the cell. Then part of the cell membrane will invaginate, pinch off, and then enter the cell as a vesicle or a little ball of material. And that will enter into the cell. That would be called endocytosis or cell eating. A special case of endocytosis is called phagocytosis, which we see with white blood cells when they attack bacteria or viruses. And then those bacteria or viruses enter the cell by endocytosis or phagocytosis. So that's one way. When material will exit the cell, that would be called exocytosis. And so there could be vesicles within the cell that are leaving the cell. They go to the cell membrane, they fuse with the cell membrane, and then they release their contents to the exterior. So these are the different ways that substances can cross the plasma membrane, either simple diffusion, some sort of facilitated transportation or active transportation, one way or the other, a form of carrier-mediated transport, or it can enter or exit the cell by endocytosis or exocytosis. Now, let us look at the inside of the cell. The component within the cell that you can't see is the cytoskeleton, which I've mentioned before as providing the shape and helping in movement for the cell. The cytoskeleton is composed of three different elements. There are the microfilaments, which are the smallest. Then there are the intermediate filaments, which are, well, intermediate in size. And then there are the microtubules, which are the largest of the cytoskeletal elements. Microfilaments form actin and myosin, which are very important in the movement of cells, in particular the movement of muscle. Intermediate filaments and microtubules, they serve as railroad tracks within the cell, all over within the cell, including in certain parts of neurons called the dendrites or axons, in which they serve as railroad tracks to help to move things along the cell. Microtubules, in particular, have many other purposes. They serve as the core of flagella or cilia, which are things within the cell that sort of stick out upon this, and they create movement. Cilia sort of have finger-like projections, which can move things along passages in the body. And of course, the flagellum is a whip-like organelle, which is found in sperm, which helps the sperm to move along when it requires. And so there are many different purposes for the microtubules, including sometimes the microtubules are arranged in a cylindrical structure called a basal body. And a basal body is two cylindrical structures at right angles to each other, and each one of these cylindrical structures would be called a centriole, and that is microtubules arranged in this particular type of structure. Basal bodies can be found at the base of flagella, or they can also be found in associated with microtubules with the mitotic spindle, which is something that we'll be seeing a little bit later. So those are the cytoskeletal elements which are within the cell, which we can't really see. But among the organelles that we can see here the mitochondria. Now, a mitochondrium is a organelle which has actually two unit membranes. So there's the unit membrane around the cell, there's a unit membrane around the mitochondria, and there's an inner membrane of the mitochondria. The inner membrane of the mitochondria is arranged in shelf-like folds called cristae. And the inner part of the mitochondria, the fluid within the inner membrane, is called the matrix. And the matrix contains the enzymes required for the Krebs cycle, which we'll be looking at later. So the mitochondria is also called the powerhouse of the cell because that is where we have the Krebs cycle, and therefore it's a very important source of ATP for the cell. Over here we have a lysosome, and the lysosomes are also called the suicide sacs for the cell. And that is because within a lysosome, it contains the enzymes required to kill or destroy the cell. Of course, those enzymes would only be released to the cell if the cell was dying or was old and it had to be destroyed. But in terms of other activities that lysosomes are actively involved in is, for example, if there was a bacteria or a virus that was ingested by the cell by endocytosis or phagocytosis, as mentioned before, then that bacteria or virus would enter the cell and fuse with the lysosome. And that would be called the secondary lysosome after they have fused. Then the enzymes within the lysosome will destroy the bacterium or the virus into its individual components. And then those components would be used by the cell for other purposes. So actually, by having the infection or whatever it is, the cell can take in the components of that whatever organism it is that caused the infection and then destroy it, cut it up into parts, and then reuse those parts for the cell for other purposes. So that is the lysosome. Then over here we have a flattened group of sacs, and this is called the endoplasmic reticulum. The endoplasmic reticulum is known as the synthesis center for the cell. The endoplasmic reticulum can have these dots on them, which are actually ribosomes, which we will look at ribosomes a little bit later in more detail. And when it has these ribosomes or dots on it, the endoplasmic reticulum looks a bit rough, and therefore it's called rough endoplasmic reticulum. Rough endoplasmic reticulum is important in terms of synthesizing proteins. So this would be the synthesis center. When the endoplasmic reticulum is smooth, it is more important in terms of the production of fatty acids. Then there is this group of sacs, which is called the Golgi apparatus. The Golgi apparatus is known as the export center of the cell. And so proteins that are produced by the rough endoplasmic reticulum may go to the Golgi apparatus if these proteins need to be released by the cell for whatever purpose. And so the Golgi apparatus would release these proteins within vesicles, and it would go to the cell membrane, either to be proteins within the membrane or to be released by the cell entirely through the process which we mentioned as exocytosis. So if you were to think of different cells that would be where these particular organelles would be very important, you may want to pause and consider what cells you think these particular organelles would be very important. Mitochondria clearly in muscle, because muscle requires a lot of energy, movement, and therefore mitochondria. A lysosome, because it could help to destroy particles, that would be very important in terms of cells that attack infection or bacteria or viruses, such as white blood cells. Then, in order to produce a lot of protein, well, there's a number of cells in the body that produce a lot of protein. Muscle cells produce a lot of protein, actin and myosin. And there are other cells, for example, white blood cells. There are some white blood cells that create antibodies. And those blood cells that create antibodies, those are proteins. And those antibodies would be produced in the rough endoplasmic reticulum. They would be processed by the Golgi apparatus, and then they would be exocytosed, so these antibodies can be released into the bloodstream to attack bacteria or viruses. And so those particular type of white blood cells, which we'll be looking at later, they happen to be called B cells. They would be loaded with rough endoplasmic reticulum and the Golgi apparatus. And then, of course, there is the nucleus of the cell. And the nucleus of the cell has a nuclear envelope surrounding the cell. The nuclear envelope has two unit membranes, and there are pores, called, of course, nuclear pores, where certain molecules are able to cross the nuclear pore to enter the nucleus itself. Within the nucleus, we have DNA, and the DNA is arranged in the nucleus. We might see it arranged as chromatin, which is a complex of DNA and proteins called histones. And that complex is called chromatin, or we might see them as chromosomes. And chromosomes is when the chromatin arrange themselves in a certain form that can be seen with a light microscope. Also, we see here the small nucleus, which is called the nucleolus. The nucleolus contains some rRNA and some parts of the DNA. Now, the nucleolus is not membrane bound. Now, a common reasonable question that they may ask on the MCAT exam is, how many plasma membranes would you cross as a minimum in order to go from the outside of a cell to the inside of either a mitochondria or the inside of a nucleus? And the answer would be 3, because you would have the plasma membrane on the outside of the cell. You would have 2 plasma membranes to cross in order to get inside of the mitochondria, in the inner part of the mitochondria. To get into the inner part of the nucleus, you would again have the outside membrane as a minimum, and then you would have the 2 membranes of the nuclear envelope. So those would be possible questions that they could ask. The other thing which is interesting about the cell is that the great majority of DNA is within the nucleus. However, there is DNA found within the mitochondria. The only place in a cell where there is DNA outside of the nucleus is found in the mitochondria. Now that we've talked about DNA, let's look at it in more detail. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. Thus, both DNA and RNA are nucleic acids. The subunit of a nucleic acid is the nucleotide. A nucleotide contains 3 main groups: a phosphate group, a 5-carbon sugar, and a nitrogen base. In terms of the 5-carbon sugar, the sugar may be ribose in the case of RNA, or deoxyribose in the case of DNA. Deoxyribose meaning there's 1 less oxygen than there is in the sugar ribose. And this is why DNA is called deoxyribonucleic acid. Then there are the nitrogen bases. They are divided into 2 main categories: purines and pyrimidines. The purines are adenine and guanine. Some students like to remember purag. So the purines are adenine and guanine. It doesn't make any sense, but it makes it easy to remember. Then there's pyrimidines. And the pyrimidines are cytosine and thymine. That is for DNA. RNA replaces thymine with uracil. So this is the nucleotide. It is also called a nucleoside phosphate. Now these nucleotides polymerize, so they add to each other in such a way that the phosphate group will bond to the OH group of another sugar. This is done via bonds that are called phosphodiester bonds. And in this way, this will continue to polymerize the phosphate to a sugar, to the phosphate to a sugar, etc. Thus, the bases will be sticking out to the sides, so that when it does polymerize, you have the backbone, which is the sugar with the phosphate groups in phosphodiester bonds. Then you have the bases sticking out, which are the pyrimidines and the purines. Now, if you could imagine this double helix as being like a winding staircase, it is as though the stairs of the winding staircase are the nitrogen bases. The nitrogen bases bond to each other in a very specific way. Adenine bonds to thymine, guanine binds to cytosine. Furthermore, they bond to each other by hydrogen bonds. And it is such that adenine always has 2 hydrogen bonds to the thymine, and guanine always has 3 hydrogen bonds to the cytosine. So in this way, we can see that by having more guanine or cytosine in the DNA, it increases the stability because there are more hydrogen bonds. The replication or duplication of DNA is done in a semi-conservative fashion. And what that means is that each strand, every time you see DNA in a double helix, one of the strands must be an old strand or the parent strand, and the other strand must be a new strand or the daughter strand. So if this particular double helix were to replicate or duplicate itself, the way it would do so, you could imagine that it would unzip itself to open up itself like this, and then each one of these strands would serve as a template so that new nucleotides can come in and bond in such a way that A to T, G to C, etc. So by unzipping, the nucleotides would be coming in and bonding and then creating a new double helix on each side. Therefore, each double helix, each new double helix would have a parent strand, a strand that was there before, and a brand new strand that is created by the new nucleotides. So those would be the daughter strands that would be created. This would therefore create complementary strands of DNA, the daughter strand and the parent strand. And because the DNA bonds in a way that there is a phosphate group to the OH group with a sugar, it so happens that the OH group is on a particular carbon on the sugar called the 3' carbon, and the phosphate group is on the 5' carbon. So the way that DNA will be produced, one of the strands will end with 5' and the other strand will end with 3'. Thus we will produce new DNA molecules through replication. And it is these DNA molecules that are often called the blueprint of life, because through DNA and RNA, the proteins of the cells can be produced, and these are the enzymes, the antibodies, the receptors, etc. of the cell. We will now look at the anatomy of the chromosome and the cell cycle. So here is a chromosome, and the chromosome, the ends of which we call the telomere, and the center where there's a constriction, there we call that the centromere. Also, just at the outer part of the centromere are proteins. It's a protein complex where the microtubules will insert in order to literally pull the chromosome apart. That complex of proteins on the outer part of the centromere is called the kinetochore. Now, when you look at each chromosome, it contains two chromatids. So this would be considered one chromatid, and there's another chromatid here. Each chromatid contains the double helix of DNA. So when we saw the double helix, you have to imagine that that double helix is super wound, turned, and twisted all around in one chromatid. So that means each chromosome contains two double helices of DNA. Now, in the cell cycle, some students like to remember the order of the cell cycle as with the mnemonic P mad-I. And so this permits you to memorize the order of what occurs in its prophase, metaphase, anaphase, telophase, and then there is the interphase. Now, in the prophase, the chromatin material, which is sort of diffuse and indistinct, becomes condensed into chromosomes. And so it is in prophase that we start to see the chromosomes more clearly in the cell. Then the next phase being metaphase, and metaphase is where the chromosomes in the cell will begin to line up along what is called the equatorial plate. So we see the chromosomes beginning to line up in a particular arrangement. The microtubules will be inserting into the kinetochores, and then you would see an area where there's the basal bodies and there's other parts of the spindle, which means other parts of microtubules which are moving outwards. That is prophase, metaphase. Then we move on to anaphase. And in anaphase, the microtubules will pull the chromosomes apart. And so each chromatid is being pulled towards the area where the basal bodies are. So you see each chromatid being pulled away from its sister chromatid. And then in telophase is where we start to get the nuclear envelopes being reformed. As the genetic material has been completely separated, the nuclear envelopes can be reformed, and then we can enter into interphase. Interphase is composed of three different components: G1, S, and G2 phases. G1, it literally stood for gap 1, and it was because when people looked in the microscope, it looked as if nothing was happening in the cell. But actually, there was a lot of activity occurring in the cell, and what it was that the cell was preparing for synthesis. In other words, the cell was getting all the proteins and enzymes and so on ready for DNA synthesis or replication. Then, in the synthesis phase or replication, that's where the chromatid will literally unzip itself, and then the complementary strands and so on, the nucleotides would be coming in, and the polymer would be formed using the enzyme DNA polymerase. And that would occur in the synthesis phase. Then in G2, which is the second gap, that is when the cell starts to prepare for prophase, metaphase, anaphase, and telophase. This part of the cell cycle, interphase, represents approximately 90% of the cell cycle. In other words, this part of the cell cycle with prophase, metaphase, anaphase, and telophase is only about 10% of the cell cycle. And this part of the cell cycle is what is called mitosis. Now consider the following classic MCAT type question. A drug is added to a petri dish containing cells which are actively dividing. If the drug inhibits the production of cytoskeletal elements, so in other words, the cytoskeleton, in which stage of mitosis will cells be arrested? Prophase, metaphase, anaphase, or telophase? You may want to pause and consider your response. Okay, so the correct answer would be prophase, even though many students would write metaphase or anaphase. because students remember that the cytoskeleton includes microtubules, and microtubules are very active in anaphase as they help to pull the chromosomes apart in their sister chromatids. And in metaphase, people do remember that the microtubules are attached to the chromosomes. However, the cytoskeletal elements in a cell are the elements that create motion within the cell for the organelles and the chromosomes, etc. In other words, the chromosomes would not be able to line up in the equatorial plate if it is not for the fact that there are some microtubules and so on to move them into that position. They cannot line up by themselves. In the cell, you can imagine DNA like royalty. It sits in the nucleus, and that's where it directs the activities of the cell. When it wants something to be done in the cell, enzymes to change, receptors, whatever needs to be done in the cell, or something that needs to be exported like antibodies or whatever, then the DNA will send a messenger to get the job done. So the information from the DNA will be transcribed in the nucleus into a messenger called messenger RNA. It is the messenger that will leave the nucleus, go into the cell, and have the message on the messenger RNA translated into a way that the cell can understand and do something, do some sort of activity. And that translation will be into a protein. And the proteins are the workers of the cell. They are out there to get the job done. So let's look at the story more specifically. To begin with, the information on the DNA, which will be transcribed and then translated into a protein, is called a gene. I use the word protein, but it also stands for polypeptides as well. A polypeptide is exactly like a protein, but much smaller. So the information which will be translated and transcribed into this information, that information is called a gene. So when the messenger RNA is being produced in the nucleus using the DNA as a template, now of course we've seen that the DNA is a double helix. The RNA molecules are single-strand molecules. And this is being produced using DNA as a template, transcribing the information. The information is not word for word for a couple of reasons. One is that we've already seen that RNA writes its messages differently than DNA. Uracil is the base in RNA as opposed to thymine, and furthermore, the sugar is ribose as opposed to deoxyribose. That's one issue. The other issue is all the information in a gene on the DNA is not necessarily useful information. Some of this information would be called introns. That's not useful. It's not expressed. The information which is expressed are called exons. So those are expressed information. Those are information that will be translated into a protein. So the RNA will have to take all the information from the gene, and then will have to start maturing itself, removing the introns, which are not important, and keeping the exons, which will be translated. So once the messenger is created, then it will leave the nucleus. So we imagine some sort of single-strand molecule, which is the messenger. So the messenger leaves the nucleus, goes into the cytoplasm or cytosol, in where its information will be translated. In order for it to be translated into a protein, it needs the proper environment. The environment for translation is provided by a ribosome. A ribosome is a complex of proteins and what is called ribosomal RNA. We mentioned earlier that ribosomal RNA is concentrated in the nucleolus of the nucleus. So ribosomal RNA and proteins will complex in the cytosol or associated with the rough endoplasmic reticulum, which is studded by ribosomes. And so this complex called a ribosome, which is rRNA and protein, will provide the necessary environment for the mRNA to be translated. Now, when we look inside and we see the messenger and the environment, what we will see on the messenger is a code. The code for genes is a triplet code. That is called the genetic code. And so when we look up closely, we will see on the messenger RNA, in the environment, some sort of triplet code of 3 nitrogen bases. For example, it might say AUG. So AUG might be the code that shows up within the ribosome. So in other words, there's the translation that will be taking place from the messenger RNA into an amino acid, which is the building block for a protein, will come from the genetic code, which is a triplet code. So AUG is present here. That code on the messenger RNA is called a codon. So we have the code, we have the environment, now we just need something that will translate this code into an amino acid and take that amino acid and transfer it onto another amino acid and another amino acid, etc. And all these amino acids linked up together will form our protein. So the molecule that will be able to read the codon, transfer an amino acid, is called transfer RNA or tRNA. So we imagine tRNA as a molecule that will have one part which will be able to identify the codon and another part that will be carrying an amino acid. This part that will identify the codon will be called the anticodon because it will bond to the codon. So this would be 3 bases that would identify this particular codon. And these 3 bases will always come with this particular amino acid. So you may want to pause here and consider what would be the names of these 3 bases on the anticodon which would identify this particular codon of AUG. Well, the complementary bases for AUG would be U, A, C. The reason is because we saw that in DNA, A bonds to T, G to C. But we also said that thymine is replaced by uracil in RNA. And these molecules are RNA molecules. mRNA, there's tRNA, and of course the environment is in part prepared by rRNA. So these are RNA molecules. So T is out of the question and we're only talking about U. So then A will bond to U instead of T, and then U will bond to A instead of T to A, and G bonds to C. So UAC is the anticodon which would attach to UAG. And when it comes in, it attached, it leaves off its amino acid, it goes away, the ribosome moves over, reads another part of the genetic code, another triplet of nitrogen bases, another transfer RNA will come in, read the codon, bond with its anticodon, release an amino acid, transfer it onto the polypeptide which is growing into a protein. So that is how the translation occurs. Now in terms of the protein, the subunit of proteins are amino acids. And amino acids are called amino acids because on one end there is an amino group, which is the NH2 group, and on the other end there is an acid group. And of course, when you talk about organic chemistry, an acid group in organic chemistry is a carboxylic acid. So it's COOH. So that is the acid group which is on one side. So it so happens that as our polypeptide is being produced and getting longer, On one end, there's going to be the amino group. That's the first thing that comes out. Then, in the amino acid, there's the carboxylic acid group. That will bond to the amino group of the next amino acid. The bond between that carboxylic acid group and the amino group, that's called a peptide bond. So the peptide bond produces the polypeptide, which produces the protein. So then at the beginning of the polypeptide, you will have one amino group sticking out. Then when you go all the way to the end of the polypeptide or protein, you will have the carboxylic acid group that will be sticking out at the other end. So that is how the protein is being produced. Now, if the ribosome is just free in the cytosol, and it meets with the messenger RNA and the transfer RNA, and this reaction is occurring and the protein is being produced, then this kind of protein might be used within the cell for some cell purpose like enzymes within the cell, etc. If this happens to be a ribosome, which is studding the rough endoplasmic reticulum, then this protein is being pushed into those flattened sacs of the rough ER. And then after it is produced by the rough ER may send it to the Golgi apparatus, where it will be further processed and then exported to the cell membrane. That is where those proteins might be used within the cell membrane itself, or might be exported for other purposes, for example, antibodies or a protein enzyme. Despite its appearance, this is not a UFO. It is a virus, and viruses are very interesting. For one, these are obligate intracellular parasites. In other words, they cannot survive without using a cell to replicate themselves. And viruses are often in this gray zone as whether or not they're alive or dead. And most people feel that they're probably not alive because they can't grow, they can't replicate on their own, they don't have independent metabolism, etc. And they have no cellular structure. But nonetheless, the important features of a virus for the MCAT exam is that it is composed of a head and a tail. The head part has a protein capsule called the capsid, and within this protein capsid is the genetic material, which may be DNA or RNA as genetic material, which is extremely unusual. The tail part is structured such that it can attach to a cell. And so a virus that is able to attach to a bacterium and infect bacteria is called a phage or a bacteriophage. As we know from personal experience, viruses can attack people as well, as you know from having a cold at some point in your life. And so when a virus attacks a cell, it will land on the cell, it will inject its genetic material into the cell, and it will force the cell to replicate more viruses. If sometimes the cell will replicate so many viruses that it will burst and release these viruses. That would be called a virulent state. Now, the virus may have two aspects to the life cycle. It may have a life cycle in which it infects and bursts cells and releases more viruses. That would be called either the lytic life cycle, lytic meaning to burst or tear apart, or it might be called the virulent life cycle. In the life cycle in which the virus remains dormant and quiet, that would be called the lysogenic life cycle. When you get into medical school, you will study microbiology in much greater detail. The microbiology for the MCAT exam is very basic. Microbiology is essentially the study of bugs, viruses, bacteria, and fungi. And so bacteria are considered to be prokaryotes, whereas fungi are eukaryotes. Prokaryotes being pre-nucleus, karyote referring to nucleus. Eukaryotes being true nucleus, literally. And so eukaryotes include fungi, but it also includes all animal cells and plant cells, etc. Commonly on the MCAT exam, they specifically try to determine whether or not you know the difference between prokaryotes and eukaryotes. And so they would tend to ask questions to delineate that difference. So I've written a number of things here: nucleus, cell wall, ribosomes, organelles, and plasma membrane. I would like, based on your knowledge, you can pause here and you can see if you remember if these particular things represent a similarity between eukaryotes and prokaryotes, or a difference between, meaning that they're present in one but not the other. So when we look at nucleus, clearly there is no nucleus in a prokaryote. That's why they're pre-nucleus. And there are nuclei present in all eukaryotes, true nucleus. For the cell wall, you might have thought that that's a difference between the two, because you may have remembered that bacteria have cell walls. And that is indeed true. Bacteria do have cell walls, but some eukaryotes have cell walls, like fungi. They have cell walls. And so it's not a difference, but rather this is a similarity. So nucleus is a difference, cell wall is a similarity. Ribosomes is a similarity. Both bacteria and eukaryotes do have ribosomes. That helps to produce the proteins which we saw earlier. Then there are organelles. Prokaryotes have no nuclei and no organelles. So they have no mitochondria, they have no endoplasmic reticulum, etc. But they do produce energy. They can produce ATP like eukaryotes do, but they don't produce them using a mitochondria. Rather, they have their own internal membranes in order to produce ATP. In terms of plasma membrane, indeed, this is a similarity. Both bacteria and eukaryotes have plasma membranes. We already saw how the plasma membrane develops and is produced and exists in eukaryotes. And for the prokaryotes, they have a cell wall, but they indeed also have a plasma membrane associated with the cell wall. So they have both. So this is actually a similarity between the two. We will now look at bioenergetics. Bioenergetics is the study that looks at how biological organisms, like our cells, produce and use energy. We're going to begin the story at the end. ATP. ATP is adenosine triphosphate. ATP is the number one short-term energy storage molecule. This is the molecule that we will break down to produce energy so that we can exercise, do sports, sit, read, learn the MCAT exam, whatever it may be. ATP is broken down into ADP, adenosine diphosphate, and an inorganic phosphate molecule. It is that reaction, the breaking down of ATP, that permits us to have the energy necessary to do biological processes. Now, there are many different mechanisms in order to produce ATP. And there are many different sources in order to go through mechanisms to produce ATP. The most common source to produce ATP is glucose. Of course, glucose is a carbohydrate. It is stored in our liver as glycogen. It is also stored in muscle cells as glycogen as well. it is broken down, it floats through our bloodstream, and then it goes to our cells. And then actually will enter our cells, and that's when we will use it. But there are other molecules that we can use as well. For example, lipids. Lipids can be broken down from fat, and then the lipids can be sent into the Krebs cycle, where the energy from the lipids will produce ATP. There are also amino acids. Amino acids are broken down from proteins, and the amino acids can be fed into the Krebs cycle as well in order to produce energy. Now, the first source of energy that we use is glucose. If we start running out of that, then we will start using the lipids. And of course, lipids is stored in our body as fat. And then when that is broken down and we still require energy, that's when we start going after the protein. And protein is stored in our body as muscle. Now, glucose is our number one source of energy, and then we look at the different mechanisms in order to produce ATP. The first mechanism is to break down glucose, and that is called glycolysis. Now, you may recall that lysis is a word that is used often in biological processes to infer that something is broken apart. So glycolysis is the breaking down of glucose. We just call it glycolysis. Now, the first step in glycolysis is that once the glucose enters the cell, and we'll see later that that process is helped by insulin, when the glucose is in the cell, the first thing that occurs in glycolysis is that a phosphate group is put on the glucose molecule. There are two reasons for this. To begin with, phosphate is a large, relatively large, negatively charged group. By putting this on the glucose molecule, it will prevent glucose from diffusing out of the cell. Because you may remember that when you have a charged molecule, it is less likely that it will cross the plasma membrane because of the lipid interior. So that's one good reason to put a phosphate group on the glucose. The second good reason is because just by putting the phosphate group on the glucose, you've changed the glucose, you've altered it. And therefore, the concentration of glucose within the cell suddenly becomes much lower. And because you'll have more glucose outside of the cell, you've therefore increased the gradient for glucose to enter the cell, which is a good thing for the cell because the cell needs sources of energy. So then the first thing is glucose is phosphorylated. It has a phosphate group placed on it. So now we have glucose 6-phosphate. Then glucose will change its form and become fructose. Fructose and glucose are both 6-carbon sugars, and so the only thing that's going to change is the order and kinds of bonds, and therefore these are isomers. Glucose 6-phosphate and fructose 6-phosphate are indeed isomers. Now fructose will have another phosphate group placed on it. So it goes from fructose 6-phosphate to 1,6-diphosphate. Now, the interesting thing about this is that by having two phosphate groups on one molecule, these are two relatively large negatively charged groups that are attached to the one molecule. Well, this is quite unstable because, of course, like charges repel just as opposite charges attract. So by having these two large like charges that are repelling each other on the same molecule, this is going to be part and parcel of the fact that this molecule is going to break apart. So you have this 6-carbon sugar with two negatively charged groups, which will break apart into 3-carbon sugars, 2 of them. And that accounts for the 6 carbons. So now we will have 2 phosphate trioses. The word trio suggests there are 3 carbons, tri, and ose suggests that it's a sugar. Just like fructose and glucose are sugars, well, so are these phosphate trioses, which are produced as a result of the lysis of the glucose molecule. Then these trioses will go through a series of steps and then produce pyruvate. And pyruvate becomes the key step in the series of steps of glycolysis, because it's with pyruvate that the decision has to be made. Is there oxygen or is there not any oxygen? Because so far, the series of steps that we have done in glycolysis did not require oxygen. They are a series of steps that just occur in the cell's cytosol, in the solution in the cell. Now the decision has to be made, oxygen or not. If there is no oxygen, then we continue along glycolysis, and the pyruvate will produce lactate, or lactic acid. And then we end up with a net of 2 ATP. So for per molecule of glucose, we end up with 2 ATP, which is relatively very little energy. However, in order to get that 2 ATP, it occurs very quickly. The other possibility is that the pyruvate will be converted to acetyl coenzyme A, that is, if there is oxygen present, and then the acetyl coenzyme A will enter the Krebs cycle, which occurs within the mitochondria of the cell. So those are the two possibilities of the conditions. Either it is anaerobic, which means no oxygen, or it is aerobic, which means there is oxygen present. One quick note about the enzymes. There are two types of enzymes that you really need to be able to recognize, and those are kinases and phosphorylases. Those are simply enzymes that can add or subtract phosphate groups, and those are enzymes that you should be able to recognize. So that is one of the possible mechanisms, is glycolysis, which will produce 2 ATP per molecule of glucose. The other possible mechanism is that the pyruvate will send acetyl coenzyme A into the mitochondria, and the acetyl coenzyme A in the mitochondria will enter the Krebs cycle. The Krebs cycle is also called the tricarboxylic acid cycle. For every molecule of glucose, you get 2 turns around the Krebs cycle. There are 4 important products of the Krebs cycle. For one, there is carbon dioxide produced as a waste product of the Krebs cycle. Because it is a waste product, we need to get rid of it. And carbon dioxide is a molecule that we get rid of in the lungs. We just blow it off in the lungs all the time. And that is a waste product of the tricarboxylic acid cycle, or the Krebs cycle. The second important product that comes out of the Krebs cycle is GTP. GTP is a molecule similar to ATP. And in fact, GTP, for every 1 molecule of GTP, it is equivalent to 1 molecule of ATP. Another important product of the Krebs cycle is NADH plus H. NADH plus H is considered to be reducing equivalents. They are sometimes called hydrogens. They are also sometimes simply called electrons. But NADH plus H is an important product coming from the Krebs cycle. And the fourth product is FADH2. And again, FADH2 is sometimes called reducing equivalents or hydrogens. So these are the 4 important products. It is interesting to note that ATP is not directly produced from the Krebs cycle. After the Krebs cycle, we go to the electron transport chain. And all these things, the Krebs cycle and electron transport chain, are associated with the matrix of the mitochondria and the inner membranes of the mitochondria. Remember, the mitochondria is considered to be the powerhouse of the cell. This is the most efficient use of glucose in order to produce the maximum number of ATP molecules. So now we get in the electron transport chain a series of events in which the hydrogens from FADH2 and NADH plus H go through a series of steps, and the final step is that oxygen receives the hydrogens, producing water as the final waste product of the Krebs cycle. So water, H2O, being a waste product just like carbon dioxide, is usually released in the kidneys, but there are a series of other ways that we can get rid of water. So this is the way that energy is produced. Now it so happens that for every one molecule of NADH plus H, you end up producing 3 ATP. For every one molecule of FADH2, you end up producing 2 ATP. So when you calculate all of this, you end up that if you take glucose as your source of energy and it goes through the Krebs cycle, an electron transport chain, in other words, you take glucose and it goes through aerobic respiration, you end up with a total of 38 ATP per molecule of glucose. Your net ATP will be 36 because it will cost you 2 ATP in order to get the acetyl coenzyme A into the mitochondria. So then you get a total of 36 ATP per molecule of glucose during aerobic respiration, but you only get 2 ATP per molecule through anaerobic respiration. Now there is something called oxygen debt. That is that you run to class because you're a little bit late, which of course is very rare for you, but nonetheless, you run to class, you sit down, and you're huffing and puffing. Well, that's quite odd, because sure you were running and so on, but now you're sitting down, you're relaxed, you're content, and yet you're huffing and puffing. Now, why should you be doing that if you're not even doing any activity at that moment? Well, the reason is that when you were running, you needed energy quickly. And the body gets its energy quickly by using anaerobic respiration. It's much faster. It has fewer steps than going through the Krebs cycle, going around twice the Krebs cycle, and then in the inner membrane, going through the electron transport chain and all the steps involved there. That takes a lot of time. So though anaerobic respiration is not as efficient as aerobic respiration, which gets 36 ATP per molecule, it's much faster. So when you were running, you were able to use the anaerobic respiration, but you built up lactic acid. And building up lactic acid in muscles is not good. And if you build up too much of it, you can actually start getting cramps. So then you run, you get quick energy, you sit down, and then your body says, well, this lactic acid that you've built up, let's oxidize it back up to pyruvate, then we'll send it to acetyl coenzyme A, and then we'll send it to the Krebs cycle, and then that will be much more efficient than just sitting with our 2 ATP per molecule of glucose. So therefore, you sit down and you start breathing heavily as you're taking in extra oxygen in order to oxidize your lactic acid up to pyruvate, acetyl coenzyme A, and then going through the Krebs cycle, etc. So that is what oxygen debt is. The brain, the spinal cord, and the peripheral nervous system are composed of nerve tissue. The fundamental unit of nerve tissue is the neuron, and you're looking at a cartoon of the neuron here. I must mention, however, there are cells called glial cells, and these are supporting cells of neurons. The most important type of glial cell is called a Schwann cell. And we'll talk about Schwann cells in a few moments. To begin with, however, in terms of the anatomy of a neuron, we have here the cell body, which contains the nucleus. The cell body is also called the soma. Then we have different processes or fibers. The processes or fibers that tend to bring signals to the cell body, these are called the dendrites. the process or fiber that sends signals away from the cell body, that would be called the axon. When we go along the axon will end in little buttons. Because it terminates in little buttons, they're called terminal boutons. They are also called synaptic terminals. Now, the synaptic terminals have a little space after, and that's called the synapse. So in other words, between this cell and, say, for example, another cell that starts down here, there's going to be a little space, and that's called the synaptic cleft or the synapse. What happens is that when there is a signal going down the axon and then along here to the synaptic terminal, in the synaptic terminal there will be vesicles, little cellular containers of different types of substances. Some of these substances will be excitatory, other will be inhibitory, meaning it will inhibit the next cell or it will excite the next cell. An example of an excitatory substance is acetylcholine. An inhibitory substance is GABA. So these are chemicals that could be in vesicles that will fuse with the synaptic terminal and then diffuse across the synaptic cleft and then affect the next cell. This area, though it's called just a synaptic cleft, it has a special name when the next cell is muscle. And that special name is a motor end plate. So this is the general setup of the neuron. Now the neuron itself, as you may remember from physics in terms of discussing what capacitance is, the neuron has a separation of charges. The inner part of the neuron is relatively negatively charged, and the outer part of the neuron is relatively positively charged. So there is a membrane potential Vm, and the membrane potential is negative because the interior of the cell is relatively negatively charged. This is very important. To begin with, you could see that the plasma membrane is acting as a capacitor because it separates and stores charge. That is a definition of a capacitor. So there's a potential across the membrane. The reason why there is a difference in charge across the membrane is based on two concepts. To begin with, there's a sodium-potassium ATPase, which is the pump in the membrane. Now, the membrane of a neuron is called a plasmalemma or a neurolemma. It's the same thing as a plasma membrane. And so within the plasma membrane is this pump called the sodium-potassium ATPase. So it's an active pump. It uses ATP, it breaks it down, uses the energy in order to pump 3 sodium out and 2 potassium in. Now you may recall from general chemistry, sodium is 1 plus, potassium is 1 plus. So if you pump 3 sodium out and 2 potassium in, then you're getting a net 1 positive charge out. Therefore, if you're continually sending a net 1 positive charge out, therefore the outside of the membrane will be relatively positively charged and the inside of the membrane relatively negatively charged. Furthermore, because there's more sodium outside the membrane, more potassium inside of the membrane, and because the plasma membrane is relatively leaky to potassium, meaning that potassium will, going along its gradient, leak out of the cell, again, there is a relative movement of positive charges out of the cell, therefore leaving the inside of the cell negatively charged. Now that's quite important. Because of this resting potential of the cell, which is relatively negative, then what happens is that when the cell becomes stimulated or excited and sends a potential along the cell, which is an action potential, what happens is that the positive charges outside the membrane start to enter the cell, which means that gates open, proteins called gates open in the plasma membrane, allowing sodium to rush inside. And because sodium starts rushing inside, instead of the membrane potential being negative, suddenly it becomes positive, because so much sodium, which is positively charged, rushes inside of the cell. This produces an action potential, and this potential will send a signal along the membrane. As positive charges start going inside, it sends a signal along the membrane, and then it ends up in the synaptic terminals, or the terminal boutons, and then you can send chemicals diffusing across the cleft and then affecting the next cell. So then all these different neurons are arranged in our brain in such a way that they have different interactions with different groups of other cells. So this is the way the action potential will work along the membrane. Now there is something special that happens in peripheral nerves, and this has to do with those glial cells, which are the supporting cells. The cells called Schwann cells grow along the axons. And the Schwann cells can actually grow along the axon and then start wrapping themselves around and around. And so what happens is you end up getting these sheaths around the nerve cell or the neuron. And you get these sheaths which are caused by cells that are supporting the neuron, that are just wrapping themselves and turning themselves around and around and around the axon. This helps to insulate the axon. Because it insulates the axon, it allows the action potential to move along the axon faster. Neurons and glial cells form the basis of the nervous system. The nervous system can be divided into the central nervous system and the peripheral nervous system. As we just saw in terms of the anatomy of a neuron, we saw that the cell body can also be called the soma. Well, if you have a collection of cell bodies in the central nervous system, that collection of cell bodies is also called a nucleus. A collection of cell bodies in the peripheral nervous system is also called a ganglion. Now, the central nervous system is essentially your brain and your spinal cord. The peripheral nervous system includes the nerves outside of the brain and spinal cord. Now, the peripheral nervous system can be further subdivided into the somatic nervous system and the autonomic nervous system. The truth is that the somatic nervous system and the autonomic nervous system anatomically also form the basis of the central nervous system as well as the peripheral nervous system. But classically, it's the peripheral nervous system is divided into the somatic nervous system and the autonomic nervous system. Now, the somatic nervous system is the nervous system over which you have control. To be more specific, it is innervated with nerves that go down to the skeletal muscles. So clearly, between those nerves and the skeletal muscles, you're going to have motor end plates. So you have control over your skeletal muscles. Those are the muscles that you use to walk and move and do all kinds of activities. So you have control over that. That is your somatic nervous system. The autonomic nervous system is the nervous system that innervates things that you don't really have control over. Things like your heart rate, the size of your pupillary muscles. These often tend to be more smooth muscles or cardiac muscles, but don't tend to be skeletal muscles. Now, the autonomic nervous system can in turn be divided into two systems. The sympathetic nervous system and the parasympathetic nervous system. Now, the sympathetic nervous system is the nervous system which is often called the fight, flight, or fright response nervous system. For example, as you're sitting there learning about the MCAT exam, imagine that a bunch of raptors walked into your room. And I'm not talking about the raptors that play for the Toronto team. I'm talking about real raptors from Steven Spielberg type movies. Now, if those kind of dinosaurs walked into your room, you probably will have certain effects coming from your nervous system that would be considered to be sympathetic nervous system effects. For example, what would happen to your heart rate? You would expect your heart rate would go up significantly because your heart's going to be pumping blood quickly in order to provide the oxygen necessary for your skeletal muscles so that you can run. And your pupillary size, what would happen to that? Well, clearly, they're going to increase in size. You can just think about Bugs Bunny when he's all excited or something's happening that's shocking or nerve-wracking. You see that the eyes pop out, and it's because your pupillary size will increase in order to get as much information as possible about what is going on. In other words, looking for possible exit routes or something of that sort. So in any kind of fight, flight, or fright responses, you can consider what would happen in terms of blood flow to certain parts of your body. Obviously, the blood flow to your gut will not increase. You don't need your gut at that point. You need blood flow to your skeletal muscles. You need energy to move or to be active, etc. So those are your sympathetic nervous system responses. Then there's the parasympathetic nervous system. That is considered to be your vegetative responses. So you're just lying down, reading a book, very relaxed, nothing too much is going on. So your heart rate tends to decrease. Your pupillary size tends to decrease because you do not need action or all the information necessary in order to run or jump or do something special. And the blood flow to your skeletal muscles will tend to decrease as the blood flow to things like your gut, more smooth muscle types of things, that can increase as you start to do some digesting. And, people might say that, oh, I heard my stomach starts to grumble or whatever. And that is just, air that is passing along the gastrointestinal tract as you are beginning to digest and food and is passing through the system and blood supply is being given to the digestive tract. So this is the way the nervous system is divided. We will now examine the endocrine system. The endocrine system is the set of glands, tissues, and cells that secrete into body fluids. For the MCAT exam, essentially, the endocrine system comes down to the amount of tissues or organs that secrete hormones into the bloodstream. So hormones can be categorized into two main categories, either protein hormones or steroid hormones. Most of the hormones that we will be discussing will be protein hormones. So if I don't say anything specific, it means it's a protein. If I say something specific, then I will identify it as being a type of steroid hormone. Now, the effect that these hormones have on cells is quite different because, for example, protein hormones, because proteins are rather large and they're charged, they have great difficulty crossing the plasma membrane, as we've discussed previously. So protein hormones tend to have their effect by stimulating some sort of receptor on the plasma membrane. Then that receptor will start a domino effect. An important feature of this domino effect is a particular molecule within the cell which is called CAMP. That is cyclic AMP. Now you recall that ATP stands for adenosine triphosphate, so AMP is adenosine monophosphate. So cyclic AMP or CAMP is a very important mediator within the cell. So in other words, a protein hormone may stimulate a specific membrane receptor. That membrane receptor will go through a series of steps that will lead to increased concentration of cyclic AMP within the cell, and then this will result in a very specific interaction or effect that will eventually change protein concentrations or have some sort of effect on the cell, and so that the cell may affect other cells, etc. So that is how the protein hormone usually would work. Whereas the steroid hormone, and we know that steroid hormone has a certain structure which is fatty based on the cholesterol molecule. And so it has this fatty structure which can dissolve in the plasma membrane and therefore cross the plasma membrane. And so some steroids, because they can cross plasma membranes and cross the nuclear membrane, they can have an effect directly on the DNA. So that's a different mechanism of action than a protein hormone would have. Now, in terms of the axis, which means the levels at which the effect will occur from a hormone to the end organ, we have the hypothalamus, the pituitary, and the end organ that will be affected by the hormone. Now, it used to be thought that the pituitary was the master organ and that it controlled all the different events that were occurring in the endocrine system. But it has since been determined that the hypothalamus has many releasing factors, which means hormones released from the hypothalamus that will influence the pituitary gland and influence what types of hormones the pituitary gland will be releasing. Now, the effect of the hypothalamus and the pituitary gland functionally is part of the endocrine system, but anatomically both these organs are in the brain, and therefore they're part of the central nervous system. Now, so the effect is the releasing factor going to the pituitary gland, having an effect, making the pituitary gland want to release particular hormones. The pituitary gland releases these hormones, they circulate in the bloodstream, then they go to a particular organ where it will have a particular function. When it hits that organ may, in response, start to produce another hormone. And when that other hormone produced by the organ starts to increase in concentration in the bloodstream, it will go back to the pituitary gland and say, slow down. So, and the reason is, because could you imagine if the pituitary gland just kept stimulating the organ to produce more and more hormone, then it would be no end. The organ would just always be producing more and more and more hormone, and the effect would just get out of control. So there is a feedback mechanism so that the pituitary gland knows that the organ is actually working, it's actually doing its job, and the pituitary gland can shut down its production of the hormone which is stimulating the organ. This feedback loop is called a negative feedback loop, which means the organ will produce the hormone, tell the pituitary gland to slow down. So this is one of the ways that an end organ can regulate secretion coming from the pituitary gland. This negative feedback loop sometimes includes the hypothalamus as well. So the pituitary gland secretes a bunch of different hormones. First of all, it is divided into two parts. There is a part of the pituitary gland towards the front, that is anterior, and part towards the back, which is posterior. The anterior pituitary gland secretes all these hormones. So let's examine each of these hormones. First, we have GH. GH stands for growth hormone. So clearly, growth hormone is important in, well, growth. And so growth hormone would have its greatest effect on bone and muscle, because you started off small and now you are where you are, because growth hormone stimulated your bones and your muscles for you to grow. Next, there is TSH. TSH is thyroid stimulating hormone. So clearly, thyroid stimulating hormone goes to the thyroid gland and stimulates the thyroid gland. The thyroid gland is kind of interesting because the thyroid gland is located in your neck and it's shaped like a bow tie. So in the thyroid gland, there are a couple of hormones that are important, but the most important hormone secreted by the thyroid gland is thyroxine. Thyroxine is a hormone that contains 4 iodine atoms. And in fact, the thyroid gland is the part of your body with the highest concentration of iodine atoms. Thyroxine is secreted by the thyroid gland in order to increase your basal metabolic rate. So TSH coming from the anterior pituitary gland, going to the thyroid gland, stimulating it. Thyroxine is produced and your basal metabolic rate increases. So let's say you're a family doctor and you're in your office. Somebody comes into your office and they're all excited and hyper and all of that. Well, you might want to do some certain tests. You may want to run some drug studies. But one of the things that you may consider is doing thyroid function tests, because it might be that the person's thyroid gland is overactive and therefore they themselves are hyperactive. That's one of the things you might want to consider as thyroxine is being produced, stimulating the production of mitochondria, making more ATP being produced, and stimulating the metabolic rate of the person. On vice versa, you might have a patient come in who is very tired and exhausted and lots of fatigue all the time. And you may again want to do thyroid function tests to see if the person's thyroid gland is hypoactive or not producing enough thyroxine. Next on our list we have ACTH. ACTH is adrenocorticotropic hormone. So adrenocorticotropic hormone clearly would go to the adrenal gland. The adrenal gland is located on top of the kidney. So you have 2 kidneys, so you have 2 adrenal glands. They're rather small, and just like other parts of the body, there's an outer part called the cortex and an inner part called the medulla. We find this in many parts of the body. For example, the human brain has an outer part called the cortex and an inner part called the medulla. The kidney has an outer part called the cortex and an inner medulla. In fact, the word cortex originates from a Greek word which suggests bark. And of course, in a tree, there's the outer part, which is the cortex or bark. And it's similar for the adrenal gland, the kidney, the brain, and so on. So the adrenal gland has an outer part called the cortex, the inner part, the medulla. The medulla of the adrenal gland does not react to this axis. It reacts separately. In fact, the medulla of the adrenal gland is considered to be part of the sympathetic nervous system. The medulla of the adrenal gland secretes adrenaline, or what we call adrenaline. So it's coming from the adrenal gland, adrenaline. Some people call adrenaline epinephrine. There is a related hormone, noradrenaline. These hormones increase the fight, flight, or fright response. So the heart rate, respiratory rate, etc. The cortex of the adrenal gland is what is being stimulated by adrenocorticotropic hormone. Now, the adrenal cortex secretes 3 categories of important steroid hormones. It secretes glucocorticoids, mineralocorticoids, and the sex hormones. The sex hormones include male hormones such as testosterone and female hormones such as estrogen. The glucocorticoids, consider the word. It ends in oids, so you know it's a steroid. It starts with gluco, so if you had to think, what in the body would it increase or have a great effect on? It's going to be glucose. So glucocorticoids increase the concentration of glucose in the body. It's called glucocorticoids because it's the adrenal cortex. And examples of adrenal hormones that increase the concentration of glucose in the blood include cortisone and cortisol. So these increase the concentration of glucose in the blood. Then you have the mineralocorticoids, which increase the concentration of minerals in the blood, specifically sodium. Aldosterone is an example, a very important example, of a mineralocorticoid, increases the concentration of sodium in the blood. By doing so, by bringing sodium back into the bloodstream, water will come into the bloodstream by osmosis. So you increase the volume of fluid in the bloodstream, therefore increasing blood pressure. So you can imagine, in response to stress, blood loss, or fluid loss, such as a runner who's doing a lot of activities, you can imagine that ACTH is going to be increased coming from the anterior pituitary gland, stimulating the adrenal cortex in different ways, different effects on the different hormones, but essentially you end up getting more glucose liberated, and you get sodium being pulled back and water being pulled back into the bloodstream. So that's ACTH. Then there is prolactin. Lactin suggesting lactate. And to lactate is to provide milk. And so prolactin is a hormone important in breast development and milk production. In fact, I once had a patient in his 30s presenting to the office, very shy as you can imagine, with a history of breast development and milk production. And in fact, he demonstrated to me that he was producing milk from his breasts, which were very developed. And so, without the help of a CT scan nor an MRI, you would be able to diagnose, as I did, that clearly there must be a tumor in the anterior pituitary gland which is overproducing prolactin. And indeed that was the case. The next hormone, FSH, which is follicle stimulating hormone. So in women, follicle stimulating hormone is secreted to stimulate follicles, and we will look at that in greater detail later. And in men, FSH is important in spermatogenesis. Some students like to remember the S in FSH standing for spermatogenesis. There's LH, which is luteinizing hormone, and we will look at its effect on the ovary and the menstrual cycle shortly. But in males, LH, some people like to remember that the L stands for Leydig, since the LH hormone is secreted by the anterior pituitary gland and stimulates Leydig cells in males, which in turn produces testosterone. So these are all the hormones of the anterior pituitary gland. The posterior pituitary gland is a little bit different. The reason is that the hypothalamus actually produces the hormones in the posterior pituitary gland. It produces it, releases it in the posterior pituitary gland, and then from the posterior pituitary gland, these hormones are released into general circulation. So the hormones coming from the posterior pituitary gland include oxytocin and vasopressin. Oxytocin has an effect on smooth muscle cells. It has an effect on smooth muscle cells of the breast to help contract them, which is important in milk letdown. It also has an effect on the uterus to help contract the uterus, which is important in delivery of the baby. Oxytocin is a hormone that we sometimes give to women when we do cesarean sections. Because when we do a cesarean section, after we remove the baby, then sometimes there could be a lot of bleeding of the uterus once we've opened the uterus to remove the baby. To help reduce the bleeding, giving oxytocin will contract the smooth muscle of the uterus, thus closing down many or most of the blood vessels. Then there is vasopressin, and the word suggests what it does. Vaso, referring to blood vessel, and pressin, referring to pressure. So vasopressin increases blood pressure. Vasopressin has another name, ADH, anti-diuretic hormone. Now, a diuretic is something that will increase urine flow. Anti-diuretic does the opposite. So ADH, or vasopressin, will bring fluid back into the body, thus reducing the amount of urine produced. So we're going to look at where ADH acts later. Special mention must be made of some hormones which are not part of the hypothalamus pituitary organ axis. For example, in the thyroid gland, there are a group of cells that secrete a hormone called calcitonin. Calcitonin decreases the concentration of calcium in the blood. Now, calcium regulation is intimately associated with cells that work on bone, because clearly in our body the greatest deposit of calcium is going to be in our bone. So we have cells that create bone, osteoblasts. Clearly, if you create bone by putting calcium into the bone, you're going to decrease the concentration of calcium in the blood. So clearly, calcitonin would stimulate osteoblasts. And there are cells called osteoclasts. Osteoclasts are cells that will resorb bone, which means it will take apart bone, thus releasing calcium into the blood, therefore increasing the concentration of calcium in the blood. So calcitonin decreases the concentration. There are 4 glands that are around the thyroid gland. They are therefore called parathyroid glands. Now, when we operate on the thyroid gland, we have to be very careful to identify these parathyroid glands and preserve them. So these parathyroid glands secrete a hormone called parathyroid hormone. It's pretty easy to remember. It's also called parathormone, or PTH. Now, parathyroid hormone, or PTH, has the opposite effect of calcitonin, meaning PTH will increase the concentration of calcium in the blood. Because it increases the concentration of calcium in the blood, it will act on osteoclasts. In the pancreas, we have several hormones that are being produced in a particular part of the pancreas called the islets of Langerhans. In the islets of Langerhans, there are cells that are called alpha cells and beta cells. Alpha cells contain a hormone, glucagon, which increases the concentration of glucose in the blood. And in your body, in fact, there are many hormones that increase the concentration of glucose in your blood. So if one of these hormones happen to be missing, you'll get away with it. You'll survive. You'll be just fine, in fact. However, there's only one hormone which will decrease the concentration of glucose in the blood. Just one. And because there's just one, if something happens to that hormone, because there's no redundancy in our system, then we end up with disease. And of course, this hormone, which is located in the beta cells in the islets of Langerhans, was discovered in the land of the Toronto Raptor, and this hormone is called insulin. So insulin is the hormone that will decrease the concentration of glucose in the blood. It has many effects, but one of its effects is to actually help glucose go from extracellular to intracellular. And as you already know, through glycolysis, once it gets in the cell, it is phosphorylated, and then it goes through the rigmarole. So insulin decreases the concentration of glucose in the blood. We will now look at the menstrual cycle, keeping in mind the influence that the anterior pituitary gland has on the main organs of the menstrual cycle, the ovaries and the uterus. The menstrual cycle is a 28-day cycle, essentially. The highlights of the menstrual cycle include the first 5 days, which is menses, or menstrual blood flow, and day 14, which is ovulation. Ovulation is when the ovum, or egg, is released from the ovary. So the menstrual cycle includes several very important hormones from the endocrine system. To begin with, if you remember that in the ovary, the first 14 days is considered the follicular phase, the first 14 days in the uterus is considered the proliferative phase, then the sequence of secretion of the hormones become quite easily. There is also in the second 14 days, for the ovary it is called the luteal phase, and for the uterus it is called the secretory phase. So these parts need to be memorized, then the hormones and the action of the hormones become quite easy. In fact, for the MCAT exam, they could ask you the action of a particular hormone, or they can actually show you the graph of the hormones, point to one of the curves, and ask you which hormone does this represent. So to begin with, the ovary is the follicular phase. So consider what hormone coming from the anterior pituitary gland would be the most important hormone in the follicular phase. Clearly, it would be FSH, which is the follicle stimulating hormone. So in terms of the first 14 days of the cycle, the most important hormone coming from the anterior pituitary gland would be FSH. So we would expect there to be some sort of curve demonstrating FSH increasing in concentration. So FSH is the most important part. So whenever you see a curve that goes up in the first 14 days but doesn't do anything special in the next 14 days, that must represent FSH. So FSH would go up and then come back down in the first 14 days. So FSH, follicle stimulating hormone, secreted by the anterior pituitary gland, goes through the blood system, goes to the ovary to stimulate follicles. So it stimulates follicles, which are groups of cells within the ovary. And these follicles start to grow, and then one follicle becomes the number one follicle, or the primordial follicle. This follicle is the one that will be secreting high levels of estrogen. So estrogen starts to be secreted in response to FSH, estrogen coming from the ovary. So now another hormone that we start seeing going in high quantity as a result of FSH is going to be estrogen, which I'll just symbolize as a large E. So estrogen is now coming from the follicle in the ovary. What does estrogen do? Well, estrogen goes throughout the body as a hormone. It is a steroid hormone, and then it goes to the uterus and leads to proliferation. The inner layer of the uterus is called the endometrium. Endo referring to inner, and metrium referring to the smooth muscle of the uterus. So the inner portion of the uterus is called the endometrium. The proliferative phase refers to the fact that estrogen will lead to the proliferation of this inner layer called the endometrium. So it becomes thicker and thicker and thicker in response to estrogen. So now we have the estrogen going up, we have FSH, which rose initially, and then we're coming towards ovulation. Now we know that the next phase will be the luteal phase. So clearly, what would be the most important initial hormone coming from the anterior pituitary gland for the luteal phase? It would be luteinizing hormone, or LH. So LH actually comes in just before ovulation as a surge. So when you see a hormone that goes up like that, just peaks all of a sudden just before day 14, that is called the LH surge. So right away, no matter what the other graphs are doing, you know that the one that does the big peak just before ovulation is the LH. And it is felt that ovulation occurs in response to the LH surge, or luteinizing hormone, coming again from the anterior pituitary gland. So this is clearly a very important hormone which starts the next stage. And then in the next stage, after it does its peak, luteinizing hormone again will start to go up a little bit in the next phase, the luteal phase for the ovary. Now, what is actually occurring? So we had a follicle, it grew. It was producing estrogen, and then at day 14, in response to LH, the follicle releases the egg or ovum. That's ovulation. Now the follicle which remains no longer has the egg. It involutes, turns inwards, and becomes yellow. And coming from the Latin root for a yellow body, we get luteal body. And a body just means, and again, a cluster of cells. So the luteal body is formed in the ovary. And so literally, if you were to dissect and look inside of the ovary, you would see that it's a very yellow cluster of cells. This yellow cluster of cells will now secrete estrogen and progesterone. Again, two steroid hormones coming from the luteal body. Now we know what estrogen does. It maintains the thickness of the endometrium. It makes it thicker and thicker. So that's what estrogen does. So we know that in response to LH, we know that estrogen went up in the first, and again it comes back down, but then it goes back up. So the only hormone that goes up in the first phase, goes back down, then goes back up in the second phase. The only hormone that does that, estrogen. Then we've got progesterone also going up. So progesterone is the hormone which is not up at all in the first phase. Then in the second phase goes up a lot, just like estrogen goes up a lot. So these are the unique features of the movements of these types of hormones. Now, estrogen thickens the lining. Progesterone leads to secretion. So progesterone goes to the lining of the uterus, the inner lining called the endometrium, and helps it secrete fluids. That's very important because the egg has just been released, so there's a possibility that there could be fertilization, and if there's fertilization of the egg where the sperm and the egg meet, we want implantation in the endometrium, and in order to have implantation, the endometrium must not just be thick, but it also has to have the proper fluids on the endometrium to help the implantation occur. So these fluids are being secreted in response to progesterone. So we have estrogen and progesterone going up. Now, if there is fertilization in this part of the menstrual cycle, then the fertilized egg will start to secrete hormones so that estrogen and progesterone can remain elevated. So that the uterus continues to get thicker and thicker and there's fluids being secreted and so on. So this will continue in response to a fertilized egg. But if the egg is not fertilized and estrogen and progesterone are high, but there's no fertilized egg, then LH will taper off. We already know FSH is no longer high because it was in the first phase. That was the follicular phase. So FSH is not involved anymore from the anterior pituitary gland. LH drops off because there's no interest in it remaining high. There was no fertilized egg. So in response to the fact that the anterior pituitary gland no longer has important hormones involved in this action, the concentrations of estrogen and progesterone start to taper off. So imagine, these were the hormones that thickened the uterine lining, kept it thick, made sure it was well vascularized, fluids coming in and so on. And all of a sudden, these hormones are withdrawn. No more hormones that are keeping the uterine lining thick. So then, after day 28, we restart the cycle at day 0, the menstrual flow. In other words, in response to the withdrawal of estrogen, which was maintaining the thickness of the uterine lining sloughs off. It just falls off. And because of that, there is the bleeding of the first 5 days of the menstrual cycle. So it's in response to estrogen withdrawal. So you've graduated from medical school and you've decided that you want to become an obstetrician or a pediatrician. You've followed a couple through a pregnancy. They deliver a beautiful baby girl and they go home the same day. At 3 o'clock in the morning, they call you frantically, and of course you answer the phone because you're available to your patients, because you're just like that, and they call you frantically saying that their baby that they just brought home is bleeding from her vagina. Well, what would you say? Would you call an ambulance, or would you just comfort them? You may want to pause and consider your response. Well, after reviewing the menstrual cycle, you could imagine that in pregnancy, because the uterus will go from something that's about this size to something that is about that size, the uterus is clearly responding to massive quantities of estrogen and progesterone. And we know that estrogen and progesterone, being steroid hormones, they tend to be able to cross barriers. For example, the barrier between the mother and the infant is the placenta. The placenta produces nutrients, it can store material, and in fact the placenta even produces estrogen and progesterone. So all these hormones from the mother and from the placenta, some of this is being exposed to the infant. So here's the infant, female, with the uterine lining that is becoming thick in response to all this estrogen exposure. Then the baby is born. The baby doesn't have high levels of FSH or LH, of course not, because the baby's not going to have menstrual cycles for years, on average not until year 12. So here's the baby, after having all this estrogen exposure, now she has estrogen withdrawal. And in response to withdrawal, the inner lining or endometrium will slough off or fall off, and therefore menstrual blood flow in response to being born. And so you would be able to comfort the parents and say this is very common. And the only reason that sometimes it doesn't occur is sometimes the flow is very low, or sometimes there is a barrier for the flow to come from the vagina, like the tissue which is called the hymen, and sometimes it might be completely serving as a barrier, and then the body will slowly resorb the blood that is in the vagina. You are only allowed one guess as to which organ this represents. Clearly it's the heart. We're talking about the circulatory system now. And the heart is a muscular organ. It is about the size of your fist. The heart is divided into left and right sides by a septum that runs down pretty much the center of the heart. So that's the septum, which divides the left and right sides of the heart. Now, when we talk about left and right sides, it's always the patient's point of view. For example, it is when you are looking at me, your left is over here, your right is over here. However, you have to consider what is the side for the patient. That's what is always being discussed. So therefore, this is the left side of the patient who is looking at you, and this is the right side of the patient. So this is the left side of the heart, right side of the heart. Now, there are actually 4 chambers in the heart. So it is again subdivided, and there are the upper parts of the heart, which are called the atria, which means there is an atrium on the right side and an atrium on the left side. The bottom parts are called the ventricles. So there is a right ventricle and there is a left ventricle. So let's see what happens to blood as it leaves the heart and leaving, let's say, from the left ventricle. So blood leaving the heart from the left ventricle. To begin with, blood always leaves the heart in arteries. Some students like to remember, away starts with A, away in arteries. So it's always like that, and so blood will be returning in the veins. So going away from the heart, from the left ventricle, will be the largest artery in the body, which is called the aorta. So we leave the body from the left ventricle in the aorta. So the aorta will subdivide into smaller arteries. And those smaller arteries will again subdivide into even smaller arteries called arterioles. These smaller arteries or arterioles will then subdivide further into capillary beds. Now, capillaries are tiny, very thin-walled vessels. You have to imagine your entire body has to have some sort of exposure to blood in order to have some diffusion of carbon dioxide, the waste product, which we've seen already, coming from the Krebs cycle and aerobic respiration. And so you need to get rid of that, and also there's exchange of water, there's a need for oxygen and glucose, etc. So you have to have some area in which there's a diffusion taking place. That occurs at the level of these capillary beds, which are a rich network of vessels. They're at the tip of your finger, they're in your heart, they're in your left knee, they're all over your body in order to deliver this exchange of materials. So this is occurring at the level of the capillary network. So this is all oxygenated blood in arteries. And then we have this exchange going on. The blood is now losing oxygen. It's picking up carbon dioxide, one of the waste products. And so we've gone from artery, huge artery, to many arteries, to many, many more arterioles, to this massive network, which is the capillary network or capillary beds. Now we start getting into deoxygenated blood. So this blood is now deoxygenated, which I'll symbolize as blue. It's not really blue. When you look at your arm and you see a vein, it might look blue to you, but it's actually an optical illusion because of the shape of the vessel and light, etc. Because if you actually were to cut it, and I'm not suggesting that experiment whatsoever, but as surgeons we see it all the time, and it's clearly not blue. So here's the deoxygenated blood, which we're only symbolizing being blue. So when it comes out of the capillary network, it now goes into tiny, tiny, tiny veins, which are venules. And then from these tiny veins, it goes into larger veins, and then eventually goes into one of the 2 largest veins in the body, which are called the venae cava. These venae cava will bring deoxygenated blood into the right atrium. There's 2 venae cava. There's one for the inferior or lower part of the body, which is called the inferior vena cava, and there's a part for the upper part of the body, which is called the superior vena cava. These both bring deoxygenated blood into the right atrium. When the blood gets into the right atrium, it is pumped into the right ventricle. From the right ventricle, blood is pumped into the lungs. So it's going away from the lungs away from the heart, and because it's moving away from the heart, it's moving in this direction. Away from the heart means that it's in an artery. So it's going away to the lungs. The word pulmonary refers to the lungs, so it's going away in pulmonary arteries. So it'll go away in the pulmonary arteries. Then it'll go into pulmonary arterioles. Then it'll go into these capillary beds in the lung. And there it will have an exchange of gases in the small parts of the lung where the alveoli are, and that's where there will be an exchange of gases where it will release carbon dioxide, our waste product of cellular metabolism, which we blow off in the lungs, and then we bring in oxygen, and that oxygen is going to be exchanged at the level of the capillary beds in the lung. Now notice, this blood coming from the right ventricle was deoxygenated. It lost oxygen in the capillary beds in the body system. So you cannot say that all arteries carry oxygenated blood. It would not be true. Blood going into the body system, that is oxygenated in an artery. But blood going to the pulmonary system is deoxygenated in pulmonary arteries. So just because it's an artery doesn't tell you if it's oxygenated or deoxygenated. It depends on where that artery is going to. So in the lung, in the capillary beds, we get oxygenation in the lung. So then we have those capillary beds now becoming oxygenated, then going to venules and veins and returning to the left side of the heart in pulmonary veins. So coming to the left side of the heart in the pulmonary veins. And then coming to the left atrium, it's oxygenated blood, and then the left atrium will pump into the left ventricle, and then the left ventricle will again pump into the body system. So notice a few things. First of all, it's as if there are two pumps here. There's a pump that goes to the pulmonary system, and there's a pump that moves towards the body system. So that's why it is sometimes considered that we have two hearts, because there are two pumps that are acting in parallel. The second thing is, because there are two pumps acting in parallel, there are two systems of circulation. There's circulation in the lung, which is called the pulmonary circulation. And there's circulation in the body system, which means to your knee, your leg, your left elbow, all the different parts of your body, your brain, and so on. And that's called systemic circulation. So there's pulmonary circulation, and then there's systemic circulation. So that's one thing. The other thing is that when you're going from the atrium to the ventricle, there are valves. And these valves are very important. On the right side of the heart, there are valves that have three cusps, three sort of leaflets, 1, 2, and a third one. So it's called a tricuspid valve. On the left side of the heart, there's a valve that has two of these cusps, and therefore it's called bicuspid. It is also called the mitral valve. That is the valve on the left side of the heart. That's between the atrium and the ventricle. Now you have to imagine these valves. They have cusps. So they're sort of shaped like this. And if you could imagine that my head is inside of the atrium and blood is coming downwards, what we would see, just imagine me blowing from above. If I were to blow, I'd go, and then it would open up and allow fluid to move from the atrium into the ventricle. Now, when the ventricle were to contract, just because of the shape of these valves, when the ventricle would contract and fluid would be coming from the ventricle, boom, the valve would close, preventing blood from going back into the atrium. So therefore, these are one-way valves. Blood can flow from the atrium into the ventricle, like so, but when the ventricle contracts, then because of the shape, the valve would close back, forcing blood not back into the atrium, but forcing blood into the pulmonary circulation. And when blood comes into the left atrium, pushed into the ventricle, when the ventricle contracts, then blood will go into the aorta and not back into the atrium. In fact, if there was a problem with one of the valves in your heart, then you would hear a heart murmur, which is an abnormal sound between the normal heart sounds. Now, the arteries in the body all have muscular walls. And so, because of these muscular walls, they can be affected to maintain a certain blood pressure. And we can calculate this blood pressure or hydrostatic pressure. And it is such that when the ventricles contract, you're going to have the highest blood pressure. And then when the ventricles relax, you have the lower blood pressure. The largest, most muscular chamber in the heart is the left ventricle. And the reason for this is that it must pump blood to the whole body system, except, of course, to most of the areas of the lung. It doesn't have to send blood to most of the areas of the lung because that happens in pulmonary circulation. But it does have to send blood to the entire blood system, body system, and therefore it has to be very large and it contracts. So when it does contract, we can calculate the blood pressure, and average blood pressure for humans is 120/80. It's measured in millimeters of mercury. The 120 represents the blood pressure when the ventricle contracts, and that is called the systolic blood pressure. The 80 represents the blood pressure in millimeters of mercury when the ventricle relaxes, and that is called the diastolic blood pressure. Clearly, the fluid that circulates in the circulatory system is blood. Blood has two main components, plasma and formed elements. Plasma represents about 55% of blood, formed elements 45%. Plasma is essentially water with some very important proteins. There are three main categories. One is albumin. Albumin is a protein that can carry other molecules in the blood system, or albumin also serves to maintain osmotic pressure in the blood vessels. There's also fibrinogen. Fibrinogen is a molecule which is converted into fibrin when necessary, and fibrin is a very important component of blood clots. And thirdly, there is a group of proteins called the globulins, and the most important part of this group of proteins are antibodies, and we'll be discussing antibodies a little bit later. So that's plasma. Then there are the formed elements. The formed elements, representing 45% of blood volume, includes red blood cells. Now, red blood cells is 99% of the formed elements, so it's almost all red blood cells. And of course, we know the red blood cells are extremely important in terms of the transportation of oxygen and carbon dioxide. Now, red blood cells are biconcave cells, and they contain hemoglobin. Hemoglobin is a molecule that has two parts, the heme and the globin part. The heme has iron in it. Globin is a protein. So hemoglobin is very important in the red blood cell. As the red blood cells pass through the circulatory system, go into those capillaries in pulmonary circulation, and that is where diffusion takes place. The red blood cells release the carbon dioxide and take in oxygen. Oxygen binds to the hemoglobin molecules in the red blood cell and becomes this sort of bright red. And so it is oxyhemoglobin which is responsible for that bright red color. And then it goes through the system and then into the capillaries and systemic circulation where oxygen is given off to the cells and carbon dioxide is taken up. Carbon dioxide bonds to the hemoglobin molecule, and creates carbaminohemoglobin, which is darker. Not blue, but it is darker than the bright red color. In muscle cells, the bright red color is from another molecule, which is very similar to hemoglobin, but it is called myoglobin. And words that have myo in it often refer to muscle. So those are the red blood cells. Then there are platelets. Platelets are cytoplasmic fragments from bigger cells. And these fragments are very important, again, like fibrin, in terms of creating blood clots. Then there are the white blood cells. And white blood cells are critical for fighting off infection. There are two main categories of white blood cells. There are the granulocytes and agranulocytes. Now, whenever you hear the word cyte, just like in cytoplasm, which means plasm in the cell, or cytosol, which is solution in the cell, cyto refers to cell. So a granulocyte is a cell that has granules, and granules are just packets of stuff. And so if you were to look at a granulocyte under a microscope, you just see all these packets of stuff. You look at an agranulocyte, those same packets aren't there, but you have all the other organelles, Golgi apparatus, endoplasmic reticulum, nuclei, stuff like that. So in the granulocytes, there are three types of cells. They are the neutrophils, the basophils, and the eosinophils. The neutrophils are the dominant component of pus. So clearly a very important part of the body's reaction to infection. Then there are agranulocytes, and the agranulocytes are mostly divided into lymphocytes and monocytes. Lymphocytes are a type of cell that we're going to discuss in greater detail when we look at the immune system and the lymphatic system. But the lymphocytes are indeed part of blood. Monocytes are cells that have various names. They are indeed white blood cells. They are also called macrophages, and they are called this when they're outside of the circulatory system. They may be called a macrophage or a phagocyte. Now, a phagocyte is a cell, just like phagocytosis, it's a cell that eats other cells. Phagocytosis literally means cell eating. So a monocyte is in the circulatory system, and it is a cell that can eat other cells, specifically bacteria and cells that are causing infection. Of course, it can also attack viruses, but we know that viruses are not really cells. We will now look at the immune system, but first we will discuss lymph, because lymph is intimately associated with the immune system. Just like blood is the fluid that circulates in the cardiovascular system, which we often call the circulatory system, lymph is the fluid that circulates in the lymphatic system. The lymphatic system has a bunch of vessels in the body, and these vessels, or that contain lymph, is concerned with the third space in your body. You see, there are three places where fluid can be in your body: intracellular, which we've seen; intravascular, which we've also seen; or fluid could be in neither the intracellular or intravascular space. That's called the third space. It's also called the extracellular space. It is also called the interstitial space. So lymph will bathe these cells. Lymph contains clearly lots of water, some nutrients, and some lymphocytes, so cells that circulate in the lymphatic tissue. These lymphocytes are divided into both T cells and B cells. Now, in the lymphatic system, you have different elements. For example, you have lymph nodes. And lymph nodes will have a high concentration of these white blood cells, which will help to fight off infection. Lymph nodes are sort of pea-sized, and you can feel them or palpate them. You can often feel them in your neck or sort of under your chin. You can also feel them in the groin, and you'll feel these pea-sized shaped organs, and these are your lymph nodes that are hooked up to your lymphatic system. So you have these little lymphatic capillaries all around your body, going up to larger lymphatic vessels that usually draw lymph to the lymph nodes so that infection and so on can be taken care of. And then eventually this lymphatic fluid will return to the cardiovascular system. There are other parts of the lymphatic system, including the spleen. And the spleen is an organ that is found on the left side under the ribs. And this organ sort of has two parts to it, a white pulp and a red pulp. The red pulp is not part of the immune system, it's red, and it sort of stores blood, red blood cells. The spleen is sometimes called a blood-storing organ. The white part is part of the immune system, and this white pulp kind of filters infection and fights off infection, and so it's a very important part of the immune system. So that's the spleen. And then there is the thymus gland, which is in the upper part of the chest, and the thymus helps to mature T cells. The T from thymus, T cells. In fact, the thymus secretes another hormone, believe it or not, called thymosin, and thymosin is felt to stimulate T cells. Now, T cells and B cells both originate in the bone marrow. So you can think of the B being in B cells being for bone marrow, even though it actually stands for bursa in birds, but that has nothing to do with the MCAT. But the B cell, you can think it meaning coming from the bone marrow. T cells also come from the bone marrow, but they're matured in the thymus gland, so they're called T cells. Now, T cells are an important element of what is called cellular immunity. Cellular immunity is a type of non-specific immunity in which the T cells will attack the infection. So whatever infection that is going on, there's bacteria that's in your body or there's a virus in your body, then for sure you're going to have neutrophils, which are on the scene very early. You have monocytes or macrophages or these phagocytes, which are attacking this bacteria and virus. You'll also have T cells, which will go on the attack, and so they phagocytize. They will eat that bacteria, bring it into the cell, and the lysosomes will fuse with it and break it apart, and the body will use the parts for other purposes. So this is all part of the attack against infection using the T cells. But there are other types of T cells. Some cells are called T4 cells or T helper cells. These T helper cells will stimulate other cells to help in the attack. So it will stimulate other T cells. It'll also stimulate B cells. It'll also stimulate macrophages and other types of cells. So these T helper cells are very important. Unfortunately, T helper cells are specifically targeted by the HIV virus, and that leads to severe immunodeficiency. Nonetheless, T cells can activate, with the T helper cell, the B cells to produce these new type of cells called plasma cells. You see, B cells are cells that are involved not in cell-mediated immunity like T cells, where a cell attacks directly. B cells are involved in humoral immunity, which is very specific to the type of infection. It's not just that a cell will arrive and just eat any problem that is in front of it. That's non-specific, T cells. B cells is very specific, and the way it is so specific is incredibly fascinating. What will happen with the B cell is that a macrophage will go find the infection, attack, phagocytize, bring in the bacteria or virus. Then it will take a part of the membrane or the outer part of the virus, and it will bring it to its surface of the macrophage. And the macrophage will call the B cell over by sending chemicals up. It will call the B cell over and it'll say, look, don't attack me because I'm one of the white blood cells, just like you are. But listen, I've just removed part of something which is attacking our body. I want you to look at this, and I want you to create a weapon specifically targeting this part of that bacteria or virus. So that part of the bacteria or virus is called an antigen. Now the B cell will create something to specifically attack that antigen. So it's actually going to make a brand new protein, a protein that's never been seen in your body before. And it will create that protein to specifically attack that antigen. The protein that the B cell will create would be called an antibody. And this antibody is also sometimes called immunoglobulin. This antibody, designed specifically to attack part of the membrane or outer part of the virus or bacteria or fungus or whatever else is attacking the body, will be able to destroy that cell in a very specific way, many different types of fashions. For example, it can destroy the cell just by the fact that the antibody hits that specific part. That cell might be destroyed by lysis or explosion. That happens, not too often, but that could happen. But sometimes it will attack those cells and clump them together. That's called agglutination. Sometimes it will attack the cell and make the cell much more desirable for other white blood cells to attack, like T cells, macrophages, neutrophils. That idea that you're attacking a cell and making it much more desirable for other cells to attack, that's called opsonization. So there's many different ways in which B cells, by creating antibodies, can specifically destroy cells. So that's called the humoral response, which is a specific response to infection because the B cell actually constructed an antibody aimed for a specific type of antigen. When the B cells start to multiply, they will create some memory cells. So that if ever you get that infection again, there's some cells that already have the antibody ready to solve the problem. So that you don't have to start from scratch where the B cell's spending time trying to design a protein to attack the antigen. Rather, you've got some memory cells ready, so your secondary response, the second time you're exposed to it, will be much faster than the first time you were exposed to the infection, your primary response. The B cell will also create lots of plasma cells. Plasma cells are B cells that are just dividing and dividing because they're sending off antibodies. They're producing much as possible these weapons to attack the antigen. So these are called plasma cells that are rapidly dividing, and the T helper cells help the B cells in order to rapidly divide and make more plasma cells. And this will attack, again, the source of infection. You are looking at a very diagrammatic view of the digestive system. This is the person's head, mouth, esophagus, stomach, small intestine, and colon. The digestive system is also called the gastrointestinal system, or the GI tract. It is also called the alimentary canal. The digestive system begins in the mouth. So you take a chocolate bar or something, you put it in your mouth, that's called ingestion. Then you start chewing on it, that's fragmentation, helped of course by your teeth. And then you have some things that are being secreted into your mouth. There's saliva and there's mucus. The mucus helps to roll up the food into a bolus. The saliva contains an enzyme called salivary amylase. Salivary amylase is the first level of digestion because digestion specifically refers to chemical breakdown. So salivary amylase is an enzyme that's secreted in the mouth by the saliva, and it helps to break down carbohydrates into their subunits. So that's the beginning of digestion. Then this food particle is a bolus, it's called a bolus, and it is placed at the back of the mouth as the tongue rolls it towards the back, into the pharynx, and down the esophagus. Now, in order for the food to actually go down the esophagus and not to go down the windpipe, which is actually the trachea, because we don't want it going into the lung, we want it going into the gastrointestinal tract, there's a little flap of tissue called the epiglottis, which will cover the glottis. The epiglottis is around the glottis. So it covers the glottis, preventing food from going down into the respiratory tract and making sure that it continues along the alimentary tract. Now we have food in the esophagus. Now the food in the esophagus will go down into the stomach, well, in part because of gravity, but not necessarily just gravity, because could you imagine peanut butter would always get stuck if it just was relying on gravity. So the alimentary tract also has another thing that helps to move things along, and that's peristalsis. Peristalsis is a concentric movement of muscle in which it is squeezed and pushes things along the alimentary tract. That occurs anywhere along the GI tract where you can have some peristalsis, not just in the esophagus, but you also have it in the small bowel and the colon to move contents through the GI tract. So then we have the contents move through the GI, through the upper part of the GI tract, the esophagus, into the stomach. Now, in the stomach, we have a lot of churning motions that occur in the stomach. We also have in the stomach a very low pH. In other words, the concentration of hydrogen ion in the stomach is very high. The pH is about 2. The pH in the stomach is low for several reasons. To begin with, the first thing is that by having a low pH in the stomach, that helps to hydrolyze food, means to break it apart. Second reason is that there is an enzyme in the stomach called pepsinogen. Pepsinogen is converted into pepsin in a low pH environment. So when pepsinogen is turned into pepsin, pepsin can break down proteins. So in other words, this is another level of digestion in the digestive tract. Pepsin in the stomach breaking down proteins into their subunits, which we know, of course, are amino acids. So that's the second reason. The third reason that the pH is low in the stomach is to destroy bacteria. Could you imagine? Dogs must have a very low pH in their stomach because you know dogs' tongues have been in many different places. So you can imagine that the pH of the stomach is low so that if there are bacteria or other things that shouldn't be in the body that happen to get into the stomach, these things can be destroyed in the environment of a very low pH. So this is what occurs in the stomach. This churning and so on that occurs in the stomach produces this fluid called chyme, C-H-Y-M-E. So chyme is produced in the stomach, and then this muscular stomach squirts the chyme into the small intestine. The small intestine is divided into 3 parts: the duodenum, the jejunum, and the ileum. And it is in that order. The small intestine is very long. And in fact, the entire GI tract, from stem to stern, is about 30 feet long, or about 9 meters long. The small intestine has a lot of surface area, and it increases its surface area in order to absorb all the products like amino acids and breakdown products of carbohydrates and so on. And it increases its surface area with villi and microvilli. Villi are undulations or finger-like projections of groups of cells, so in order to increase the surface area. Microvilli are finger-like projections of single cells, so that increases the surface area of individual cells. And we know that microvilli have microtubules as a core, just like flagella. Now, we've already looked at the endocrine system, and we said that that was the bunch of glands and tissues, cells, organs, which secrete their products into body fluids. For example, hormones into the bloodstream. So it goes around in the body and then it affects certain organs. However, if there is a gland or tissue that secretes its products to the outside of the body, rather than being called endocrine, it's called exocrine because the product exits the body, so it's an exocrine organ. For example, in the skin, there are glands that secrete their products onto the skin in order to make the skin supple or smooth or oily or whatever. And these products are called exocrine glands. Likewise, there are products that secrete into the GI tract. These products are called exocrine products, and the glands are exocrine glands. It is considered so because the GI tract is exposed to the outside and it releases product to the outside as well. So it's called exocrine organs. There are two important exocrine organs for the GI tract, is the pancreas and the liver. We've already seen the endocrine part of the pancreas in that it secretes insulin and glucagon, and these are important hormones. But the pancreas also secretes pancreatic juice, and this pancreatic juice contains many different important enzymes, which are exocrine products, which are secreted into the duodenum via a special duct called the common bile duct. And this pancreatic juice contains many enzymes such as nucleases, which break down nucleic acids into their subunits. And it also secretes things called pancreatic amylase, does the same thing as salivary amylase, breaks down carbohydrates into subunits. It also secretes lipase, which breaks down fats. And it secretes many different enzymes that breaks down proteins in specific ways, like trypsin, chymotrypsin, carboxypeptidase. So these are all products secreted by the pancreas in response to the fact that you ate a chocolate bar. And so what happens is that there are some enzymes like secretin and CCK, which these are hormones secreted by the duodenum, which will go to the pancreas and say, time for pancreatic juice because here comes some chyme from the stomach. Pancreatic juice is also high in bicarbonate. And the reason is that if the fluid from the stomach just was squirted into the duodenum with that low pH in the stomach, a pH of 2, and if that was happening all day long, then everybody's going to start developing ulcers because the acid will start eating away at the duodenum. So the pancreas secretes bicarbonate, and bicarbonate will react with the acid and help to neutralize the acid. So that all is part of the pancreatic juice. There's also the liver. The liver will produce bile from liver cells. Liver cells are called hepatocytes. So the liver will produce bile. Bile is stored and concentrated in the gallbladder. The gallbladder will then squirt the bile down the ducts, eventually the common bile duct, just like pancreatic juice. This enters the duodenum. Now, bile emulsifies fat, which means it increases the surface area of the fat so that the enzymes that came from the pancreas, like lipase, can start to break down the fat into its subunits. So we have enzymes breaking down into subunits. We have bile emulsifying the fat. Bile also helps in the absorption of fat-soluble vitamins like A, D, E, and K. Those are the fat-soluble vitamins: vitamin A, vitamin D, E, and K. Some students just remember ADEC are the 4 fat-soluble vitamins. So when all this is occurring and this activity is occurring and you're digesting these particular molecules, then the small bowel will start to absorb them. So it absorbs the amino acids, it absorbs the nucleic acids, and so on. And the way that it will absorb it is because there are networks of capillaries around the small bowel which will absorb these nutrients and send them along a special vein called a portal vein, along the special portal vein to the liver. So if it has to do with the liver, it's called hepatic, like a hepatocyte is a liver cell. So this special vein going to the liver is called the hepatic portal vein. So it goes to the liver and then to another series of capillaries so that the liver can process these nutrients, can detoxify anything that might be toxic to the body and process glucose and turn it into glycogen, and other such things can be done by the liver. Fats have a different route. Fats do not go to the liver for processing. Rather, fats will go into the lymphatic system and eventually get into the cardiovascular system, as we discussed earlier, that the lymphatic system does bring products into the cardiovascular system. In order to get into the lymphatic system, fats are absorbed by the capillaries of the lymphatic system. The capillaries of the lymphatic system are called lacteals. So now we have all these nutrients that the body is absorbing because of the great surface area of the small bowel, because it has digested the macromolecules into smaller components, and therefore it's being taken in by the body. And then the fluid continues along the small bowel and then goes into the colon, which is the large intestine. Now, the small bowel or small intestine is called small because its diameter is small, but it's the longest part of the alimentary canal. The colon is called the large intestine because it has the widest portion in the alimentary canal. The colon is divided into different segments. It begins with the cecum, which has a small thing attached to it called the appendix. And then we have the right colon or ascending colon, the colon that goes across the body, which is the transverse colon, then down the left side, the left colon or descending colon. Then there's a part of the colon that curves, which is called the sigmoid colon. It ends in the rectum, and then there's the anus. Now, the purpose of the colon is essentially to absorb water, and it's important in terms of electrolyte balance. There is little or no digestion that occurs in the colon, and that is the production of feces, the color of which is produced mostly from breakdown products of red blood cells. And there is a lot of bacteria that is contained in feces. Excretion is the elimination of substances, usually wastes, from the body. Excretion usually starts at the level of the cell. For example, one of the waste products of aerobic respiration is carbon dioxide. So the cell excretes carbon dioxide. It's picked up by red blood cells, as we've discussed before, carbaminohemoglobin in the red blood cells goes through the body up into pulmonary circulation, released in the capillary bed in the lungs, blown off into, diffuses into the alveoli, then blown off through the bronchioles, bronchus, trachea eventually, and then out. It leaves our body, and that is the process of excretion. There are other things that are excreted. For example, red blood cells, when they are broken down into the body, they produce these bile pigments that are excreted into the GI tract along with bile, and then it goes through the GI tract and it is expelled eventually from the rectum, another way of excretion. However, so much waste products are excreted by the urinary system that often we call the urinary system the excretory system. And those products include ammonia and urea, which are breakdown products of protein or amino acid metabolism. Then there is creatinine, which is a breakdown product of muscle metabolism, and so on. In fact, water is one of the dominant things that are excreted, of course, by the kidneys. And water is a breakdown product of aerobic respiration, just like carbon dioxide is a breakdown product of aerobic respiration. So urine is what is excreted by the kidney. And the kidney itself, of course, there are two kidneys, as mentioned when we talked about the adrenal gland. And like the adrenal gland, the kidney has an outer part, which is the cortex, and an inner part, which is called the medulla. It excretes the urine into the renal pelvis, renal referring to kidney. Then it goes from the renal pelvis into the muscular tube called the ureter. And even here, the ureter engages in peristalsis, which is that concentric sort of muscular contraction to move things along. And so the urine passes down the ureter into the muscular sac called the bladder. And then the muscular sac, the bladder, can push the urine down through the urethra. Now, this is the global view of what occurs, but now we're going to look at the functional unit of the kidney in order to see how urine is actually produced. The functional unit of the kidney is the nephron. The nephron is a filtration, absorption, secretion unit. It is divided into two main parts. There is this part here, which is called the renal corpuscle. And then there's this part here, which is called the renal tubule. The renal corpuscle is composed of a ball of capillaries called the glomerulus. This ball of capillaries has an arteriole that's going towards it and one going away. When it's going towards, it is afferent. When it goes away, it is efferent. So there's an afferent arteriole, ball of capillaries, and an efferent arteriole. Surrounding the glomerulus is Bowman's capsule. Now you have to imagine the relationship between the ball of capillaries, the glomerulus, and Bowman's capsule is as if you took a really soft balloon. You made a fist, and your fist is the ball of capillaries. Then you push your fist into the balloon. Then the balloon will begin to surround your fist. The balloon is like Bowman's capsule, and your fist is the glomerulus. So that is the relationship here. It is at this stage that we have the first step of the function of the nephron, which is filtration. Blood is coming in here, And so it's very dependent on what the blood pressure is, how much filtration will occur. And as blood comes in, then some fluid will end up into Bowman's capsule. But Bowman's capsule will filter out things like cells. The cells are not going to cross. Proteins will be prevented from crossing because proteins are very useful in our blood system, in our bloodstream. And so these things will be filtered, and so other products will end up coming in to Bowman's capsule. Then we have the system of tubules. So tubules are just a series of small tubes. And the tubules can be divided into certain areas. There's this part here, which is a bunch of convoluted tubules. And since they're close to Bowman's capsule, they're called proximal convoluted tubules, as opposed to the convoluted tubules which are further away, which are called distal convoluted tubules. In between the proximal and distal convoluted tubules is the loop of Henle. And there are different limbs or parts to the loop. There's a part that goes down, which is the descending limb, and the part that goes up, which is the ascending limb. And eventually the convoluted tubules end up going towards the collecting duct, which collects the urine. However, as the fluid goes through these tubules, it is always called a filtrate until it ends up in the collecting duct, and that's when it is called urine. Now, in the proximal convoluted tubule, the body will resorb up to 85% of the fluid that just came out into the Bowman's capsule. And in fact, this sort of reminds me of my sister's purse. Because when she tries to clean her purse, instead of just removing the things she doesn't want in her purse, she dumps out everything, and then she ends up putting back 85% of what she dumped out. So the body ends up dumping out a large quantity of fluid, and right away it resorbs 85% of that fluid. And this resorption is necessary. It's obligatory, and therefore it's called obligate resorption. So this occurs, and then the filtrate continues along the loop of Henle. In the loop of Henle, this is where the filtrate is concentrated. This is because of movement of sodium and potassium in the medulla of the kidney. There is a concentration in the loop of Henle. In fact, the longer the loop of Henle, the more concentrated the filtrate would become. In fact, I recall looking at electron micrographs or really close views of the loop of Henle of desert rats. And the loop of Henle is extremely long because for a desert rat, it's best to preserve water, and therefore its loop of Henle is very long to super concentrate the filtrate that it is producing. So here is where it is concentrated. Then it goes on to the distal convoluted tubule. In the distal convoluted tubule, this is where we have facultative resorption, which means we have control over the resorption that is occurring here. There are hormones such as aldosterone that can act here. Aldosterone, as we know, secreted from the adrenal cortex and will resorb sodium, and therefore water will come in passively. So this occurs here. Also, ADH, antidiuretic hormone, can act either on the collecting duct or on the distal convoluted tubule in order to help resorb water into the bloodstream. Then the filtrate passes from the distal convoluted tubule to the collecting duct, which collects the urine and passes it along a system of ducts that ends up going into the renal pelvis, the ureter, and the bladder. The new MCAT changed in 1991 and changed again for 2003 emphasizes DNA and genetics more than any other subject in the biological sciences, and that includes organic chemistry. Now, we've already looked at DNA and mitosis. Now we will look at meiosis and other specific aspects of genetics. Now, in terms of meiosis, it is very important in gametogenesis. Gametogenesis is the genesis of, which means the creation of, gametes, which are the eggs and the sperm. Now, there's something unique about meiosis, and that is that meiosis results in a division and reduction of the number of chromosomes. And there is logic to this, because in mitosis, where we have a certain number of chromosomes, and in humans that is 46, there is a division, and then you end up with the same number of chromosomes. So you have one cell that starts, then you have duplication of DNA through semi-conservative replication, and then you have two cells that result, each of which also have 46 chromosomes. 46 chromosomes in humans is our diploid state. That's the state in which we have 2n, where n is equal to 23. So that's 46 chromosomes, which is the number of chromosomes that is available in almost all of the cells in our body. However, because we have all these different cells dividing with 46 chromosomes, but we are made from one sperm and one egg, so the sperm fertilizes the egg. Clearly, if they're going to add their genetic information together, they must only have half the number of chromosomes that we have in most of our cells. In other words, they must have 23 chromosomes in the sperm, 23 in the egg, so that when it comes together for fertilization, then it creates a situation where there are 46 chromosomes. Then that can divide via mitosis, and then you get tons of cells all with 46 chromosomes. So then you end up with 46 chromosomes. There must be some system in which your cells that have 46 chromosomes could then be reduced to 23 chromosomes to make gametes, eggs and sperm, and then the story repeats itself. So meiosis has two stages. It begins, just like mitosis does, with prophase. So prophase for meiosis is a little different, because in prophase for meiosis, when we start to see the chromosomes condense so that the nucleolus disappears, the nuclear membrane disappears, we start to see the microtubules and the centrioles starting to get ready for the other parts of the cell division. So we start seeing some of the chromosomes, and so they decondense, so that means they become more obvious and more clear. But what's interesting about this first prophase, or prophase 1 in meiosis, is that the chromosomes can cross over, which means parts of the chromosomes literally will cross over another chromosome. Because of this crossing over, these chromosomes can actually exchange parts. So they exchange genetic material, which is very important in terms of understanding diversity and different changes that can happen genetically later on. So this all happens in prophase. Then in metaphase, and this would be, of course, metaphase 1, just like in mitosis, in metaphase, you get lining up on the equatorial plate. Well, here again, in metaphase 1, we get lining up of the chromosomes. But the difference is that the chromosomes that line up in mitosis, each one lines up in mitosis. And then each chromosome is pulled apart into separate chromatids. So that's very different, because then you would have 46 chromosomes lined up in mitosis. But here, in metaphase of meiosis, the first part of meiosis, we only have 23 pairs of chromosomes. A pair of chromosome means that one chromosome will line up together with its homologous chromosome, so that you'll get a pair of chromosomes coming together. In fact, there'll be 4 chromatids in each pair. And so sometimes it's called a tetrad, referring to 4. It's also called synapsis. So there is the synapsis of the chromosomes. And then, of course, we have the microtubules that are extending towards, and we have the normal mitotic spindle that exists. But this is all in metaphase 1. Then the next phase is anaphase. And in anaphase, the chromosomes are pulled apart. Now in mitosis, it was chromatids that were being pulled apart. But here, it's actual chromosomes. So you would have the mitotic spindle carrying away a chromosome. And on the other side, you have the same thing. An actual chromosome being pulled away. So this is prophase 1, metaphase 1, anaphase 1. So now you're starting to get reduction of the number of chromosomes. Instead of having 46 in one cell and 46 in another, because actual chromosomes, the entire chromosome, is being pulled away from the other entire chromosome, then you're going to have 23 in one and 23 in the other. This is the reduction division. Then in telophase, this is where you get reforming of the nuclear membrane and the nucleolus and so on. In meiosis, you get a very short interphase. Remember, from mitosis, we said P-mati, or P-M-A-T-I. So you have, after telophase, interphase. And in meiosis, you have a very short interphase, because then you start getting back into the prophase number 2. Now, the rest of the cell cycle will be just like mitosis. So in prophase 2, we start to see the nuclear membrane will disintegrate, the nucleolus goes away, the chromosomes condense, and then the chromatids are being pulled apart into separate cells, etc. So it goes exactly like we described for mitosis in the second stage of meiosis. The first stage of meiosis is different, and this is called the reduction division. So it is with meiosis that sperm is created. And the sperm starts off with a primordial cell, or a basic cell, which are the spermatogonia. And they divide into primary spermatocytes, which start off the meiosis division. And then the primary spermatocytes become secondary spermatocytes. So they go from 2N to having just N. So going from the diploid state to half of the number of chromosomes, which is the haploid state. And then those secondary spermatocytes will go on to create spermatids and sperm. And of course, sperm have the whip-like organelle called the flagellum. Similarly, gametogenesis in females, which is called oogenesis, begins with the oogonia, which is 2N. The oogonia produces a primary oocyte, which goes into meiosis. During meiosis, the primary oocyte, through a reduction division, because the primary oocyte, like the primary spermatocyte, is 2N, then it undergoes a reduction division and may eventually produce a secondary oocyte. The secondary oocyte, which is 1N, eventually becomes the ovum, which is released by the ovary during ovulation. This is day 14 of the menstrual cycle. The egg then migrates through the fallopian tubes where it may be fertilized. Now, the difference between the production of sperm and egg, one of the important aspects is that when you start with one spermatogonia, you produce 4 sperm. The reason is that you start with one cell that is 2N, it undergoes a reduction division. At telophase 1, it's going to separate, and then you'll have 2 cells. each of those two cells will undergo a type of mitotic division afterwards. And because it's undergoing a type of mitosis, each one of the cells will produce another two cells. So then you end up with 4 cells from the first spermatogonia. Now, for the egg, starting off with an oogonia, you have the potential of producing 4 ovums, but that does not occur. What happens is that you only produce 1 ovum for 1 oogonia, but you had the potential of having enough genetic information for 4. But what happens is that the 3 other will degenerate. And so the genetic material that could have produced 3 other nuclei will degenerate in just a collection of genetic material which will not be used, and those are called polar bodies. Genetics is the study of the principles and mechanisms of heredity. Central to the discussion of heredity is DNA. Now, we've already looked at DNA. We saw that it is a double helix structure, and we've looked at chromosomes and how each chromosome is composed of 2 chromatids. We saw that this area, the central area, was the centromere, and the distal area was the telomeres. Now, the DNA is wound up using proteins, some of which are called histones. And so the DNA is wound up with proteins to form each chromatid. In other words, each chromatid contains the double helix of DNA. This is why in mitosis the chromatids can separate and yet can still replicate according to semi-conservative replication, because each chromatid will contain the double helix of DNA, which will unzip and serve as a template for new DNA strands to be produced. Now, on the chromosome, as we've already discussed in terms of DNA, there's an area of DNA that codes for 1 protein or 1 polypeptide. We call that area a gene. That segment of DNA is called a gene. The actual location of the gene on the chromosome or on the DNA, the location is called a locus. Now, these are 2 homologous chromosomes. This is sort of a magnification of meiosis, when we saw that the homologous chromosomes will migrate together. So these homologous chromosomes are 2 chromosomes, one coming from your mother, the other coming from your father, so one of maternal origin, the other of paternal origin, and these 2 chromosomes code for the same products. That is the definition of homologous chromosomes. And that makes sense in terms of meiosis, because we saw that the homologous chromosomes will separate in the reduction division in meiosis. So it makes sense because, at least this way, in each cell you will have enough genetic information to code for whatever products you need. Because right here you have enough genetic information to code for all the products twice. This has all the information, this has all. So the homologous chromosomes separate. So we have 23 pairs of homologous chromosomes. Now, on the homologous chromosomes, if we were to look very closely, there would be a gene here, for example, and there would be a gene here that would code for the same product. Now, in general, both chromosomes, each chromosome will code for the same product, so there will be a location somewhere where we can say, for product whatever, the size of your ear, this particular product, here is where it's coded for on this chromosome. So they're both coding for the same thing at this particular location, and that is what an allele is. These are 2 alleles. So there are two genes that code for the identical product. Now, if these two alleles code for the same thing, the same size of whatever, the same color of whatever on your body, if they do the same thing, they're called homozygous. If they code for two different products, then they're called heterozygous. Now, sometimes one of the alleles will be dominant over the other, meaning that if, even though it is heterozygous, one of the alleles is so strong that when you have both of these alleles, one will dominate the others. So that would be called a dominant characteristic, whereas the one which is dominated would be called a recessive characteristic. An example of that would be the color of eyes. One might code for dark eyes, which would be dominant over light eyes. So that if you have the gene for both, your eyes may be dark because the gene for dark eyes is dominant over the other. Of course, this is a simplification. Now let's symbolize the idea of dominant and recessive. Let's say that the gene for dark eyes is symbolized by this, a large E. The idea here is that the capital letters are used to symbolize dominant genes and smaller case letters are used to symbolize recessive genes. So if we look at the different possibilities that there could be, we could have a dominant gene which would be for dark eyes, and if you could possibly have two alleles for that gene, because you would definitely have two alleles. It's either you have one dominant and another dominant, or the other option is you have one dominant and the other is recessive, or the other option is that you have both that are recessive. Now, if you have two dominant, it's clear that you would have dark eyes. If you have one dominant and one recessive, well, then you would still have dark eyes because the dark eyes dominates over the recessive allele. Then if you have both that are recessive, then in this case you have nothing dominating over the other, therefore it would be expressed as though you have light eyes. So then there are two things that are going on here. One is your set of genes. This is described as your genotype. That's your set of genes, either your capital E, your capital E, or your small e. That's your set of genes. The other thing is your phenotype. How are those genes expressed? What do you see on the person? How can you tell? What can you measure? What can you determine? And so that concept is your phenotype. And that is the interaction between your genotype and your environment. That's your phenotype. So when we have these set of genes, we notice that we have three different genotypes, but we only have two different phenotypes. Either the eyes are dark or they are light. But in this particular example, there's nothing in between. And so we have examples of dominant genes, recessive genes, phenotypes, and genotypes. There are situations in which, because this is one dominant gene, but there are situations in which genes can be codominant. There might be a couple of genes that are dominant over a couple of other recessive genes. An example of incomplete dominance or codominance is seen in the ABO blood types, and we're going to look at that next. As you may know, there are 4 different blood types. They are A, B, AB, and O. So these are the 4 different blood types. In other words, because these are measurable, these are things that we can see about you, these are called phenotypes. So there are 4 phenotypes for the blood types. Now, the way it works is as follows. On a red blood cell, there are antigens. And these antigens are designated this way. So if you have the antigen A on your particular red blood cells in your body, you're blood type A. If you have the antigens B, you're blood type B. AB, if you have both A and B on your red blood cells, then you're blood type AB. And if you have neither the antigens A nor B, then you're blood type O. Now what happens is that these antigens are also called agglutinogens, and that is because if they are attacked by antibodies, the red blood cells will clump, and that is agglutination. So the way it works is that if you have type A blood, therefore you have A antigens on your red blood cells, then in your bloodstream, in your serum, you're going to have anti-B antibodies, meaning you cannot deal with type B blood as a transfusion, for example, because you will attack it and you will have agglutination because you have anti-B antibodies. Of course, you're safe because you have only A antigens on your red blood cells. If you're the type of person who's type B blood, then floating around in your bloodstream, in the serum, is anti-A antibodies. If you have type AB blood, then you have A and B on your red blood cells. Therefore, in your serum, you cannot have antibodies against A nor B, or you would destroy your own cells. And then finally, type O blood. Type O blood, since there is no antigens on the red blood cells, there are antibodies against A and B. Now, you may recall that type O is considered to be the universal donor. Some students like to remember there are two O's in the word donor, so they like to think of it as being related to the type O blood. So type O blood is the universal donor. You may want to pause now and try to consider how is it possible that type O blood can be given to any other person if type O actually has anti-A and anti-B antibodies. Well, the reason is that when blood transfusions are made, the serum is removed. Therefore, type O can be given to any person because the red blood cells have no antigens. So if you give just the red blood cells, not the serum is removed, you give just the red blood cells to a person with type A, no problem. The red blood cells go into the bloodstream, and the person does not attack those red blood cells because there's no such thing as an anti-O antibody because there's no antigen on those particular red blood cells. And so for B, you can transfuse type O blood, and for AB, you can transfuse type O blood as well. So, and type O blood obviously can be transfused to a person who has type O blood. So type O is considered to be the universal donor because yes, it has anti-antibodies, but that's removed, and the red blood cells have no antigens, so that can be provided to these other people. If you have type O blood, you can only receive blood from someone who's type O, because any other blood will contain antigens. And people with type O blood have anti-A and anti-B antibodies, so they will attack anything that provided antigens. So this is how the blood system works, and those are the 4 different phenotypes. Now, in terms of the genes that represent these different phenotypes are quite varied, and you must know that the gene for A is dominant, the gene for B is also dominant. So these are co-dominant genes. The gene for O is recessive. Therefore, in order to have type A blood, you can have, and this is just the symbol for the gene, it's symbolized often with an I, a capital, and also a capital A next to it. So if you have the alleles that have the dominant for A and a dominant for A, so those are your 2 alleles on your particular chromosomes, your homologous chromosomes, then that would code for type A blood. But this also would code for type A blood. If you had A dominant and then you had the recessive O on your other gene, then that would also code for the same thing. Both of these would code for type A blood. And for type B blood, if you were to have IB and IB, or you had IB and IO as your genes, both of these genotypes code for the same phenotype because the B is dominant over the O. Now, in order to have AB, all you would need to have is the gene for A and the gene for B. And since they are codominant, therefore, they would code together for AB. And then for O, well then here, because it's recessive, you'll need to have the gene for O and the gene for O. And so if you have these two alleles for the recessive gene, then they indeed would code for type O blood. Therefore, you have 1, 2, 3, 4, 5, 6 different genotypes to create 4 different phenotypes. So now we can see that in your body, for a particular trait, you may have 2 alleles on your homologous chromosomes. But in a population, there may be multiple alleles for a particular trait, just like we see here. Now, also talking about blood, we do need to mention the Rh factor. The Rh factor is a different gene found on a different locus on the chromosome. So it's not the same as these ABO blood types. So the Rh factor, it's either on or it's off. In other words, you're either Rh positive or you're Rh negative. And the Rh positive is dominant. And in fact, 85% of our population is Rh positive. Now, Rh is different from the ABO blood types, again, for another reason. When you're born with type A blood, you have anti-B antibodies. If you're born with type B, you've got anti-A antibodies automatically. But people who are Rh positive will not have any Rh antibodies. But if you're Rh negative, you might think that you have anti-Rh positive antibodies. Not really. You're not born with them, at least. You need to be exposed to Rh positive blood in order to develop the antibodies against the Rh antigen. Because clearly, if you're Rh negative, you don't have the Rh antigen. So you're not going to have the antibodies yet until you're exposed to the blood. Once exposed to the blood, you develop these anti-Rh antibodies. Then if you get a chance to attack a blood cell that has the Rh positive on it, then it will attack and it will agglutinate. That's why when we say that O is the universal donor, that is true. But it's usually O negative blood that will be provided in emergency situations when someone is bleeding to death or something, and there's not enough time to get more specific blood. We will take O negative blood to do the transfusion, to provide the least risk of having any reaction against the blood. Now, the time that this becomes a problem is sometimes when a woman is Rh negative, during her first pregnancy, if her baby is Rh positive, there might be some exchange of blood during the delivery of the baby. And so the mother might have been exposed to the baby's blood. So there's a little transfusion of blood that occurs there. And because of this, the mother will develop anti-Rh positive antibodies because she's been exposed to it in the baby. Now, if the mother were to have another baby, and if that other baby is also Rh positive, then the mother who has the antibodies against Rh, some of those antibodies may cross the placenta and start attacking the baby. And this would be fatal for the baby if left untreated. Now we're back to our homologous chromosomes. So again, each chromosome in the pair codes for the same products. So if you were to look at the chromosome and you looked at a particular allele on one chromosome, particular allele, and they both coded for the same trait, we now come to our first law in genetics. And that is that if you were to look at these particular alleles that code for the same trait, they are going to end up separating in meiosis, and therefore they will be segregated because each one of these alleles will end up in a different cell because the homologous chromosomes paired and then they were pulled apart. Each will have that particular allele. So this is the Mendel's first law of segregation, and that is that alleles coding for the same trait will be segregated, meaning they'll be pulled apart, put into separate cells. Now, you've got, in humans, 23 pairs of chromosomes. So we have a whole bunch of these pairs of homologous chromosomes, half from maternal origin, half from paternal origin. When these chromosomes separate and go into different cells, it is not the case that 23 from paternal origin will end up in one cell and 23 from maternal origin will end up in the other cell. No, because the chromosomes line up, but they don't say all the guys on the left, all the women on the right. They don't do that. They line up down along the equatorial plate, and then they are separated randomly, independently. Therefore, it could be the case that the one of maternal origin goes left, and you look down, maternal origin goes right, look down, left, right, left. It could be in any particular order. And therefore, because they will separate independent of what the other pair of chromosomes does, that brings us our second law. And that's Mendel's second law of independent assortment. Now, given those laws, and of course, like anything else, there are some laws that are broken, and so there are some anti-Mendelian concepts. For example, we expect that the homologous chromosomes will sort themselves independently. But sometimes there's information on one chromosome that sorts independently. Now that is not something that Mendel would have considered. But the reason this occurs is because of what we saw earlier. In meiosis, there's crossing over. So therefore, one would expect this whole chromosome to move over, but sometimes the chromosome will cross over in prophase with its homologous chromosome, and they will exchange parts. And so if this chromosome gives up a certain part, then most of the chromosome might go one way, while part of the chromosome is on the other chromosome going in the opposite direction. So sometimes information on the same chromosome will sort itself separately. And so knowing that there is this sort of crossing over that occurs, the next question is, well, how can we determine what sort of rate of crossing over is happening? And it has been determined that when you have genes that are very close together, well, then we say that they're highly linked, meaning that the chances that those two genes that are close together on the chromosome or in the DNA, when they're close together, it's highly unlikely that they're going to separate. But if you have genes that are far apart along the chromosome, then the chances that they get separated by crossing over is much higher. Besides crossing over, there is another exception to Mendelian genetics, and that is the idea of sex linkage. Now, according to Mendelian genetics, we expect there to be 23 homologous pairs of chromosomes. So here we have two pairs of chromosomes, and we see that these two pairs of chromosomes have the same sort of look or appearance or morphology. And so we can look at specific areas on one chromosome representing an allele present at a particular locus on that chromosome, and then we can look at the same locus on the other chromosome and look at the other allele. They both might be recessive, one might be recessive, the other dominant, or they both might be dominant alleles. So we expect to see 23 pairs like this. However, there is an exception. There's actually 22 pairs that have the same morphology for all of humans. And those 22 pairs of chromosomes, which would be like this, those 22 pairs of chromosomes are called the autosomes. Then there is another pair of chromosomes, which is called the sex chromosomes. And in females, there are 2 X chromosomes, and in males, there is an X chromosome and there is a Y chromosome. Now, the X chromosome has a similar morphology to other chromosomes, but the Y chromosome is actually a very small, tiny clump of DNA with histones and so on. And so it has an appearance which is very different from the other chromosomes. Now consider this. This has because the morphology is the same, we know that we can find the allele on the other chromosome which is coding for the same genetic information. However, when you have an X chromosome and a Y chromosome, we will see that in males, therefore, there's going to be things that are coded on the X chromosome which are not present on the Y chromosome because the Y chromosome doesn't have as much information as the X chromosome does. So in females, where there would be 2 X chromosomes, then it would be similar to the other chromosomes in which the X chromosome would have another allele on the other X chromosome that will be coding for the same genetic information. So you would have dominant and recessive, and it would be the same situation. However, for males, because there's going to be information coded on the X chromosome, which is not present on that little clump on the Y chromosome, therefore, there will be genes that may be recessive for the female, but for the male, that gene suddenly becomes expressed because there is no other dominant gene in order to dominate or be superior to the recessive gene on the X chromosome. Therefore, that would actually be expressed in the male. In other words, this is the situation where a woman can have a recessive allele. Therefore, because it's recessive, and if the woman is heterozygous, so she also has the dominant gene, therefore the phenotype will not be expressed. The recessive phenotype will not be expressed. However, because of Mendel's laws of genetics, in which those X chromosomes in meiosis will be separated, they will go into different eggs. One egg will have one X chromosome, the other will have the other. Now, in the X chromosome that will have the recessive allele, if that bonds with another X chromosome, in other words, fertilization to create a human, and if it comes together, 2 X chromosomes together, in which one X chromosome has the recessive allele, the other has the dominant, therefore no problem. You have another generation in which that particular woman will be a carrier of the recessive allele, but she will not express the phenotype because she has the dominant allele. Now let's put the situation to the male. Now if there is the egg with the recessive X chromosome with the recessive allele on it, then it is fertilized by sperm carrying a Y chromosome as the sex chromosome. Then this will create a male. But because the X chromosome carried a recessive allele and the Y chromosome does not contain that allele at all, that recessive allele, which was not expressed in the daughter, will now be expressed in the son. So the son will have the disease. He will have the phenotype. So this is a classic case in which a woman has children, and among her children, none of her daughters have the disease, but all are carriers of the genotype. So they're all heterozygous. Then all the sons have the disease. They have the phenotype. So this is called sex linkage. And this is when the disease is linked to the sex chromosomes. Now we've looked at the exceptions for Mendelian genetics. Now we will examine what the consequence is of Mendelian genetics. And the important consequence of this goes as follows. To begin with, recall that capital letters represent a dominant trait, whatever that trait might be. Coding for a particular protein, hair color, nail strength, whatever it might be. So this is a particular trait, and we have it as being homozygous. Homozygous meaning it's both the same, and it's dominant. And this one is homozygous but recessive. Now let's say that the mother had the homozygous dominant trait. The father had the homozygous recessive trait. So it means that the mother, in all her gametes, she's going to have a dominant gene in every one of the gametes, because these will separate independently, and therefore she will always, in all her gametes, after meiosis, she'll have capital A in her gametes. And for the male, he would have the recessive gene in all of his gametes. So if these were to mate, what you would have is, in every single situation, you would have a sperm with the recessive gene and an egg with the dominant gene in every single situation. So all of their children are going to have the dominant A and the recessive A. All of the children will have as their pair of homologous chromosomes for that particular trait. They will have dominant A, recessive A. In other words, they will be heterozygotes, or also we can call them hybrids. So all the children in the population that comes from parents that, as described, they will all be these hybrids. So now, if we were, this particular generation would be called the first filial generation, symbolized by F and 1. So this is the first filial generation. Filial simply means the sons and daughters of the parents. So this is the first generation, sons and daughters of the parents. They would all have this hybrid. Now, if we were to look at the second generation, the next generation, this is how we would do so. We would recognize that in every one, whether male or female, because this particular trait will assort independently, or will segregate independently, because it will do so in the gametes, half of the gametes will have the dominant gene. The other half will have the recessive gene. That goes for male and that goes for female. Half will be each. So if we had a population of hybrids and they were to mate, and you knew that in each of these hybrids they had half their gametes dominant and half were recessive, then what would happen in the next generation of sons and daughters, also called the second filial generation, or F2? So let's examine what would happen in F2. To begin with, we can work this out using a Punnett square. And all that means is we draw a square like so, and now we will put along one side and the other side what the gametes will be, recognizing that we'll put the males down one side, the females down the other. And if we go with the males down one side, it's going to be the same thing, which is that each male would have half the gametes would be the dominant A, so one half would be the dominant A, and one half would be the recessive A. And then going along the top, we do the same thing. So one half would have the dominant allele, or A, and the other half would have the recessive allele. then all we have to do is put in the square what the product would be. For example, the product here would be the product between this and this. So we would have 1/2 times 1/2, which would be a quarter, and we would have capital A, capital A. Down here we have 1/2 small a times 1/2, so that's a quarter, and it would be capital A, small a. And over here, likewise, we have 1/4, and it would be capital A, small a. And then down here we would have 1/4, and we would have small a, small a. So if we were to look at the population to determine what would happen to a population that began with this hybrid or a heterozygote for a particular trait, what would happen to that trait given a population? Well, what would happen is we would get 1/4 of the population with this particular genotype, AA. We would get 1/2 of the population, 1/4 plus 1/4, with capital A, small a, and we would get 1/4 of the population with the recessive genotype, which is small a, small a. So in terms of the genotypes, we would get 1, 2, 2 to 1, because it's 1/4 to 1/2 to 1/4, so that creates this ratio, 1 to 2 to 1, which would be the ratio of the genotype, which is homozygous dominant, to the genotype, which is heterozygous, and the genotype, which is homozygous recessive. However, in terms of the phenotype, the ratio is going to end up being 3 to 1. And the reason it's 3 to 1 is because what is going to be expressed would be the dominant gene. And so here the dominant gene is expressed. Here it is expressed because even though it's heterozygous and the person would be a carrier for the recessive genotype, it will actually be expressed as the dominant phenotype because it has the dominant allele. So this is going to be the dominant. This would also be the dominant allele will be expressed. So then in 3 cases, you get the dominant allele being expressed, and only in 1 case do you get the recessive allele being expressed. So the ratio for the phenotype would be 3 to 1. The ratios for the genotype would be 1 to 2 to 1, and that is given a population. So in terms of giving a population, you could say, well, there will be a 50% chance that someone's going to end up with the hybrid as a genotype. There'll be a 50% chance given a population. So in other words, you can't say for any particular person being born, you can't say, oh, well, you're going to definitely be this or that. You can say that there's a 50% chance that that person being born will be a heterozygote for that particular trait if both parents were heterozygotes for that particular trait. So it is a probability that you're talking about, not specifically what any individual would have. Welcome to General Chemistry for the MCAT exam. In order to understand chemistry and the interaction of chemicals, one has to understand the atom. It is very key to the interaction of chemicals to understand the electrons that are around an atom in order to determine the unique features of atoms. these electrons around the atom and the way that they are organized is referred to as the electronic configuration. There is certainly some overlap between our review of the atom in chemistry and our review in the atom in physics. Now let's look at the most simplistic view of an atom. This was the view of the atom that was put forth by Rutherford. And what he had suggested was that there was a nucleus at the center of an atom, and that nucleus contained neutrons and protons. And then in orbit around the nucleus were the electrons. This view of the atom is sort of based on our understanding of the solar system. And that is that there is the sun in the center of the solar system, and then there are planets that are revolving around the sun. And this is how this simplistic view of the atom was developed. However, experiments were done that were to show that we cannot detect exactly where an electron is in space around the nucleus. In other words, when we have the sun at the center of the solar system, we can always tell where the Earth is with respect to the sun at any given moment. But when you have a nucleus and an electron which is in orbit around the nucleus, we cannot detect precisely the position of the electron at any given time. And therefore, we could only discuss the probability of finding the electron somewhere in space around the nucleus. We can't say exactly where it is. We can't pinpoint its position in orbit. We can only discuss the probability of where it can be found. And so Schrödinger presented an equation in order to determine how we can describe where the electrons are around an atom. When you take Schrödinger's equation, which you do not need to memorize for the MCAT exam, when this equation is solved, you get 4 solutions. And these 4 solutions are important for the exam. These 4 solutions are called the quantum numbers. Thus, there are 4 quantum numbers, and these quantum numbers will describe any electron in any atom. The first quantum number is called the principal quantum number n. The principal quantum number n can take on certain values. Those values are 1, 2, 3, etc. Each one of these values has a letter associated with it. The number 1 is K, L, M, etc. These are capital letters. So n is the principal quantum number, and it describes the shell. I like to think of the shell as the big picture. And that is to say that you have a nucleus, and then around it, at some point in space, there is a shell, an electron shell, which describes the probability distribution of finding an electron in space. In other words, instead of being able to say that this is the nucleus and here's the electron, you can't say that because the electron is moving at a speed which is much too great. But what you can say is, here's the nucleus, there is a shell around the nucleus, and that shell represents 90% probability of finding the electron somewhere in that shell. Where? We don't know. But it's somewhere in that shell, 90% chance. So if we're talking about shell K, that would be the first shell. So we have the nucleus, we move away from the nucleus, and there's one shell. If we shell L would be the second shell. So wherever the nucleus is, we move away from the nucleus. That's the first shell. We move further from the nucleus, and there we have a second shell. and so on. So quantum number n, the first quantum number, or the principal quantum number, is what I regard as the big picture. It determines the shell of the atom. So we have the nucleus, we have the shell, which is the big picture. Within each shell, there are electrons, or there may be just one electron. But this electron has a mass. The mass of the electron is very small, particularly in comparison with a neutron and a proton. The mass of the electron is something like 10 to the minus 31 kilograms. It's extremely small mass on an electron. You don't need to memorize that number. So the mass is moving in space, so that we have a mass with motion, so the mass has a velocity. Mass times velocity, you may remember from physics, is momentum. Thus, the electron has a momentum associated with it. Thus, we get the second quantum number, which talks about the momentum of the electron, but because the electron is always changing directions, because it's moving in some sort of form, like a sphere, therefore its momentum is angular, because it keeps changing direction. So the second quantum number, L, is also called the angular quantum number. It's also called the subshell, so it's within the shell. It is also called the orbital. So the quantum number L provides the subshell or orbital of an atom. It has particular values as well. Its values that it can take are 0, 1, 2, right on up to n minus 1. There are letters associated with these values. These are large case letters. The letters associated with the subshell L are small case letters. S, P, D, etc. So these are the letters that are associated with these values. So if you're talking about the orbital P, therefore it's the same thing as L is equal to 1. Each one of these orbitals has a different shape. The S orbital is spherical. So you have the nucleus, and the S orbital describes a sphere around the nucleus. You have the P orbital. The P orbital describes a dumbbell shape. So if this is the nucleus, the P orbital describes a shape where it is lobed above the nucleus and it is lobed below the nucleus, sort of dumbbell shaped, and that describes one P orbital. And the D orbital, well, you don't need to know that for the MCAT exam. It's a very pretty shape with a dumbbell and a donut around it, but you don't need to know that for the exam. So these are the main orbitals that you need to be able to describe. In the S orbital, it can take on a maximum of 2 electrons. The P orbital can take on a maximum of 6 electrons. We'll look at how that figures out in a moment. Let's go on to the third solution from Schrodinger's equation, so the third quantum number. The third quantum number has to do with the fact that we have a nucleus, we have the shell, we have the subshell within the shell, which describes the orbital, and then the third thing is we have this electron, which has a charge, and it's moving in space. By definition, when a charge is in motion, it has a magnetism associated with it. So the fact that this electron is moving in space around a nucleus, there is a magnetism associated with it, and so we get our third quantum number, which is m sub L. This describes the third quantum number, and it is called the magnetic quantum number. So the magnetic quantum number has certain values that it can take on. And these values can be described as plus or minus L to plus or minus 1 to 0. And we'll see how this comes together when we look at our first atom. Now, we have the nucleus, we have the shell. The electron or electrons are in the shell. There's an orbital associated with it, which is the subshell, and then there's the magnetic quantum number, which describes this charge moving in space. The fourth quantum number refers to the fact that not only is the electron moving around the nucleus in space, and therefore has a magnetism associated with it, but the electron is also spinning on its own little axis. For example, the Earth is spinning around the sun, but the Earth is also turning around its own axis as it's spinning around the sun. Therefore, the electron has a very tiny magnetism associated with the fact that it is turning and spinning on its own axis. Now, there's only two possibilities. Either the electron is spinning clockwise, or it is spinning counterclockwise on its own axis. Thus, there are two quantum numbers, two possibilities for the fourth quantum number. It could either be plus a half or minus a half. And this refers to the spin, clockwise or counterclockwise, on the electron as it's turning around the nucleus. So here we have the fourth and final quantum number, the magnetic spin quantum number, m sub s, and it can have the values plus or minus 1/2. Now, let's make an atom and see what it looks like. So, if we were to take a nucleus and we were just to add one electron, we were just going to go and look at what the first shell would look like around a nucleus. And we're talking about any shell from any atom, no matter what the atom looks like, we're going to look at what the first shell would look like. Well, if you have the first shell, then n is equal to 1. So we're talking about the K shell. If n is equal to 1, then L, the subshell or orbital, whose maximum value is n minus 1, well, 1 minus 1 is 0, so the only value that we can have for the subshell or orbital L will be S. Therefore, we know the shape of the first shell. We have the nucleus, and the shape of the first shell will be spherical about the nucleus. Obviously, what I am trying to suggest to you in your imagination is not drawn to scale, because the electrons are very much further from the nucleus than my hands are from the fist. But nonetheless, just to give you the concept, so you have the nucleus here, the first shell is spherical around the nucleus. Then, how many electrons can we fit in the first shell? Well, if the L is equal to 0 and m sub L, the magnetic quantum number, its values are plus or minus L to 0, well, plus or minus L would be plus or minus 0. Therefore, the magnetic quantum number would only be 0, only one option. If n is equal to 1, L is equal to 0, m sub L is equal to 0. But the magnetic spin quantum number has two possible options, plus or minus a half. Thus, if we're talking about the first shell, there are two possibilities in the first shell, plus or minus a half, and that's it. Therefore, there is a maximum of 2 electrons that can fit in the first shell. And those 2 electrons are clearly in the s orbital. So we've looked at the nucleus, we've looked at the first shell around it. Now we move out and let's look at the second shell and what an atom would look like as it fills up its second shell. Well, the second shell is n is equal to 2, the big picture. So it is the L shell. So in the second shell, n is equal to 2, we have L having values of 0 up to n minus 1. n minus 1 in this case would be 2 minus 1, which is 1. Thus, the maximum value for L would be 1. So L would have two possible values, 0 or 1, either S or P as orbitals. Thus, we have the nucleus. The first shell is spherical. We move away. Then we have the second shell, n is equal to 2. And in this second shell, n is equal to 2, there are two subshells, two orbitals, S and P. The S orbital in this second shell is spherical. That's the shape of all S orbitals. The P orbital is dumbbell shaped, meaning the nucleus is here. We have the dumbbell shaped orbitals extending into the second shell. It will extend in the Y direction. It will extend in the X direction, and it will extend in the Z direction towards you and away from you. So that is the P subshell. And as I mentioned before, two possible electrons in the S subshell. P, there are 6. Then M sub L, remembering that there were two possible values for L, 0 or 1. Well, for M sub L, when we have 0 as a value for L, we have plus or minus L, making 0. Therefore, we end up being magnetic spin plus or minus a half, two possibilities again, identical to the situation of the first shell. In other words, in the first shell, we had an S orbital, and its maximum number of electrons were 2. When we move out to the second shell, there's an S orbital, and there's a maximum, once again, of 2 electrons that fit in the S orbital. In the P orbital, where L is equal to 1, now we have the values of M sub L as plus or minus 1, which means plus or minus 1, plus or minus L, right to 0. So we have 3 possible values for the magnetic quantum number, plus 1, minus 1, and 0. Those are the 3 possible values for the magnetic quantum number. For each of these 3 possible values, we have a magnetic spin of plus or minus a half. So we have plus or minus a half, plus or minus a half, plus or minus a half, meaning 6 possibilities in the P orbital. And that is why the P orbital can fit a maximum number of 6 electrons. So my question to you is this: what is the maximum number of electrons that can fit in the second shell? You may want to pause and consider your response. The answer would be 8 electrons. The reason is because in the second shell, n equal to 2, where we have possibilities of 0 or 1 for L, 2 electrons in the S, 6 electrons in the P, 2 plus 6, 8 electrons. This brings up our first equation, which you should memorize. And that will make your calculation a little faster. And the equation is that the maximum number of electrons in a shell is equal to 2n squared. Thus, in this particular instance, where I asked what is the maximum number of electrons in the second shell, well, the second shell would be n is equal to 2, 2 squared, 4, 4 by 2, 8. And so this is another way of calculating the maximum number of electrons in any particular shell. So we mentioned the maximum number of electrons in this orbital is 2, in the P orbital is 6, and I'll also tell you that the maximum number of electrons in the D orbital is 10. We are about to look at the shorthand for electronic configuration. In other words, the shorthand for taking the quantum numbers, which we just looked at, and expressing it. But what would be of value is keeping in mind the periodic table. This will be useful for many of the discussions that we're going to be having over the next couple of subjects. So this is an extremely symbolic look at the periodic table, in which you see in the first group of the periodic table, you have what is called the alkali metals. In the second group of the periodic table, you have the alkaline earths. Running across the periodic table, you have the transition metals. And then other notables is that you see oxygen and sulfur, which are in the sixth group. And then in the seventh group, where you have fluorine and chlorine and bromine, these substances are called the halogens. And in the eighth and final group, we have the noble gases, which used to be called the inert gases, but they have been found to be reactive under extreme conditions, and so they are more correctly called the noble gases. A periodic table will be provided to you during the MCAT exam for your use at any point for the different science sections: physics, biology, chemistry, organic chemistry, and general chemistry. Now, in terms of the shorthand which is expressed for electronic configuration, you might see something like so: 1s2, 2s2, 2p4. What this stands for is the following. The number in front of the letter stands for the quantum number n, which is to say the principal quantum number. So this is the principal quantum number n. The s stands for the orbital, so the quantum number L. And the 2, or the superscript, that stands for the number of electrons. So this is 1s2, meaning that this shell, subshell, is fully booked. It's complete. It has its 2 electrons. Here is the second shell, and it has 2 orbitals, as we saw before, s and p. The s is completely filled with 2 electrons, and the p is partly filled because it could have a total of 6 electrons. It happens to have 4. Now, if you were given this and asked what substance does it represent, well, what you would do is, if this is a neutral atom, and that means all the electrons is equal to the number of protons, then you would just count the number of electrons. 2, 4, plus 4, 8 electrons. So there are a total of 8 electrons here. And then if you were to be asked what type of substance is it, or in what group in the periodic table, or what's the name of the substance, well, you would just look for atomic number 8, because that would indicate the number of protons that it has by its atomic number, and that would be equal to the number of electrons from the electronic configuration that we've been provided. And so if you look on the periodic table, which would be provided to you during the exam, you would find that this represents oxygen. Now, what happens if they give you electronic configurations, but it goes on and on and on? It's much longer. They go up to the 3rd shell and 4th shell, etc. How do you know for sure how each of the orbitals will be filling up? Well, there's actually a way of just remembering it. This is not scientific whatsoever. It's just a way that it'll be easier for you to remember how to fill up the shells in sequence. So the way you do this is you write the following. You write 1s, 2s, 3s, 4s. Then you write 2p, 3p, 4p. Then 3d, 4d. So once you've written this down, then you draw diagonal lines, like so. And now you're going to read it in this direction. So this is how you would read it to fill up the orbitals. So you start here, you read 1s2. Then you go back up here, 2s2. Up here, 2p6, because that would be the maximum number of electrons that can fit in a p orbital, 3s2. You go back up here, 3p6, 4s2. Up here, 3d10, 4p6, 5s2, etc. So this is just a way in order to simplify your ability to fill up the orbitals should you be presented with a problem with a series of orbitals for the electronic configuration. Based on the electronic configuration of atoms, atoms are able to be placed in a periodic table because of their similarities and their differences. But these similarities are shown in a very periodic or regular way, and we'll be able to see that now. The periodicity refers to the fact that there is this regular pattern that we can come up with by examining the periodic table. The first topic will be ionization potential. Ionization potential is the energy needed in order to remove the least tightly bound electron. So if you have some substance, X, and that substance is ionized, so it becomes X plus an electron, there is a certain energy associated with this. The energy associated with this reaction is the ionization potential. So if you can imagine your nucleus with electrons and shells around it, there has to be some electron, which is likely the furthest away from the nucleus, which is the least tightly bound electron. In order to remove that, there's an energy, and that is the ionization potential. The trend in the periodic table is as such. As you go across the periodic table, the ionization potential increases. So we'll put ionization potential as IP. It increases. As you go down the periodic table, the ionization potential decreases. Why? Well, the reason is, as you go down the periodic table, you are starting to add shells, meaning you start off with your first shell, n is equal to 1. You go away, second shell, third shell, fourth shell, further and further away from the nucleus. Because each time you're adding a shell and you're going further and further from the nucleus, then the bond between the positively charged nucleus and the negatively charged electron, which becomes further and further away, becomes weaker and weaker. And so because the bond is weaker, it's easier to remove the least tightly bound electron. It requires less energy to remove it. Therefore, the ionization potential is less. But when we go across the periodic table, we're not adding shells. We're just adding electrons to the same shell, and we're adding protons to the nucleus. So the nucleus becomes more and more positive, and the shell, the outer shell, becomes more and more negative. So as we go across the periodic table, the atoms become smaller. They become heavier because there's more protons and you're adding electrons, but they become smaller because the force of attraction between the electrons in the outer shell and the nucleus, which is becoming more and more positive, becomes greater and greater and greater, thus reducing the size of the atoms. Because the atoms are being reduced in size, because the attraction is becoming greater and greater between the shell and the nucleus, therefore, the energy required to remove the least tightly bound electron becomes greater. Therefore, the ionization potential increases. The other important concept regarding the periodic table and its trends is that of electronegativity, which we'll just write it as such. Electronegativity. Electronegativity measures the pull on an electron when the atom is in a molecular environment. Electronegativity is a key concept which we'll be using over and over again in the organic chemistry review. So the trend of electronegativity in the periodic table is very similar to the trend of ionization potential, meaning as you go across the periodic table, you have an increase in electronegativity, and as you go down the periodic table, you have a decrease in electronegativity. Thus, as you go across the periodic table, you will find over here fluorine, and fluorine is the substance, because as you go down, the electronegativity decreases, so you go up, the electronegativity increases, and you go across, it increases, and it is fluorine right here which has the greatest electronegativity on the periodic table. We are now about to look at chemical bonds. Now, chemical bonds can either be within a molecule, which would be intramolecular bonds, or it can be between molecules, which would be intermolecular bonds. We're going to look at both. For example, let's look at bromine. Bromine is Br2. So here is Br2. So here's a bromine molecule just hanging around, not doing anything particular. It is covalently bonded, meaning it is sharing a pair of electrons equally. And why not? The two atoms are identical. Therefore, one atom doesn't pull on the bond more than the other. They share this bond equally. So this would be a perfect covalent bond, a bond between two atoms within a molecule, an intermolecular bond. Now let's say that we presented something large and negative to this system of bromine molecules, lots of bromine molecules, and we present something huge and negatively charged. Well, this thing that is negatively charged is now going to change the molecules. And the reason is this negative charge here will repel the electrons in the bond. It will push the electrons away because like charges are repel, just like opposite charges attract. So these like charges repel, therefore these electrons are going to be pushed over towards this bromine. As these electrons are pushed towards this bromine becomes a little bit negative because it used to be completely neutral, sharing an electron bond, but now it becomes a little bit negative as these electrons are being pushed onto this bromine atom. So the symbol for a little bit negative is delta negative. This bromine over here, because it's losing electrons that it used to have, so that negative charge it used to have around it is now being pushed over here, this bromine starts becoming a little bit positive, or delta positive. Once this bromine becomes a little bit positive, and it's near a negative charge, it becomes even more attracted to this charge. And the more that it becomes attracted to this charge is the more the charge repels the bond, and the more this becomes delta negative. Now, imagine that there is another bromine molecule sitting right next to it. This bromine molecule was also neutral. However, now that this bromine atom is becoming increasingly delta negative, its delta negative charge repels the electrons here, making this delta negative and this delta positive. Now we have a force of attraction. Therefore, we've created a situation in which there are now intermolecular bonds, meaning an attraction between two different molecules of a substance. And that attraction is based on very basic concept, which is opposites attract. This force of attraction, this bond between these two molecules, is attracted by what is called van der Waals forces. Van der Waals forces is talking about this induced bonding, electrostatic interaction between these molecules. Among the different types of intermolecular bonds, van der Waals forces are considered to be among the weakest. But nonetheless, they are important in terms of chemistry, and these can affect the boiling points of substances, because if a substance would boil easily, well, if you increase the intermolecular bonds, the bonds between the molecules, then the substance would want to hold itself together and remain in the liquid form, and therefore it'll be harder to boil it, harder to break those intermolecular bonds and send the substance into the vapor phase. So if you had bromine without van der Waals forces, just normal everyday bromine, without this charge being introduced, then the bromine would boil at a certain temperature. But if you induce the dipole, and now you have this interaction between bromine molecules, then it would require a higher temperature to boil, more energy in order to break these intermolecular bonds. Incidentally, the fact that you introduced a charge to create this change in distribution of charges within the molecule, and we will see in a few moments that this change in charge within a molecule, when there is a separation of delta negative and delta positive charges, a net separation of charges, we call this a dipole. And when you induce a dipole, it is called an induced dipole. Not too complicated. Anyway, let's move on to hydrogen bonding. We are now going to discuss hydrogen bonding. Consider the following. Here's water, and here's CH4. CH4 is an organic chemical, and it's called methane. Meth has to do with the fact that it has only one carbon, so it starts with meth. And this is an alkane because it's a hydrocarbon. Now, if you were to look at the molecular weight of these substances by examining the periodic table, you'll see that hydrogen is 1 times 2, so that's 2, and then we have oxygen, which is 16. So this is 18 grams per mole. Then for methane, you have carbon, which is 12 on the periodic table. Hydrogen is 1 times 4, so that's 16 grams per mole. So water is just a little bit heavier than methane. Well, what that means is that under normal circumstances, if you had two chemicals whose properties were very, very similar, and one was heavier than another, you would expect that it would be more difficult to push that substance up into the vapor phase. In other words, the heavier molecule would have a higher boiling point, would require more energy to push it into the vapor phase because it's a heavier molecule. Well, water is just slightly heavier than methane. And at room temperature, methane is, well, it's a gas, methane gas. And water is, well, it's a liquid. In fact, water is a liquid all the way up to 100 degrees. You have to push water all the way up to 100 degrees in order to be able to get it into the vapor phase. But yet, their molecular weights are so similar. And yet, water requires so much more energy in order to get it into the vapor phase. Why? Hydrogen bonds. because water has bonds between the molecules and it holds it together in the liquid form and makes it more difficult to push it up into the vapor phase. Now, if we were to look at a water molecule, we can symbolize it like this. And so this is a water molecule, H2O. And then if we looked at the periodic table, we would find that oxygen is to the right on the periodic table, hydrogen is to the left in the periodic table. Therefore, electronegativity of oxygen would be higher because oxygen is more to the right. So oxygen attracts electrons to itself. Therefore, oxygen would be a little bit negative, delta negative, and the hydrogens would be delta positive. When you have many different water molecules in your substance, they arrange themselves in a particular way. The oxygen has a couple of lone pair of electrons. The other neighboring water molecules will be attracted to the delta negative oxygen because they have delta positive hydrogens. So we have the opposites attracting, delta positive to delta positive in this way, and this creates these intermolecular bonds. So what is a hydrogen bond? Is this a hydrogen bond between the hydrogen and oxygen? No, because this is an intramolecular bond. Hydrogen bonds refer to intermolecular bonds between different molecules. The force of attraction is based on the fact that there's a difference in charges, delta positive to delta negative. What about other substances? What about H2S? Can H2S hydrogen bond? Certainly. It's the same thing. Sulfur, it will be found just under oxygen in the periodic table in the 6th group. And therefore, sulfur has similar properties to oxygen. Certainly, sulfur, because of its location in the periodic table, would be much more electronegative than hydrogen. Therefore, there will be a difference in charges between the two. Therefore, the hydrogen of one hydrogen sulfide molecule would be attracted to the sulfur of another molecule because delta positive and delta negative and opposites would attract. What about this substance? This substance is called ethyl alcohol or ethanol. Eth comes from the point that it has 2 carbons. So this substance, would it hydrogen bond? Certainly, because we have an oxygen here which is delta negative according to the periodic table, because it is right close to this hydrogen, and the hydrogen and the oxygen being so separated on the periodic table, there is a significant difference in their electronegativities, and so we have a delta negative oxygen, a delta positive hydrogen. So the delta positive hydrogen of one ethanol molecule will be attracted to the delta negative oxygen of another ethanol molecule. Do these hydrogens hydrogen bond? No, because between carbon and hydrogen there is very little difference in electronegativity, and therefore there is not a charge difference such that these hydrogens would engage in hydrogen bonding, but certainly these hydrogens would. Now we will look at bonds within the molecule, intramolecular bonds. 2 dominant types, ionic bonds and covalent bonds. Ionic bonds are where there is a complete transfer of electrons. For example, NaCl going to Na plus Cl minus. This is a complete transfer of electrons. This is an ionic bond. There are 3 important things to say about ionic bonds. 1, the melting point is very high. Why? Because 2, the bonds are very strong. Imagine we looked at hydrogen bonds and how they're important because there's an attraction between delta positive and delta negative, partially positive and partially negative. Well, these are fully positive and fully negative charges. So the force of attraction is very powerful. So the bond is very powerful, and therefore the melting point is very high, meaning it takes a lot of energy to break those bonds and to break apart the solid form of this substance. So high melting point, bonds are strong, and thirdly, what is important about these substances is that because they're charged, they are conducting substances. Covalent bonds, well, that's the opposite. In a covalent bond, we have a complete sharing of electrons. So an example of a covalent bond would be chlorine, which is Cl2, or bromine, Br2, which we saw before. So in chlorine, Cl2, the two atoms are identical, and therefore there is no reason why one would pull the electrons more than the other as long as there's no external force. So this is a perfect covalent bond. Well, in chemistry, most bonds are neither ionic nor covalent. They are a combination. And we will look at that next. The Lewis dot structure is very important in general chemistry, and the reason is it allows us to keep an eye on the outermost shell electrons. And in general chemistry, that's where the action is, because reactions are occurring with the outermost shell electrons. So let us look at the Lewis dot structure for hydrogen. Hydrogen in the periodic table is number 1, and therefore its neutral form has 1 electron in its outermost shell. So we'll symbolize it like this. And we'll add it with fluorine. Now fluorine in the periodic table, you'll find it in the 7th group. It is atomic number 7, and therefore it has 7 protons and therefore 7 electrons in its outermost shell. So we will add 7 electrons to its outermost shell. There's 2, here 4, 6, and 1 more. Well, there are many substances in chemistry that prefers to have 8 electrons in its outer shell. It completes the shell. The reason why they used to call these the 8th group in the periodic table the inert gases is because they didn't seem to be very reactive. And the reason is they're very, very stable substances. So the noble gases are very stable, and it's because they have a complete outer shell. And so there are many substances that try to be like a noble gas. And so in their effort to try to be like a noble gas, they will try to have, for example, 8 electrons in their outer shell. That is called the octet rule. So fluorine is one of these substances, and fluorine has 7 electrons, but it can get 8 if it bonds with hydrogen. And by bonding with hydrogen, fluorine will end up being surrounded by a total of 8 electrons, making this very stable situation. So we have hydrogen thus bonded with fluorine. But this is not a true simple covalent bond. After all, fluorine is the most electronegative element on the periodic table, and hydrogen's way on the other side. So fluorine is drawing electrons to itself, and fluorine thus is going to be seriously delta negative, and hydrogen is going to be delta positive. Thus, the bond is not shared evenly or equally. Thus, we call this bond a covalent bond with ionic character. So, as I mentioned before, the majority of bonds in chemistry are not covalent and they are not ionic, but they're somewhere in between. And this is a classic example of a covalent bond with ionic character. Of course, the way we generally describe such a bond is simply calling it a polar bond. Sometimes compounds can achieve the octet rule by forming multiple bonds. For example, carbon dioxide. Carbon has 4 electrons in its outer shell. Oxygen has 6 electrons in its outer shell. Oxygen, of course, is in group 6 and thus has 6 electrons in its outer shell. So we draw 2, that's 2 for oxygen. Here's another 2, that's 4 for oxygen. And here's another 2, that's 6 for oxygen. So we do the same thing on the other side. Total of 6 for oxygen. So oxygen can bond with carbon with its 4 electrons in this manner. Thus, the octet rule has been achieved. Because oxygen is surrounded by 2, 4, 6, 8 electrons. This oxygen also 2, 4, 6, 8 electrons. So this is a more stable position for it. And carbon has 2, 4, 2, 4. So a total of 8 electrons surrounding the carbon. So each of the atoms is able to achieve the octet rule by doing this special type of bond. And this bond, the way we would generally write it, is simply carbon double bonded to oxygen, double bonded to another oxygen. And this is a multiple bond. Instead of the single bond that we've previously seen, this is a multiple bond. And of course, because of the difference of electronegativity, carbon being to the left and oxygen being to the right, carbon would be delta positive, each oxygen would be delta negative. Another example of the octet rule being achieved with multiple bonds is nitrogen, or N2. And nitrogen would be in the 5th group in the periodic table. Nitrogen has 5 outer shell electrons. And so if we draw nitrogen here in N2, each nitrogen has 5 outer shell electrons. So we'll draw 1, 2, 3 for this nitrogen, and there's the other 2. So this has 5 electrons. Here we draw 1, 2, then we draw 1, 2, 3. So we have 5 electrons here. Now by bonding in this manner, each nitrogen is surrounded by 8 electrons. So this nitrogen has 2, and then there are 6 here, so it's a total of 8 electrons. And this nitrogen has the 6 here and the 2 here, total of 8 electrons. Therefore, each nitrogen, by bonding in this way, by using a multiple bond, is able to achieve the octet rule. So now we have a 1, 2, 3, a triple bond. As you notice, each line represents a pair of electrons, an electron pair. So there's 1, 2, 3 electron pairs. In other words, 6 electrons in the bond between the 2 nitrogens. So these are multiple bonds. There are 2 things to say about multiple bonds. To begin with, multiple bonds are unsaturated. Anytime you see a multiple bond, it means that the atom is unsaturated, meaning that the atom is not bonded to the maximum number of other atoms that it could be bonded to. For example, carbon can be bonded to 4 different atoms, like for example in CH4, methane, which we saw earlier. It could be bonded to 4 different hydrogens But here, carbon is only bonded to 2 oxygens. It's not bonded to 4 different substances. Therefore, this carbon is said to be unsaturated. It's an unsaturated compound. And this is the case, of course, with nitrogen as well, because it's not bonded to the maximum number of atoms that it could be bonded to in theory. So multiple bonds are unsaturated. The second thing is, multiple bonds are stronger. That is to say that by having a double bond, that is stronger than only having a single bond. I am not saying, however, that the second bond in the double bond is stronger than the first bond that was laid down. That is not the case. The second bond is actually weaker. However, by having 2 bonds, it is stronger than just having 1 bond or a single bond. And by having 3 bonds, that is even stronger still than having 2 bonds or just having 1 bond. Resonance is a very important concept to understand, since it is one of the foundations of organic chemistry. We will look at the resonance of sulfur dioxide. First, in terms of what the molecule would look like, using the octet rule, remembering that sulfur is in the 6th group and oxygen is in the 6th group, so both have 6 electrons in their outer shell. So here's sulfur with its 6 electrons in its outer shell. So we'll put 2 here, we'll put oxygen here, another oxygen here, and we'll put their lone pair electrons as well. So here we have this oxygen achieving its octet, 2, 4, 6, 8. This oxygen, 2, 4, 6, 2, 4, 6, 8. The sulfur, having its 6 electrons, 2, 4, then it has 6, so it has 2, 4, 6, 8 electrons in its, for it to achieve its octet. So each will contribute 6 electrons. Now, what we have here is a multiple bond. We have a double bond on the left, and we have a single bond on the right-hand side. But here becomes the problem. Here's sulfur. This is the other way of writing it. This is the exact thing, just simplified. But the problem is, why isn't it like this? What's the difference? Well, there is no difference. There is no reason why the double bond should be on the left side any more than why it should be on the right side. Because there is symmetry in terms of the electronegativity of these 2 substances, there's no reason why one would have a double bond more than the other. And so, if you were to do an experiment to determine what side the double bond was on for sulfur dioxide, you would find that there is no double bond in sulfur dioxide. And you would find that we simplify things by writing the bonds as we do. But in reality, because of this symmetry, there is no double bond here at all. What there is, in actuality, you would find with your experiment that sulfur is bonded to oxygen, but that there are electrons which are delocalized, meaning they're not, the double bond is not there, it's not on the other side, but rather it is spread over the molecule. And so you wouldn't find that there is a single bond in sulfur dioxide, nor would you find that there's a double bond. You would find that there's about a bond and a half on each side. And so this idea that there's electrons which are delocalized over a period, they're not localized just in somewhere, they're delocalized over the molecule, that's called resonance. And electrons are able to increase their stability when they are able to have a resonance structure. Now notice, the word is a bit of a misnomer because some students hear the word resonance and they think that there is resonating, meaning it's going from this to this and back and forth from this. It's not. The actual molecule is this, but we symbolize it either this way or this way or this way. These are just symbols, but the true molecule looks something like this, where the electrons are delocalized over an area. We are now going to look at the concept of molecular polarity in the context of BCl3. This is boron, this is chlorine. So boron, if you look at the periodic table, is going to be to the left of chlorine, and chlorine being in the 7th group with the rest of the halogens. So we have boron, B, and we have chlorine, Cl, Cl, Cl. Now, because boron is to the left in the periodic table, boron would be delta positive, and the chlorines are all going to be delta negative, as each of the chlorines have a greater tendency, more electronegativity, greater tendency to attract electrons to them. So they are attracting the electrons in the bond. So these are polar bonds. There is no doubt of that. So the question is this a polar molecule? The answer is no. Yes, these are polar bonds, no doubt about that. Each of these connections have a difference in electronegativity. However, if you look at the overall molecule, because of the symmetry, the center of positive charge, well, that would be here. There's only one place where there's something which is partially positively charged. The center of negative charge for the molecule would be also right here. Because of this symmetry, this is a triangle in which at each corner there's a delta negative charge, so the center of negative charge would also be here. Whenever you have the centers of positive and negative charge at the same position, therefore the molecule is not polar, and therefore the molecule has a zero dipole moment. A dipole moment is something that you do not need to know the details of. You don't need to know how it is derived in terms of using polarized light and watching how molecules move or do not move when there's polarized light. But what you do need to know is the definition, and that is there is a dipole moment present when there is a separation of the centers of positive and negative charges in a molecule. When the centers of positive and negative charges in the molecule are in the same position, then there is a zero dipole moment. Let's look at a couple of other examples. For example, water. For water, we have oxygen, hydrogen, hydrogen. So the question could easily be, is there a non-zero dipole moment or is there a zero dipole moment? Well, the center of negative charge will simply be here. That's where it's delta negative. This is delta positive, this is delta positive. The center of positive charge would be this point between the hydrogens. And therefore, there would be a difference between the center of negative and positive charges. Therefore, water would have a dipole moment. We will now look at hybridization. Hybridization refers to the changes that occur within an atom when the atom is about to bond with another atom. For example, let us first look at the electronic configuration of a carbon atom. Now, in order to write the electronic configuration of a carbon atom, you can look at the periodic table locate carbon, atomic number 6, and because it's atomic number 6, which means it has 6 protons, it must also have 6 electrons. So then we can start filling up our orbitals according to the idea that we have to use up 6 electrons. So we're going to look at the carbon atom. So in the carbon atom, we're going to look at the atom as follows. This box will place the first electrons, which will be 1s. In the next box, we'll place the 2s electrons, and in this box, the 2p electrons. Remember, we have 6 electrons to place. The idea that we're going to draw these diagrams using boxes, it has a very complicated name. They're called box diagrams. And anyway, it helps us to have sort of a bookkeeping for the electrons to watch where they go and where they will end up. So in the first box, in the 1s box, we know that there's a maximum of 2 electrons that can fit in this box from what we've already discussed about electronic configuration. So we're going to put 2 electrons in this box. We remember also that the magnetic spin quantum number can be plus a half or minus a half depending on which way the spin, clockwise or counterclockwise. So we will symbolize the 2 different spins by placing 1 arrow upwards, so it's just really a half of an arrow that we're drawing, and 1 arrow downwards. So this is the symbol for 2 electrons in the 1s box. In other words, this is 1s2. So we go to the second shell. We have 4 more electrons. So we place 2 electrons in the 2s shell. So here's 1 electron, and here's another electron. So we have 1s2, 2s2. 4 electrons have been placed. There's 2 electrons left. So we place 2 electrons in the p. So we'll have 1s2, 2s2, 2p2. So we place our first electron in the p. And the second electron is such that because it's all in the same orbital and electrons are negatively charged, the electrons would rather be as far apart as possible, as energetically possible. And so the next electron, instead of being in the same box, will move on to the other box, 1 level higher, because it would rather not be as too close to the other electrons. So here we have 1s2, 2s2, 2p2. That's the carbon atom. Well, something will change in the carbon molecule. So here's the 1s box, 2s box, and here's the 2p box. Well, this is what happens. In the carbon molecule, the 1s box, it's the same. But the change occurs over here. And this is the reason. If you can imagine carbon having its nucleus and its electrons around it, it would have the first shell, then a second shell. And in the second shell, it would have 4 electrons. 1, 2, 3, 4 in shell number 2. So it has these 4 electrons in its outer shell. Now let's say that it wanted to bond, like for example with hydrogen. So we have carbon atom, we have hydrogen atom, and they're going to bond. Well, this would require significant energy to bring these 2 atoms together, because on the outer surface of the atom there are electrons. They are negatively charged, and the outer surface of the hydrogen there are negatively charged electrons. So as the 2 atoms are approaching, there is an attraction between the electrons of one and the nucleus of the other, but there's also this repulsion between the electrons as these atoms are being brought together. Because there is the repulsion between the electrons, when the atoms are approaching, there is an elevation of the energy. And because there's an elevation of the energy, one of the electrons in the carbon molecule pops up to a higher level. So we have 1 electron here, one here, and one here. So we end up with one electron in the s orbital, one s electron, then we have three p electrons. We have three p electrons. Now it's going to make something brand new, a hybrid. It's no longer going to be spherical. It's no longer going to be exactly dumbbell shaped. It's going to be a new arrangement of the electrons in order to be able to bond with this other atom. And that new arrangement is not S, it's not P. It is 1 plus 3, 4 sp3 hybrids. So this is a brand new arrangement of the electrons forming an sp3 hybrid, and there are 4 sp3 hybrids because there are 4 electrons. So 4 possible bonds with this new sp3 hybrid. So for example, you have carbon forming one of its sp3 hybrids. Here's another. Here's another. Here's another, which is going away from you into the board. These ones are coming out. This is in the plane. And then here I'll draw some spheres at the end of these bonds. And those spheres represent hydrogen atoms. So this would be a representation of CH4. Now you will notice that this forms sort of a pyramid-like structure. And this structure is called a tetrahedron. And the angle between these bonds is called the tetrahedral angle, which was 109.5 degrees. It's 109.5 degrees, which is the normal tetrahedral angle. Now let's say we have another molecule like ammonia, NH3. Well, NH3 has its sp3 bonds, and it's bonding its sp3 hybrid bonds with a hydrogen. So this is NH3. Very similar to CH4, except NH3 has a lone pair of electrons. So these lone pair of electrons, which I'll symbolize like this, are on the nitrogen, and then nitrogen has these 3 bonds. So a common question is, what would be your comment on the difference of the angle between these bonds and between the lone pair and these bonds in the nitrogen? Well, this angle here would be greater than 109.5 degrees. The reason is, when carbon has a bond to another atom, its electrons are moving away from carbon in order to bond with the hydrogen. But when you have a lone pair of electrons, the electrons are remaining closer to the nitrogen atom. Because the electrons are closer in the molecule, therefore these electrons, being negatively charged, will repel the bond with the other hydrogens. And because it repels, it pushes these away because they're like charges. So it pushes this away and makes the bonds down here smaller, but the bonds up here larger. Now we can summarize the hybrid orbitals. So if you were to have a 1s electron plus a 1p, you would create 2 sp hybrids. And as soon as you know that there is an sp hybrid, then you know that the shape of the molecule must be linear. And an example of that would be BeCl2. Now, if you were to have 1s electron and 2p, then you would have 3 sp2 hybrids. So there will be 3 different hybrids, and the shape would be trigonal. And we saw an example of that, which was BCl3, where boron was at the center of a triangle with chlorine in the corners of the triangle. That would be, there are 3 hybrids there, 3 separate bonds created, and so those are sp2 hybrids. And when you have 1s plus 3p, you get 4 sp3 hybrids. And this shape, of course, is the tetrahedron. So this would be a tetrahedron, and we saw the example of that, which is CH4. So let us look at the tetrahedron with a couple of different molecules and a couple of different hybrid orbitals. Here is a compound that has many different hybrid orbitals. The compound has 2 carbons, and it's a hydrocarbon. So it has 2 carbons, so it's eth, and it's a hydrocarbon, so it's an alkane. Therefore, it's called ethane. So this compound, each carbon has 4 different bonds. So right away, you know that it has to be at the center of a tetrahedron. Each carbon is at the center of a tetrahedron. And so we also know that the carbon would have sp3 bonds. So this bond right here would be an sp3 to sp3 bond, meaning this carbon has had a hybrid orbital, which is sp3. It hybridized in order to form the bond, and this carbon did the same. And so this bond would be sp3 to sp3. All these other bonds, for example, this one over here, would be an sp3 to an S, because hydrogen only has 1 electron in its outer shell, and that electron is in the only possibility for its orbital, which is an S. So therefore, it's an sp3 to S bond. These bonds that involve s orbitals are called, the Greek letter for S, a sigma bond. So these are all sigma bonds because they involve the s orbital. Now, this is ethane. Let's look at ethene, also called ethylene. Because it ends in -ene, you know it's an alkene. All alkenes have double bonds. So here we have carbon, double bonded to carbon, hydrogen, hydrogen. Note, each carbon is only bonded to 3 other atoms. Because it's 3 other atoms as opposed to 4 in ethane, 3 hybrid bonds, so it's an sp2 hybrid. So these are sp2 to s bonds. So these are still called sigma bonds because they involve the s orbital. Here, we're going to have one of the bonds being sp2 to sp2. So that would be a sigma bond. But there's another bond, and that bond would be from the extra electron that wasn't used in the hybrid bond. Because it's called an sp2, that means, as we've just seen, it has 1 s electron and 2 p electrons. But when we looked at the carbon molecule preparing to bond, we saw that the carbon molecule would have in its outer shell 3 electrons in the p orbital. So there's only 2 in the hybrid bond. That means there's an extra p electron in each of the carbon atoms. So those p electrons are bonding to each other. And this p to p bond is called a pi bond. So that's a pi bond. The sigma bond is stronger than the pi bond. That's what I was referring to earlier when we talked about multiple bonds. The first bond, being the sigma bond, is stronger than the pi bond. But having 2 bonds certainly is stronger than just having 1. So we've completed our review of bonds. Now we're going to move on to gases. the fundamental concepts behind gases includes pressure, volume, and temperature. In terms of pressure, it is very important to remember that the units of pressure for the SI system is a Newton per meter squared. A Newton per meter squared is also called a Pascal. The other numbers that you must memorize is that 1 atmosphere is equal to 101.3 kilopascals. Kilopascals, in other words, that's 1,000 pascals. So that's 101.3 thousand pascals, and that is equal to 760 torr, which is equal to 760 millimeters of mercury. In terms of temperature, the key units for temperature, of course, are degrees Celsius. So if we have x degrees Celsius, that is equal to x plus 273 Kelvin. Because degrees Kelvin is the units that will be most often used when we are doing our calculations. So the conversion factor between degrees Celsius and Kelvin is 273. The next important concept in gases is that of STP. STP stands for standard temperature and pressure. Standard temperature and pressure is a temperature of 0 degrees Celsius, which means 273 degrees Kelvin, and a pressure of 1 atmosphere. Common mistake that students make is to use the standard temperature at 25 degrees Celsius. The reason some people make that mistake is because the standard temperature for liquids is 25 degrees Celsius, but the standard temperature for gases is 0 degrees Celsius, which is equivalent to 273 degrees Kelvin. The other important concept when discussing gases is that of partial pressure. The partial pressure of a gas in a mixture is the pressure that the gas would exert if it were alone. In fact, Dalton's law states that the sum of partial pressures in a mixture is equal to the total pressure. This is a very important concept, and it is commonly tested on the exam. For example, let's say that we have a container, and in the container we have two gases, oxygen and nitrogen. If in the container oxygen has a pressure, if it were in this container all by itself, it had a pressure of 2 atmospheres. And if in the same size container nitrogen had a pressure of 1 atmosphere, if we were to put both these gases in the same size container, the total pressure in the container would be the sum of the partial pressures of these gases. So the total pressure in the container would indeed be 3 atmospheres. Furthermore, as a result of Dalton's law, it is possible to state that the mole fraction of oxygen in this container is simply 2 over the total pressure, which is 3. So the mole fraction of oxygen in this container would be 2/3, and the mole fraction of nitrogen in this container would be 1/3. And this is a result of the fact that each gas acts independently in the container. So the pressure of the oxygen is the same pressure that it would have if it were alone. Adding nitrogen to the container does not alter the pressure caused by oxygen. But adding nitrogen into the container, if nitrogen adds pressure, therefore the total pressure will change. And you can calculate the mole fraction simply by taking the partial pressure of the gas divided by the total pressure in the container. Now, another commonly tested concept, that of Avogadro's law. Avogadro's law, or principle, states that for a given temperature and pressure, equal volumes of a gas contain equal number of molecules. And in fact, at STP, standard temperature and pressure, temperature of 0 degrees Celsius or 273 Kelvin, pressure of 1 atmosphere, we have Avogadro's number of particles. Avogadro's number of particles is 6.02 times 10 to the 23rd. And that is 6.02 times 10 to the 23rd is the number of particles per mole, number of atoms or molecules per mole. That's Avogadro's number, and the volume associated with this is 22.4 liters. Certainly, these are numbers to memorize. So, what is the significance of Avogadro's law? It is very important. What it is saying is that if we have, for example, two different rooms filled with different gases, the gases are at standard temperature and pressure. Now, these gases can be very different. One gas can be some normal gas like nitrogen, and the other gas could be something very complicated, some organic chemistry type of gas that's in that room. But both gases are at standard temperature and pressure. Well, Avogadro's principle is stating that if you take a container which is 22.4 liters in size, and you walk into room A, you open up the container, and you let the gas diffuse inside, and then you close the container. Then you can walk into the hallway, and someone can go up to you and say, how many particles do you have in that container? Right away, you can say 6.02 times 10 to the 23rd. You can go into another room with a completely different gas, open up a container, which is 22.4 liters, allow the gas to diffuse in the container, walk into a hallway. Once again, how many particles do you have in that container? 6.02 times 10 to the 23rd. Just that straightforward. And if someone were to ask you, how many moles do you have in that container? You have 1 mole. Even if you don't understand the complexity of the gas, what it is, what the molecules are shaped like, you still know exactly how many particles there are in there based on Avogadro's principle, meaning that you know if the temperature and pressure are standard and you have a container of 22.4 liters, then you know you have a mole, and you know you have Avogadro's number of particles in every mole. So to make this as practical as possible, whenever you see the number 22.4 on the MCAT exam, or any factor of that, for example, if you see 11.2, if you see 44.8, right away you should zero in on that. Those are volumes that should tell you something. 44.8 tells you we're dealing with 2 moles of a substance if it is at STP. And 11.2 means it's half a mole. And those are numbers that they will throw at you, and you have to immediately be able to recognize them based on Avogadro's principle. And certainly any factor of 6.02 times 10 to the 23rd is something that you should immediately recognize based on Avogadro's principle. Now we will look at the equation of state. Thus we have the equation of state. PV equals nRT. P, the pressure, V, the volume, N, the number of moles, R, the gas constant, and T, the temperature in degrees Kelvin. In terms of R, I usually memorize the R in terms of liters atmosphere per degree, and the unit that I remember is 0.08. That's the R that I remember. But you can remember R in terms of any of the units that you wish to, because depending on what units used for pressure and volume, you will have different number for the gas constant R. But for me, I tend to try to put everything in terms of liters and atmospheres, and therefore 0.08 is the R that I tend to use. But you can use any one you want as long as you're consistent in any problem solving to make sure that all the other variables are in the same units as the gas constant that you're using. Now let's manipulate the equation a little bit. If we bring temperature over to the other side, we get PV over T is equal to nR. Now R is the gas constant. If you are not altering the number of moles, meaning you have a gas at a particular temperature and pressure and volume, and then you alter that in some way, but you don't change the number of moles, you're not adding gas to it or removing gas from it, well then you would get that PV over T is a constant. And what that means is that P1 V1 over T1 is equal to P2 V2 over T2. That's if you're changing the pressure before and after, the volume before and after, and the temperature before and after. So you have PV over T is equal to a constant. Now let's say that you were not changing the temperature. The temperature was remaining constant, but you were changing the pressure and volume. Well, then you would just take off the variable temperature, then you would get PV is a constant. That means that P1 V1 is equal to P2 V2. And once again, if you weren't changing the volume, then the volume would be constant. And therefore, you would have P1 over T1 is equal to P2 over T2. And finally, if the pressure was constant, then you would have V1 over T1 is constant. So there, V1 over T1 is equal to V2 over T2. So these are different ways that the equation of state can be manipulated or changed to be altered for specific situations. Of course, they're given specific names, but that's not as important as in terms of how you can use the equation to solve problems. The other manipulation, which is often used on the MCAT exam, is the following. Number of moles in terms of mass and molecular weight is equal to mass over molecular weight. So we have PV is equal to mass over molecular weight RT. So this is equivalent to PV equals nRT, because of course mass is in grams, molecular weight is in grams per mole. So if you cancel the grams, then you get moles on top, and that's the same thing as being the number of moles. So that's just dimensional analysis, looking after the units. So if we were to write the equation this way, if we were to divide both sides by volume, this is what we would get. Pressure is equal to mass over volume, RT, and we bring the molecular weight over to the side. Molecular weight. Why is this manipulation so important? Because when we talk about gases, sometimes instead of simply providing the number of moles, they may provide you with the density. And mass over volume is density. So this here becomes R rho, or D, depending on how you symbolize density, but usually density is symbolized by the Greek letter rho. And so we have pressure is equal to density times RT over molecular weight. So this is how they can ask you a question and you know the equation PV equals nRT, but you didn't remember how does density fit in, but then you make this simple change or alteration in the equation in order to see how density can fit into the equation. Graham's law. Graham's law deals with the rates of diffusion and effusion. Diffusion is defined as the spontaneous spreading of a gas through another. Of course, in reality, diffusion is the spontaneous spreading of anything through another. After all, liquids diffuse, and even solids diffuse. Rocks diffuse into rocks. over millions and millions of years. And in fact, if you sat down long enough, your butt would diffuse into your chair. Of course, that takes a lot more time than this video will last. Nonetheless, effusion is the streaming of gas through a fine hole. The rate of either diffusion or effusion is given by 1 over the square root of the molecular mass. So this is more of a proportionality. So the rate is proportional to 1 over the square root of the molecular mass. Essentially, what this is saying is, if you have a heavy particle, a particle that has a great molecular mass, many, many grams per mole, that particle will diffuse slowly because it is the inverse of the molecular mass. Therefore, the rate will be less as you increase the molecular mass. The bigger the particle, the more the inertia, the slower it will diffuse or effuse. The smaller particles will have a greater rate of diffusion or effusion. Now we're going to look at liquids. Vapor pressure is one of the fundamental concepts to do with liquids. Let us consider the following. This is a petri dish, and in it there's just some liquid. For example, let's say there's water in here. And then this is a bell jar. So it creates an enclosed space. Well, if we were to place the petri dish inside of this enclosed space, this bell jar, what would happen is that if we were to look just at the surface of the water, we would see that there are some molecules at the surface of the water that have enough energy necessary to enter the vapor phase. There's also going to be some molecules near the surface of the water that are going to condense and go from the vapor phase to the liquid phase. Well, because this is an enclosed space, there's going to be molecules going into the vapor phase, other molecules going into the liquid phase, and after a while, there's going to be a dynamic equilibrium, meaning the amount of vapor and the amount of liquid will be constant. So that's an equilibrium, but it's a dynamic equilibrium, meaning there's still a lot of motion going on because molecules are still going into the vapor phase and coming back into the liquid phase and going back and forth. So there is a motion that is occurring, but an equilibrium has been obtained. Now, let us imagine that we can go right into the surface of the water and see exactly what's going on. What we will see is that there are little molecules continually entering the vapor phase, coming back into the liquid phase, entering the vapor phase, others coming into the liquid phase, and so on. So this is continually occurring on the surface. Well, the fact that there are these molecules that are continually entering vapor phase and coming back into the liquid phase and going up and down and so on, it is applying a pressure on the surface of the liquid. The pressure that is being applied, remember, pressure being force per unit area, and this pressure that is being applied on the surface of the liquid, that is what we call the vapor pressure. Now, let us look at some of the things that can affect vapor pressure. For example, let's look at two substances. CCl4. So that's called carbon tetra for 4 chloride. Carbon tetrachloride and water. Consider which of these two would have a higher vapor pressure. You may want to pause and consider your response. Well, carbon tetrachloride, which would be, of course, carbon has 4 bonds, therefore it's in the middle of a tetrahedron, so the molecule would look something like this. And of course, these are all polar bonds, like we've discussed before, but this is a non-polar molecule, so it has a zero dipole moment. It's a non-polar molecule. This obviously is a polar molecule with a non-zero dipole moment, as we've seen before. And we know that this molecule engages in hydrogen bonds. So, if we had these two substances at the same temperature, which would have the higher vapor pressure? Clearly it would be carbon tetrachloride, because more molecules of carbon tetrachloride would enter the vapor phase, because there's not something holding it in the liquid phase. So they'll have more molecules in the vapor phase, and therefore that's capable of applying greater pressure on the surface of the liquid. As opposed to water, which has hydrogen bonding, therefore the liquid phase is being held together by these hydrogen bonds. There's much fewer water molecules entering the vapor phase. Therefore, because there's less vapor, there's less pressure that could be applied to the surface of the water. Therefore, intermolecular forces have a great effect on the vapor pressure. Now we just looked at how some particles have enough energy to enter the vapor phase. Well, the Maxwell distribution plot will allow us to calculate more precisely how many particles have enough energy to enter the vapor phase, or how many particles have enough energy to do whatever we arbitrarily decide we want to examine. The Maxwell distribution plot looks at a plot of the fraction of particles on the y-axis against the energy on the x-axis. When we look at particles and their distribution of energies at a given temperature T1, it has a bell curve distribution. The bell curve begins at the 0 point, and it has a certain distribution based on the amount of energy and the type of particles that are experiencing that particular energy. The important points about the Maxwell distribution plot include the following. First of all, if we have a temperature T1 and we have the same particles, and then we provide a higher temperature. So we're going to give it temperature T2. Obviously, that's going to give a distribution of energy which is higher, because we're increasing the temperature, so we're increasing the energy of the particles. Well, the key point about the Maxwell distribution plot is that the curve does not simply shift over when you increase the temperature. Rather, the curve always begins at 0, and then the curve, as you get higher and higher energies, the curve will simply not rise as high. It will remain flatter, but it will go further. So this temperature here will be T2, where T2 is a greater temperature than T1. So here we have, the reason why this is so important is because on the MCAT exam they can easily ask you about what change would there be in terms of the distribution of particles, and then they'll draw a bell curve starting at 0, and they'll ask you what happens at a higher temperature. And then one of the possible answers will be that the bell curve shifts to the right. And of course, many students will put this as being the correct answer, because there seems to be some logic associated with that. But it is not logical for the following reason. That is that no matter how much you increase the temperature, there's always going to be some particles that are going to have a low energy. That's a normal distribution. Furthermore, if you imagine these particles having more energy and bouncing around and hitting each other, there's going to be some collisions of these particles that will occur in such a way that a particle will have a low energy, even if it's just for an instant, because it was hit by many particles at a particular angle, at a particular place in the container. And so the curve will continue to spread out, go further, but it would flatten out, And so more particles experience a higher energy, no doubt about that, but there will always be some particles, though a small fraction, that will experience lower energies. If you had to calculate precisely how many particles have a particular energy or higher, then you look at the area under the curve. So say that you wanted to look at the energy right here, and you wanted to know how many particles have this energy or higher, and say that happened to be the energy required to make a particle enter the vapor phase, or to make the particle boil, or something of that nature. Then what you would do is you would look at this point here, and then you would examine the area under the curve. And it's not as if for the MCAT exam you need to do integrals or any complicated mathematics. It's very basic math, but it's just the concept that the area under the curve will provide you with the number of particles with this energy or higher. Let's say a few years from now, your niece comes to your medical practice and asks you for some help with her homework. And she asks you, what is a boiling point? And so then you scratch your head and you think back to your MCAT preparation, and you tell her that a boiling point is the temperature at which the vapor pressure equals the prevailing atmospheric pressure. And indeed, you would be correct. But she's not quite satisfied with your response. And so she asks you, but what are the bubbles all about? You know, when you put water on a stove and you boil the water, there are bubbles. What is that? Why does that happen? You may want to pause and consider your response. Well, I recall that in first year of college, I had one of those first year college pots that sort of moved, tilted from one side to the other. And when it tilted on one side, it would boil on that side. When it tilted on the other, it would boil on that side. So obviously something is happening where the element touches the water, or is closest to the water, and heats the water. Something is happening at that point. Well, just like the Maxwell distribution plot, there is a certain energy that is provided to the molecules when it's closest to the heating element. And when these molecules have that energy, they rapidly expand into vapor. So the heating element vaporizes the water, turns it from liquid to gas immediately. Then the vapor will take on a shape that has the maximum volume of vapor with the minimal surface area. And that is defined as a sphere. So a sphere is created in this manner, and then objects that are most dense go to the bottom. Objects that are least dense rise to the top. And thus the vapor bubble rises to the top because it's least dense, and that's where the bubbles come from. So that is the boiling point. It is the temperature at which the vapor pressure is equal to the prevailing atmospheric pressure. Melting point. The melting point is the temperature at which solid and liquid can exist at equilibrium. The important point about the melting point is that it is pressure dependent. Some people think that ice is slippery. Well, it's not exactly true. Ice is not very slippery. In fact, people who do a lot of skating will know that just ice is not slippery. Water on ice, now that is very slippery. And what happens is that if you apply pressure to ice, you reduce the melting point. Therefore, you can actually turn the solid into liquid. More specifically, a skate is designed so that it applies a lot of pressure to a very fine area. That pressure is applied to ice. It literally melts the ice by lowering the melting point of the ice, melting it, turning a fine layer of liquid water on the ice. Now that is very slippery, and that allows the person to be able to skate without much friction. Le Chatelier's principle. Le Chatelier's principle, which is often tested on the exam, states that when a stress is applied to a system at equilibrium, the system will change in order to relieve the stress. For example, if we have this simple reaction, solid plus heat giving a liquid, well, this type of reaction would be called an endothermic reaction, meaning it's a reaction that requires heat. If this reaction, instead of requiring heat, heat was given off in the reaction, so heat would be part of the products of the reaction, then we would call that an exothermic reaction. Some people remember that if the heat exits the reaction, then it's exothermic. So this is an endothermic reaction, and so if we were to apply Le Chatelier's principle to this reaction, if we were to put more solid in this equilibrium, and we were to add solid, then the stress would be solid. Then it would push the equation to the right-hand side. It would shift the equation to the right. If we were to increase the temperature, so that means giving more heat, then heat would be the stress, and therefore more liquid would be produced because there would be a shift to the right in order to relieve the stress of heat. If you were to add liquid to the equation, then liquid would be the stress, and then the equation would be shifted towards the left. And finally, if you were to decrease the temperature, so consider, if you increase the temperature, you're adding heat, it goes to the right. If you decrease temperature, then it would be the opposite, and then the equation would shift to the left. You are now looking at the phase diagram for water. In the phase diagram, we see that it is a pressure versus temperature diagram. There are three main areas in the phase diagram. They represent solid, liquid, and gas. Clearly, at very, very low temperatures, minus 100, minus 200, you're going to mostly have solid. So you take an extreme example in order to reveal for yourself what part of the graph represents what, or you just memorize it. If you look at very high temperatures, you're going to have mostly gas. The gaseous phase of water is what we often call vapor. And then in between solid and gas, we would have the area representing liquid. Any point along this part of the curve between solid and liquid is a pressure and temperature at which solid and liquid exist at equilibrium. Any point along this part of the curve is where liquid and gas would exist at equilibrium, and any point along this part of the curve is where solid and gas would exist at equilibrium. This point is where solid, liquid, and gas would exist at equilibrium, and therefore it is called the triple point. When a substance goes from solid to liquid, of course, we call that melting. From liquid to gas is vaporization. When something goes from solid to gas, that is called sublimation. When a substance goes from gas to either liquid or to solid, that is called condensation. Shortly, we are going to start looking at something called colligative properties. That's where liquids become solutions. That's when we add things to the liquid and then we see how it will be altered. These colligative properties are properties that depend on the number, not the type of molecules involved. So it could be any type of molecule, but it is the number of molecules that are going to make a difference in terms of the properties that we will be examining in solution chemistry. In fact, when you take a molecule and you add it to, for example, water, the molecule, as long as it dissolves in the water, the molecule will start to interact with the water molecules. This interaction, this new interaction, is going to change the properties of the water in many basic ways. For example, by adding the molecule into the liquid phase of the water, more water molecules are now going to have to interact with this new molecule. That means there are fewer water molecules available to enter the vapor phase because more of them are interacting with this new molecule. Because there are fewer water molecules in the vapor phase, that means in order to boil the water, you're going to have to push harder. You're going to have to provide more energy. Therefore, the boiling point will increase because there are fewer water molecules in the vapor phase. So by adding something into the liquid, and we call that something a solute into the liquid, creating a solution, right away you're going to increase the boiling point of the solution. So whatever water's boiling point was before, which is generally 100 degrees Celsius, all of a sudden the boiling point will be higher, 101 or 102, depending on the number of particles that we add to the water. Similarly, when we add this solute to water, which is called a solvent, so we add the solute to the solvent, creating a solution. Because the water molecules, again, are starting to interact with this new molecule that has been added to the water, it disrupts its ability to form crystals or the solid form. Therefore, you have to decrease the temperature even more in order to produce solid water, or the solid form of water, which is of course ice. So therefore, the boiling point is going to increase and the freezing point will be depressed. And that is a consequence of these colligative properties adding molecules that will interact with the liquid phase. So let us look at how the graph will be altered by this. By adding a solute to the liquid, we're going to alter the graph such that the area where the liquid is will be increased. So I'm drawing arrows here because the area of the liquid is going to be expanded because the boiling point will be increased. So there's more temperature at which you have liquid solution. And the freezing point will be depressed. So there'll be more temperature again at which you will have the liquid form. Now we're going to look at some of the concentrations that we often discuss when discussing solutions. You are now looking at the units of concentration which are often used in solution chemistry. First off, we have mole fraction. Mole fraction is defined as the number of moles of a component divided by the total number of moles. Mole fraction is often symbolized by a large X. Molality. Molality is the number of moles of the solute divided by 1 kilogram of the solvent. So often the solvent in solution chemistry is water. So then we're talking divided by 1 kilogram of water. Molality is often symbolized by a small case m. Molarity. Molarity is the number of moles of the solute divided by 1 liter of solution. Molarity is often symbolized by a large m. So the number of moles of a solute per liter of solution would be something like, say, salt water. The number of moles of the solute would be how many moles of salt were placed in the water. Per liter of solution is just how many liters of salt water is present. Then there's normality. Normality is the number of equivalents of acid or base per liter of solution. Normality is normally symbolized with a large N. We'll talk about normality for a moment because often people are not as comfortable with the concept of normality. Well, first off, normality can be calculated using the equation NV is constant. That means the normality, number of equivalents of acid or base per liter of solution, times volume is a constant. That means that N1V1 is equal to N2V2. So exactly what are equivalents of acid or base? Well, we know that one of the definitions of an acid is that it can give protons. And we know that bases are proton acceptors. So we can look at a unit of acid, meaning, or equivalent of acid, meaning how many protons are dissociable, meaning how many protons can be given off by that substance. And in terms of a base, we can look at a base as how many OH minus groups it has that can be dissociated, because we know that an OH minus group, which is basic, can accept a proton. More specifically, let us look at HCl, and let's pretend HCl was 1.2 molar. So if we have 1.2 molar HCl, that means we have 1.2 moles per liter of HCl. So how many equivalents per liter do we have? What would be the normality of HCl? Well, for every mole of HCl, there is one equivalent, one hydrogen that can be dissociated. So we have one equivalent of acid per mole. This is 1.2 moles per liter. So if we multiply that by 1.2 moles per liter, well then moles cancel, and we end up getting 1.2 normal. It will be 1.2 equivalents of acid per liter. So what happens if we have 1.2 H2, so 1.2 molar H2SO4? Well, now we know we have two dissociable acid components or equivalents, and therefore we can have 1.2 moles per liter times 2 equivalents of acid per mole. The moles cancel, and we end up getting 2.4 normal. So here we look at a couple of components of acid. Examples of bases, NaOH. Clearly NaOH has one dissociable OH minus per mole. And so whatever its molarity will be, it will be equivalent to its normality. Here's calcium hydroxide, which is CaOH2. Therefore, there are two OH groups which can be dissociated, and therefore it will have, its normality will be twice as much as its molarity. So these are examples of normality, and all of this comes down to this basic equation where N1 V1 will be equal to N2 V2. Raoult's law helps us to numerically describe a concept which we've already discussed, and that is the idea that if you have a liquid or a pure solvent and you start to add a solute to the solvent starts to interact with the solute, and therefore the vapor pressure will be lowered because there are fewer molecules to enter the vapor phase. And so we can calculate the vapor pressure P of the solution, which is given by the mole fraction of the solvent times the vapor pressure of the solvent. So in other words, the highest number is going to be the vapor pressure of the solvent. So that was the pressure that was in the liquid before you added anything to it. Then as you start to add solute to the solvent, creating your solution, the mole fraction, which used to be 1 because it was completely solvent, slowly the mole fraction, the fraction of solvent in the solution, starts to decrease. And as this fraction starts to decrease, so too does the vapor pressure of the solution. And so Raoult's law, which is very important, helps to describe this relationship, and thus it is one of the colligative properties in solution chemistry. The next colligative properties that we will look at are boiling point elevation and freezing point depression. Given our discussion about vapor pressure and the phase diagram of water, hopefully you understand at this point why there must be a boiling point elevation and a freezing point depression. Further, the minimum is that these are expressions that must be memorized. It must be very clear in your mind that this is the way things will occur as a consequence of colligative properties. In terms of boiling point elevation, it can be calculated, and the delta T, which is the change in temperature, which will be leading to the elevation of the boiling point, is given by IBM. What an easy equation to remember. Anyway, IBM, and this time it stands for, I stands for the number of particles. B is the boiling point constant. M is the molality. It's not as if you have to memorize a boiling point constant. If it is required, it will be provided to you. The number of particles means the number of particles in that molecule, the number of particles after dissociation. M is the molality in the units moles per kilogram solvent. Some important points about this equation. To begin with, it's a delta T, change in temperature, meaning that let's say that the solvent was water, and you did your calculation, and you come up with delta T, after you just plug these in, you come up with delta T is equal to 2. Great. So your delta T is equal to 2. Guaranteed on the MCAT, one of the answers will be 2. But wait, what was the question? If the question was, what will be the difference or the for the boiling point elevation, then you would be correct. It's 2. But if the question was, what is the new boiling point of the solution, then the answer certainly wouldn't be 2 degrees Celsius. The new boiling point of the solution, this only provides delta T, so you have to add the T to the 100 degrees, which we know is the boiling point of water. So the answer would be 102 degrees Celsius. And so you wouldn't choose 2 as the answer. So delta T is a very important part of the equation to understand that you have to manipulate it. And of course, the exam will also have 98 degrees to just see whether or not you thought that it was boiling point depression instead of boiling point elevation, which occurs definitively as a consequence of the colligative properties. Another important point. Let's say that we took sugar and we put some sugar in water. So we know that the sugar will dissolve in the water. And we took that water and we tried to find its new boiling point. And its new boiling point was, let's say, it was 102 degrees Celsius. So then they ask you, take the same amount of salt, the identical molality of salt, and add it to the water. What would the new boiling point be? You may want to take a moment to consider this and pause and then come back and we'll discuss it. The new boiling point would be 104 degrees Celsius. And this is the reason. The boiling point constant is constant. It is the constant for that solvent. So we didn't change solvents. We didn't change from water to ethylene or something. So we stayed with water. So the boiling point constant remains the same. Molality, that remains the same because we said we'll take the identical quantity of salt as we did sugar. But what about the number of particles? Well, for salt, because salt is NaCl, and that dissolves into Na plus Cl minus, well, we have 2 particles. And that means that the number I for salt will be 2, whereas for sugar it was just 1. So because this becomes 2, whatever the delta T was before, now it's going to be 2 times that number. So if the delta T was 2 degrees, meaning you had a boiling point elevation from 100 to 102 degrees, so the delta T is 2, well, now the delta T will be 4, because we now have 2 particles instead of just 1. Because the delta T is 4, the new boiling point will be 104 degrees Celsius. And finally, with regards to freezing point depression, it is essentially the same equation, where we get delta T is equal to I F M, where I, again, is the number of particles, because this refers to colligative properties. F is the freezing point constant, and M the molality. Arrhenius, Bronsted, and Lowry are all scientists that essentially suggested that an acid is a proton donor, which means it can release a proton, like we looked at when we discussed normality, and that a base is a proton acceptor. The acidity or basicity of a solution can be measured from its pH. And pH is defined as the negative log of hydrogen ion concentration. Of course, this needs to be memorized, and furthermore, most questions regarding acids and bases for the MCAT exam is reduced to your understanding of the use of logarithms and the rules of logarithms. There is also the term Kw. Kw is the water dissociation constant, and it is equal to the concentration of hydrogen ions times the concentration of OH minus, which in turn is equal to 10 to the minus 14. If you were to take the negative log of all these terms, remembering that one of the rules of logarithm is that the logarithm of A times B is equal to the logarithm of A plus the logarithm of b. Remembering this and taking the negative log of each of these terms, we get negative log of kW, so that is defined as pKw. Just like the negative log of hydrogen ion concentration is pH, negative log of kW is pKw. So that's equal to the negative log of hydrogen. Negative log of hydrogen is just pH, plus negative log of OH minus, and that is POH. And so you see the relationship between pH and POH, because now we will develop the conversion factor for it. And that's equal to, well, it would be equal to negative log of 10 to the minus 14. Negative log of 10 to the minus 14. Based on the rules of logarithm, if you have a logarithm, and logarithm, this logarithm is base 10. If you have logarithm base 10, and you have 10 as the factor here, you have log base 10 of 10, then you can take the exponent and bring it to the front. So this is equal to 14, because we'll have a negative sign, and negative 14, so that's 14 log 10. And log 10 is equal to 1. So all this is just equal to 14. So pH plus POH is equal to 14. That means if you're ever given the pH of a solution, you can easily calculate the POH because you know that the sum is equal to 14. We will now calculate the pH of a strong acid. There are many strong acids. The most common ones are hydrochloric acid and H2SO4, sulfuric acid. Let us look at the dissociation of hydrochloric acid. Here's HCl, and dissociating into H plus Cl minus. So it is a proton donor. It released a proton, H plus, and therefore it is an acid. What would you guess the Ka is associated with this reaction? Now a Ka is the acid dissociation constant. It is an equilibrium constant. Ka is equal to the sum the factor of H plus Cl minus over HCl. This is a classic equilibrium constant in which we have the factor of the products over the factor or whatever we have as the reactant. So products over reactants. So if you had to guess what the Ka of this was, it would be near infinity, essentially. In other words, this is such a powerful acid that essentially all of it will be dissolved into its products. And so you have an extremely high value for the products and an extremely low value for the reactant, because all of it is dissolving. It's such a powerful acid. So Ka, the value of this equilibrium constant, is extremely high. So if we were given that we started the reaction with, say, 10 to the minus 3 molar of HCl. Molar, as we described before, is moles per liter, and that represents the concentration of HCl. If we were to start with 10 to the minus 3 molar of HCl, what would be the pH of solution? Well, if we start with 10 to the minus 3, because we know that our Ka is essentially infinity, meaning this will completely dissolve or dissociate, therefore we can suggest that whatever molarity we start with is going to be the molarity of each of these substances later. We start with 10 to the minus 3, then when it dissolves, we will have 10 to the minus 3 molar of hydrogen ions, and we will also have 10 to the minus 3 molar of Cl minus ions. The number of moles of each don't change because for every 1 mole of HCl, we have 1 mole of hydrogen being produced. Therefore, 10 to the minus 3 molar HCl, we will have 10 to the minus 3 molar hydrogen ions. So if we were to ask for the pH of a solution that started with 10 to the minus 3 HCl, then we would just say that pH, which is equal to negative log of hydrogen concentration, negative log, and in this situation, the hydrogen concentration we know would be 10 to the minus 3. And if we have log 10, we have log 10 base 10 to the minus 3, we bring the minus 3 to the front using our rules of logarithm. Therefore, we would have 3 log 10. And log 10 base 10 is just 1. So the pH of the solution would be 3. Just for your own interest, the pH of Pepsi or Coke is about 2. Anyway, we're going to next look at the pH of a weak acid, and we will look at how we can calculate that. We will now calculate the pH of a weak acid. There are many types of weak acids, but the classic weak acid that's given in problems is that of acetic acid. This is simply a symbol for acetic acid, since this is not exactly the chemical formula, but we will look at that closer when we look at organic chemistry. So this is the symbol for acetic acid, which is a weak acid. It happens to be an organic acid. So we know that this is a weak acid because when we look at its dissociation, acetic acid giving off a proton, our definition, one of our definitions of an acid, and producing the proton and the anion acetate. We know it is a weak acid because of its acid dissociation constant. It is not infinity or near so, as in the case of HCl or H2SO4. Rather, the acid dissociation constant is something, 1.75 times 10 to the minus 5. Now, 10 to the minus 5 is obviously a very small number. You know, it's 0.00001, and so it's obviously a very small number. And because the acid dissociation constant is the product of the products divided by the product of the reactants, so that means if you have such a tiny fraction, that means you have very, very, very little product being produced. And so there is very little of this being produced. That is shown by the fact that the Ka is such a small number. That's how we know that this is a weak acid. So, if we were given a similar problem, just like the previous problem, HCl, we will use 10 to the minus 3 of acetic acid. We saw how 10 to the minus 3 of HCl would produce a pH of 3. Now let's start with 10 to the minus 3 of this substance. So the way we solve these problems is you first write what you find initially. So initially, I'll just put a little I. Initially, we have 10 to the minus 3 of acetic acid, and we have nothing of these other substances. Now note, they will rarely ask you questions in which they will have different ions in the problem. But if for whatever reason you're talking about the dissociation of acetic acid, and let's say for whatever reason they added another substance into the solution as well at the same time, something like the acetate anion, either by using sodium acetate or some other type of solution to provide this acetate anion, well then you would simply write in here whatever concentration of acetate there was. But if the solution is that of acetic acid and that is being added to solution, well then if that's the only ions that are being added, then we have 10 to the minus 3 is the initial, and then there's nothing of the others yet. Then we would look at the change. During the reaction, how will things change? Well, the acetic acid would lose some amount. Remember, it's a very small amount that is turning into the products. But nonetheless, there will be some amount X that would be lost from the 10 to the minus 3. And whatever number of moles per liter is going to be lost here, that would be the amount that would be gained here. That's similar to what we saw with the HCl. And the reason for that, again, is because the relationship in this equilibrium is that for every 1 mole of this, we have 1 of this and 1 of that. Had this been a problem of H2SO4, we would have seen H2SO4, and we could have seen 2 protons being given off and SO4 minus. Because the relationship between H2SO4 and the protons would be 1 to 2, then you would have twice the amount being produced. Of course, H2SO4 is not an example of a weak acid. It's an example of a strong acid. But I'm just trying to underline for you the importance of the relationship in the equilibrium. So, this one would lose X, this would gain some quantity X, and this would gain, again, some quantity X. Therefore, we write initial, the change. Therefore, at equilibrium, what do we end up with? At equilibrium, we would have 10 to the minus 3 minus X, and here we would have X, and here we would have X. So we just add up the columns. Now we want to be able to move on to calculating, well, what would the pH of the solution be? Remembering that the Ka, the acid dissociation constant, is the product of the products over the product of the reactants. Therefore, Ka, which is 1.75 times 10 to the minus 5, is equal to the product of the products, which is going to be x squared over 10 to the minus 3 minus x. So we have a component squared, we have an x, which is to the power of 1. In order to properly calculate or accurately calculate the answer for this, you would need to use the quadratic equation, which is that you'll have to dust it off from your high school memory to use the quadratic equation. However, it is very unusual to have to pull out the quadratic equation for the MCAT exam, though you are responsible to know it. So what happens in this particular problem, in order to avoid using the quadratic equation, keeping in mind that relatively little product is being formed, that means if you start off with 10 to the minus 3 and only 1 in 100,000th of this is going to be released as x, well, 10 to the minus 3 minus x is going to be essentially 10 to the minus 3. It's going to be, this number will hardly be changed because the x that is being subtracted from it is so incredibly small compared to this number. And we know that only because the Ka is such a very small number, 0.00001. So a very small amount of this is going to be given off. And because of that, we can estimate this denominator as just 10 to the minus 3. Once we've done that, now it becomes very easy to solve. It's basic algebra. We have 10 to the minus 3 in the denominator. So then, in order to bring that up, we multiply both sides by 10 to the minus 3. Therefore, 10 to the minus 3 would cancel on the right-hand side of the equation. On the left-hand side of the equation, we would have all of this times 10 to the minus 3. 10 to the minus 3 times 10 to the minus 5, well, all you do is you add the exponents. So you have 1.75 times 10 to the minus 8. That's just adding the exponents, 10 to the minus 5 times 10 to the minus 3. You add the exponents, and it happens to be a negative exponent. And all this is equal to x squared. All these steps you should be able to do very quickly because you don't have a calculator, you've got other problems to solve on the exam, and so you want to be very efficient with your math. Other little tricks is that when you look at this and you have x squared, well, you want to take the square root of both sides. Taking the square root of the right side would give us x, and that is our hydrogen ion concentration is x. So we get x by taking the square root of the right side. Let's take the square root of the left side. The square root of 10 to the minus 8 is simply, it's a square root, is the same thing as the power of 1/2. And therefore the power of 1/2 would cut minus 8 into 10 to the minus 4. Now we need to take the square root of 1.75. And that is simple. The reason it is simple is that you should have memorized the squares of every number between 1 and 15, and you should be able to quickly recognize them. In other words, you should know that 15 times 15 is 225. And you should also know that 12 times 12 is 144. And that's so important for the MCAT exam because, again, you don't have a calculator. And so if you see a problem and one of the answers is 1.44, or they use the number 1.44, immediately you should be suspicious because 1.44 suggests somewhere in your memory, hey, 144, the square root of 144 is 12. Perhaps there's going to be an equation somewhere in this problem that has to do with the square root of this number 1.44 because that makes me think of the number 1.2. So you have to consider these different manipulations so that soon as when I see 1.75 and I have to take the square root of it, well, knowing all the squares between 1 and 15, I recognize immediately that the number 169 is a very close approximation of 1.75. And the square root of 169 is 13. Therefore, the square root, which is a close approximation of 1.75, would be 1.3. So here we have a solution with very few steps. And we have 1.3 times 10 to the minus 4, and that's x, which is our hydrogen ion concentration. So what would the hydrogen ion concentration be? Well, we would have to take the negative log of all these terms. And then we would end up with the negative log of 1.3. And then we'd have the negative log of 10 to the minus 4. Once again, minus 4 comes in front of the logarithm. And then we end up with plus 4 as our pH equals x. Therefore, we have 4 as our pH. X is actually going to be the negative log of x, which is going to be our pH. That would be more accurate. Now we would end up with 4 minus log 1.3. Again, knowing your rules of logarithms, remembering that the log of 10 is 1. The log of 1 is 0. Therefore, the log of 1.3 is some number between 0 and 1, but very close to 0. So it's much closer to 0 than it is to 1 because it's closer to the log of 1 than it is to the log of 10. So we're going to subtract a number very close to 0, so a very small fraction, from 4. In other words, just by looking at the possible answers, you would know that the pH of this solution is going to be 3 point something, very close to 4. Whether it be 3.95 or 3.9 or something, it doesn't matter. They're not, if you have to estimate, they're not going to make the answer so tight that you could not reasonably estimate. So they'll have the answers far enough apart that you could reasonably, rationally estimate. If the numbers are very tight, it means that you cannot estimate. You have to use all your significant figures. Judging from what we've already seen, the pH of an acid must be relatively low, and the pH of a base must be relatively high. This is clearly because if the pH is equal to negative log of hydrogen ion concentration, if you were to have a very small hydrogen ion concentration, for example, 10 to the minus 14 would be a tiny hydrogen ion concentration, because 10 to the minus 14 is 0.0000001, and so it's a very, very small fraction. So 10 to the minus 14, negative log of that would give us a pH of 14. So pH of 14 is essentially basic, very basic. There's terribly small amount of hydrogen ion in that particular substance, and much more POH, which that could be calculated as well, much more hydroxide ion concentration. And so that's one of the tricks that they have on the MCAT exam, is that they'll say, oh, this is a very strong acid, isn't it? Yes, of course. So it has a very high pH, doesn't it? The opposite. Strong acid, very low pH. So you have to have that very clear in your mind. Now let's consider that we do have an acid with its very low pH, and we start adding a base to it. A strong base, like for example, sodium hydroxide. So we're adding a strong base to our acid. Slowly the pH will begin to rise. And it will get to a point where the pH will be basic, because we've added so much NaOH that the pH of the solution would be basic. Well, that is called a titration. And this is a titration curve. And this is how we're going to look at the pH of the solution as NaOH is being added. So clearly, when there's very little NaOH added, the pH of the solution would be very low. As you add a lot more sodium hydroxide, the pH of the solution gets very high. Now, there is this part in the curve, which this type of curve is called a sigmoidal curve, and there's this part in the curve where the curve sort of changes directions. The curve sort of is curved this way here, and then it's curved this way here. Sort of changes directions here. In mathematics, that's called the point of inflection. But for chemistry, this is called the equivalence point. That's the equivalence point. It's the point at which the curve changes direction. The reason why this is an important point is that this is the point at which the number of equivalents of acid and base are equal. That's the equivalence point. In fact, at the equivalence point, we get that the pH is equal to the pKa. To summarize the common things that they will ask you about on the MCAT exam for this, first, they will test to see whether or not you understand that that is the shape of a basic titration curve. And and you will be able to verify that they're not pulling any tricks by looking at what was added. And if it's sodium hydroxide being added, clearly the more you add is the higher the pH. And this is the general shape of the titration curve. The other common point that they like to test is that of the equivalence point. Because once you know that this represents the pKa of the substance, because the equivalence point is where the pH is equal to the pKa, so that value would be the pKa, then you would know where the curve should be depending on what the equivalence point would be of the substance. For example, let's use a strong acid with a strong base. So we have HCl plus NaOH. So hydrogen will give off its proton. NaOH will give off its one equivalent of base. And we have this producing. So we would have H plus OH minus that would just be water, and we have Na and Cl, that's salt, NaCl plus H2O. This is a classic reaction. In fact, many students sort of remember that if you add an acid plus a base, you get a salt and water. That's not 100% accurate, but I think the better or more general statement, and there are exceptions to it, but the more general statement to remember is that if you have an acid plus a base, you get a salt and a neutral compound. And water, of course, is an example of a neutral compound. Now, if you were to look at this and try to design, what is the pH of NaCl plus water? Well, NaCl doesn't have any hydrogen or OH minus ions, and water has an equal amount of H plus and OH minus. That according to the equilibrium constant for water dissociation. So the pH of this solution would simply be 7. So we know that the equivalence point of the curve for the titration curve of HCl for when you're adding increasing volumes of NaOH, the equivalence point would be at a pH of 7. Oxidation states. Oxidation states are a type of electron bookkeeping. They usually represent very easy marks on the MCAT exam. There are just a few basic rules to remember. The first rule is that all elemental atoms or forms have an oxidation number equal to 0. So that includes things like hydrogen, phosphorus, sulfur. Any elemental atom or form has an oxidation number equal to 0. The second rule, any ion has an oxidation number equal to the charge on the ion. For example, if you have iron, Fe2+, therefore its oxidation number would be equal to +2. Third rule, the oxidation number for oxygen is equal to -2. So this is the oxidation number for oxygen in all compounds except in F2O, which of course you recognize fluorine as being the most electronegative element in the periodic table. Its oxidation number is going to be -1. And so we have -2 for fluorine because -1 times 2. So then the oxidation number for oxygen is +2 in this situation. The other exception are peroxides. And in peroxides, for example, hydrogen peroxide, hydrogen has an oxidation number of +1. So we have +1 times 2, that's +2. So therefore, in order to be neutral, oxygen must have an oxidation number of -1. So those are the exceptions. The fourth rule is that hydrogen has an oxidation number of +1. Once again, there is an exception. The exception is the category of substances called hydrides. For example, sodium hydride. Sodium has an oxidation number of +1. Therefore, to remain neutral, hydrogen has to have an oxidation number of -1. You will recognize hydrides, hopefully, from organic chemistry, or you will, because hydrides are powerful reducing agents. Because of that concept of a hydrogen that has a negative charge that would be very attracted to a partially positively charged carbon nucleus. Of course, that will be discussed in organic chemistry in greater detail. Then there's 5. The fifth and final rule is that the sum of oxidation numbers is going to equal to the charge on the substance. So it equals to the overall charge. So let's look at a couple of examples. For example, H2SO4. So using the rules of oxidation numbers, hydrogen is +1, so this is +2. The oxygen is -2. -2 times 4, that's -8. This substance is neutral. There's no overall charge. So all this needs to equal 0. And sulfur, the oxidation for sulfur is our unknown. So plus X. So this is not going to be the most complicated equation you'll have to deal with on the MCAT exam, but clearly 8 goes over to the other side, blah, 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 and then we get X is equal to 6. So the oxidation number for sulfur would be 6. Let's look at another situation, a little bit more complicated, just a little bit. Cr2O7 to the -2. So now we have a situation in which there is an overall charge and it's -2. You may want to pause and calculate what you would provide as the oxidation number for chromium. In order to calculate it, we say -2 for oxygen, -2 times 7, so we have -14, and that's going to equal -2. So we have chromium, -14 equals -2. We bring the 14 over to the other side. We have Cr2 is equal to 12. So that means that the chromium, 2 chromium, is equal to 12. So chromium is going to equal to 6. So the oxidation number is 6. 3 more points to make about oxidation numbers. The first is that oxidation numbers can change from molecule to molecule. Clearly that is the case, as I've shown these exceptions, but we often see that in substances like sulfur. In different molecules, sulfur can have very different oxidation numbers, so you have to calculate them. So they may vary from molecule to molecule. The second point is that they may be fractions. It is unusual, but if you calculate an oxidation number on the MCAT exam and it ends up being a fraction, the first thing to do is to check your work. Make sure you didn't make a mistake. And if you did go through it properly, then yes, you can have confidence knowing that an oxidation number can be a fraction. And the third point is that the oxidation number is not equal to the valence. Now, the valence is what we discussed earlier, which has to do with the outer shell electrons. But the oxidation number that we calculate or develop or memorize, this is not necessarily equal to the valence electrons. Solubility product. The solubility product has to do with weak electrolytes. Many substances, when they're dissolved in water, will ionize. And for example, this is lead chloride, and it produces lead and chloride. Lead, this is Pb2+, and Cl-, 2+, the plus indicating that this is a cation, and Cl- being an anion. Some people remember from high school, it's because anion has An ion, that anions are negative ions. Nonetheless, because these are ions being produced from this solid, the ions can conduct electricity because they're charged. Because they can conduct electricity, they're also called electrolytes. Some substances dissolve readily in water, like strong acids, which we saw before, or NaCl, which is salt, which dissolves into 2 components, which we discussed in terms of colligative properties. Well, lead chloride and many other substances are very weak electrolytes, meaning that they're solid forms, they tend to stay together, and because they tend to stay together, very little will dissolve into water. So because they are weak electrolytes, and because, relatively speaking, the amount of the solid form, and sometimes that solid form will be called a precipitate in water, so because the solid form predominates, therefore it is more or less constant compared to the small amount of product being produced. For this reason, there is a constant called KSP, and that constant refers to the solubility product, meaning the product of the products. So the KSP expression for this reaction would be, just like all the other equilibrium constants, except that we don't have to take the reactant into account because that is more or less constant. So KSP is given by the product of the products. So we have PB and we have Cl. Once again, these square brackets representing the concentration, and this is squared because there is a 2 in the equation, in the balanced equation. So we have PB Cl minus squared. So if we're given the concentrations at equilibrium of PB and Cl, we are able to calculate the solubility product. We expect that the solubility product for a reaction which has a weak electrolyte would be very small, meaning that the KSP would be 10 to the minus something or the other, but it would be a number which is relatively small, 0.00 something or the other. And we expect that because very little product is being produced with these weak electrolytes. We also would expect that the lower the solubility product means the least likely that the substance would dissolve, which means the more likely that the substance would precipitate or to form a solid form when it is in a liquid. The next issue about the solubility product is the word solubility. Sometimes they may ask you about the solubility of a particular substance. The solubility is in moles per liter, and it refers to how much of that substance would actually dissolve. Sometimes solubility is symbolized by S. And if we were to look at this reaction and to talk about the solubility that would occur, then we would symbolize for the lead, we'd symbolize it by S. We'd symbolize the solubility of Cl minus as being S, but it would really be 2S, because whatever the solubility of the lead is, there's going to be twice as much of the Cl minus being produced because of the stoichiometry of the reaction. So we have 2S. So that when we were to write the KSP expression based on the solubility, this is based on the concentrations, the actual concentrations. But if we were to be asked about the solubility, then we would write S representing the lead, and then that would be multiplied by, for Cl minus, we would have 2S as being the amount. So we would have 2S squared. So this is KSP would equal to S times 2S squared. And that would be equal to, well, we would have 2 squared, which is 4. And then we have S squared, which is S squared, times S, so that is cubed. So we would get KSP equals to 4S cubed. So if we had to calculate the solubility for the reaction, if we were given the KSP, then we would be able to work it out based on this calculation that the KSP would be equal to 4S cubed, where S is the solubility. Hess's law. Hess's law refers to certain manipulations that can be done concerning heats of reaction. You have to imagine that in chemistry, many different types of reactions can occur. Associated with these reactions are certain changes in terms of energy or heat that may evolve from the reaction or may be required for the reaction to occur. The changes in these heats that of the reaction is the delta H of the reaction. We mentioned before that if energy or heat leaves the reaction, is released from the reaction, that's an exothermic reaction. And if energy or heat is required for the reaction to occur, that is an endothermic reaction. The delta H for an endothermic reaction is positive. The delta H for an exothermic reaction is negative. Because these different chemical reactions have different delta H's, but the delta H's are always dependent upon the initial and final circumstance of the reaction, they are called state functions. That means it doesn't matter how the reaction occurred, it doesn't matter what happened in between the final and initial circumstance for the reaction. It only depends on the heats initially and finally, and that provides us with our delta H for the reaction. So both chemical reactions and the formation of chemicals have heats associated with them. The formation of a chemical will have a heat associated, and that would be called delta HF, or the heat of formation of that chemical. Hess's law comes from the fact that delta H, or heats of reaction, are state functions. And because they're state functions and they only rely on the final and initial circumstance of the situation, then we can do certain manipulations. For example, if you were given a reaction and you were given the heats of formation of the chemicals in that reaction, using Hess's law, all you would need to do is take the sum of the heats of formation of the products, subtract the sum of the heats of formation of the reactants, and you would have the overall delta H of the reaction. So these reactions have delta H's associated with them. Here, this reaction has delta H1. This reaction here with calcium hydroxide has delta H2. Now let's say that you were provided these two reactions and you were provided with the actual values of their delta H. Then you were provided with another reaction and were asked, what is the delta H of this reaction based on your knowledge of the delta H of these other two reactions? You may want to consider how you would respond to that. So you may want to pause now. Well, the way you would do this is you would line up the equations that you were given with the equation that you want. So we have 2H2 here. Well, that's also on the left side. We have oxygen on the left side. That's also on the left side. And we have water. So water is not there. So water has been eliminated somehow. But the rest of the equation lines up exactly like this. So we're not going to alter the way the first equation is written. Now the second equation, we have calcium hydroxide on the left side. But over here we have calcium hydroxide on the right side. Furthermore, we have 2 times it. Then we have calcium oxide, which is on the left side. So clearly we're going to have to switch the equation around. And we're going to have to multiply this equation by 2. And that seems to work out quite nicely, because then we'll have 2 times water. And when we switch this over to the other side, and we have 2 times water on the left-hand side, we have 2 times water on the right-hand side of this equation. So when we add up the equations, we would be able to cancel the 2 times water from the both sides. Then we would end up with exactly this equation. So the only manipulation we would need to do before we add these 2 equations is we would need to flip this over, which is to invert the equation, and we would need to multiply through by 2. To invert the equation, we therefore would turn this delta H2 into negative delta H2, and then to multiply it, the equation through by 2, we also multiply the delta H2 by 2. In other words, the delta H for the given reaction, the total delta H or enthalpy for the given reaction would be given by delta H1, because that reaction was not changed, minus 2 times the value for delta H2. And this would give the overall enthalpy for the final reaction. We will now examine the rate law. The rate law refers to the rates of reactions. Now you could consider the rate of reaction sort of like the speed at which the reaction occurs. Normally when you think of speed, you might think of miles per hour or meters per second. But in a chemical reaction, because they are chemicals, we are really talking about the concentration per second. In other words, moles per liter per second. That represents the rate of the reaction. Now here's a typical reaction. So we have reactants, say A and B, whatever they may be, and then producing certain products. Now we can write the rate law for the reaction. The rate law is that the rate of the reaction in moles per liter per second is equal to the rate constant K times the concentration of the reactant A times the concentration of the reactant B. Now each reactant is raised to a power. The exponents are not necessarily equal to the stoichiometric coefficients. Because if you look at this reaction, clearly the stoichiometric coefficient for A must be 1, and the stoichiometric coefficient for B also must be 1. And so it is not necessarily the case that X and Y must equal 1, because in the balanced equation, the stoichiometric coefficients equaled something in particular. You can't make that assumption. What you need to do is, in order to determine what the exponents are, you have to examine a particular experiment. Then by looking at the experiment, you can determine what the exponents must be. For example, in this experiment, you examine what the initial concentration and initial rates of reactions are, and then by examining the rate law, we will be able to determine what the exponents must be. So, in order to do so, we look at an experiment in which one of the particular reactants will not change. Then we look at what happens when the other reactant does change, and we examine its effect on the rate of reaction. So if we look at the first two experiments, in the first two experiments, the concentration of A remains constant. It is 0.10 moles per liter, and then it is 0.10 moles per liter. So it does not change in the first two experiments. Now in the first two experiments, the concentration of B doubles. It goes from 0.10 to 0.20. Now we have to examine what is the effect of doubling B while A is constant. What is the effect of doubling B on the rate of the reaction? Well, the rate of the reaction goes from 1.23 times 10 to the minus 3 up to 2.46 times 10 to the minus 3. So it doubles. Doubling B leads to doubling of the rate of the reaction. So if B doubles, if you have 2 times B, you end up with 2 times the rate of reaction. So it's a perfect 1-to-1 ratio in terms of what happens. In terms of having a factor of B, you have the same factor for the rate of the reaction. Therefore, the exponent for B must be equal to 1. So we've worked out what the exponent of B must be because we were able to keep A constant and see how the rate of the reaction would change as B changes. Now, let's keep B constant, change A, and see what effect it will have on the rate of the reaction. So A changes when we compare experiment 1 and experiment 3. We go from 0.10 up to 0.20. So we doubled the concentration of A. B remains the same, 0.10 to 0.10. So while B remains the same, we double A. Let's see the effect on the rate of the reaction. The rate of the reaction goes from 1.23 times 10 to the minus 3 up to 4.92 times 10 to the minus 3. In other words, the rate of the reaction quadruples. It is 4 times. So we double A, we get 4 times the rate. So if we had 2A, something happens with this exponent to make it 4 times. Clearly, x must be equal to 2. So we have x is equal to 2. And that means 2a, the 2 is squared, making 4, and therefore we have 4 times the rate. Now we can see how you would develop the exponent for any kind of reaction that they would provide. We have now noted that it's 2 for A and 1 for B. There's other ways of saying this. The most common way of saying it is that the rate of reaction with respect to A is second order. The rate of the reaction with respect to B is first order. So you can talk about the individual components and what the rate of the reaction would be with respect to that component, and that is equal to its exponent. If you want to talk about the overall rate of the reaction, then the overall rate of the reaction will be the sum of the exponents. So it would be 2 plus 1. The overall rate of the reaction would be third order. It's a third order reaction. This concept of the order of reaction will also come up later when we look at organic chemistry. Now, a couple of other things to note about this. 1, imagine what you would give as a response if you changed the concentration of A, you doubled A, but the rate of the reaction didn't change. You tripled A, the rate of the reaction didn't change. Whatever you did at A, the rate of the reaction didn't change. What would you consider to be the order of the reaction with respect to A? You may want to pause and consider your response. Well, the order of the reaction would be 0. The exponent would be 0, because no matter what you do with A, it doesn't affect the rate of the reaction. So you would say that the rate of the reaction is 0 order with respect to A. But in terms of the overall rate of the reaction, once again, you would add up all the exponents, whatever they may be, and that would give you the overall rate of the reaction. So that's one point. The other point is, be careful on the MCAT exam with your exponents. Now here, they are all 10 to the minus 3. But they might put one of the exponents as 10 to the minus 4 or something like that. They'll do that very commonly on tables, whether it be for general chemistry or any of the basic sciences. The important thing for you to do, just to keep everything consistent in your table, just cross out whatever they have which is different and rewrite it on the side in a way which is consistent with the rest of the table so that you don't make any mental errors. So for example, if 1.23 was 10 to the minus 4, then in order to make that 10 to the minus 3 like the others, we would just write 0.123 times 10 to the minus 3. Some students like to remember that if you add a number up here, then you subtract a particular part of the decimal. You move the decimal more to the left. And if you subtract a number from here, say you add minus 1, then you move the decimal to the right. So if you have 10 to the minus 4, then it's like moving the decimal to the right. So 12.3 times 10 to the minus 4 would be equivalent to 1.23 times 10 to the minus 3. That kind of comfort with exponents is important for the MCAT exam. Well, we've looked at reactions and the rate of reactions. We also know that associated with reactions is a change of energy or a change of heat, which we call delta H, the enthalpy. And this produces reactions which can be endothermic or exothermic. The next thing that we will look at are the potential energy diagrams that are relative to this particular endothermic or exothermic reactions. You are now looking at potential energy diagrams. Now consider the complicated chemical reaction A plus B goes to C plus D. Therefore, A plus B would be our reactants, and C plus D would be our products. Now, of course, there are changes in energy during reactions. And so consider these two different possibilities. To begin with, we start with a particular energy for our reactants. We end off with a particular energy for our products. So in one possibility, the energy of our products is much lower than the energy for the reactants. So here's the reaction progress, meaning as the reaction is occurring, there is a change in energy. The change here is that A plus B has much more, the reactants have much more than the products. So therefore, energy must have been released in the reaction. If energy is released or energy exits a reaction, we call that an exothermic reaction. And that means that the delta H for the reaction, the enthalpy for the reaction, would be negative. So if you were to look at the difference between the energy of the reactants and the products, well, that would be about like this. That would represent our delta H for the reaction. So that's the delta H, which would be the difference of the energy of the reactants and the products. And in this particular case, this is clearly an exothermic reaction because the products have less energy, which means that energy must have been released in the reaction. Therefore, delta H must be negative. If you have difficulty remembering that delta H must be negative in this type of reaction, remember that the delta H will be calculated by the total energy of the products minus the total energy of the reactants. So I don't know what the energy of either is, but I do know that the energy of the reactants is clearly here higher than the energy of the products. So if you had products minus reactants and the reactants is a larger number, then you end up with a negative number. So the delta H here would be negative. In this particular reaction, we start off with a low energy for our reactants. We end up with a higher energy for the products. So in other words, energy was gained. Energy must have been added for this reaction to occur. That is an endothermic reaction. That means that the delta H must be positive. And the delta H would be, of course, here, would be the difference between the reactants' energy and the product's energy. So this would be the delta H of this reaction. Now the reaction sort of, during the reaction progress, as it's proceeding, sort of goes up into this lump and then it comes back down. Well, this area up here is what is called the activated complex. In organic chemistry, we more commonly call it the transition state. This is the time and place where the bonds are being created and broken in order to make the new products, because these A and B is going to react together, and therefore there's going to be new bonds that are being created and destroyed and so on. And so this is occurring at the activated complex at the top of the curve, both here and both here. And then we get the particular products being formed. Now, the difference in energy between the reactants and the activated complex is called the activation energy. So that's between here and here. This is the activation energy, and it is symbolized by an E with a small letter A, a subscript A. That's the activation energy here. And so it is the difference of energy between the activated complex and the reactants. So here's EA for this reaction. When you have a small EA, it is more likely that the reaction will occur spontaneously. When you have a large EA, it is unlikely that the reaction will occur spontaneously. So here is a reaction. We know we start with a high energy, we end up with a low energy. Energy is released. It is more likely that this will occur spontaneously with its low activation energy than this reaction with this huge activation energy. In order to get over this mountain or lump or hill, it would be very difficult, if not impossible, for this to occur spontaneously at least. So it would require energy to help bring it over into the products. Now there are ways that the activation energy can be reduced. In particular, a catalyst is a compound that could be added to a reaction. It won't be used up in the reaction, meaning it is regenerated at the end of the reaction, yet it increases the rate of the reaction. The reaction will occur faster by using the catalyst. The catalyst will indeed reduce the activation energy. There might be a series of steps, but each step will have a lower activation energy overall in order to produce the products. So in summary, if you were to compare this particular curve to what effect a catalyst would have on it, well, what it would do is that you would start here with your reactants, and then the catalyst would create a curve, something like this, so that the activation energy would be lower. The activated complex will be in a lower position, a lower energetic position because of the use of the catalyst, and therefore the activation energy will be lower. So these are common curves that you may see on the MCAT exam, so it's important to quickly recognize them and to know what your delta H is, your activation energy is, and the particular effect that a catalyst can have on these curves. Now we're going to move on to electrochemistry, and in particular, electrolysis. Electrolysis. If you open your mind to electrochemistry, you'll find that it is a very straightforward subject. Here we have a battery. It is connected by some wires to electrodes. These electrodes are in some sort of fluid. Now let's say that that fluid was molten salt, meaning that it's Na plus and Cl minus in solution only. So all we have is Na plus and Cl minus in solution. Then we have these electrodes, and these electrodes are, say for example, an inert substance like platinum, so that the electrodes are not going to get involved in the reaction. So we're going to be looking at what happens to NaCl as a result of having these electrodes here. Then we have the battery, and you may remember from physics that as soon as you see a battery, your Pavlovian reaction is to look at the large electrode and to label that as positive, and to look at the smaller electrode and label that as negative. So this is the positive electrode, and this, because there are electrons emanating from this terminal of the battery, this would be negative. So the battery is going to result in a charge difference in the electrodes. So that's fine. Now in solution, we have Na plus and Cl minus. Well, imagine that because electrons are flowing into this electrode, making this electrode more and more negative, sodium, which is positively charged, is going to be attracted to the electrode, which is negatively charged. Chloride, which is negatively charged, will be attracted to the electrode, which is positively charged. Now the word electrolysis. You may remember from biology that the word lysis often refers to something which is being split apart. Well, electrolysis is the word. And the reason is because the battery is providing electrons and creates a charge difference. That's the electro part. The lysis is the fact that before the charge went to the electrodes, sodium and chloride were just hanging around in solution, kind of ionically bonding. And so sodium and chloride was involved in an ionic bond, very relaxed, nothing special going on. But soon as these powerful charges come in to the electrodes, then the sodium and chloride atoms that were being bonded ionically suddenly are being torn apart, separated, such that the negatively charged chloride goes to the positive electrode and the positively charged sodium goes to the negative electrode. So this is electrolysis. Now, there is a mnemonic that you can memorize that will help you remember where the anode and cathode are. That mnemonic is Leo is a jerk. I suppose this would be more correctly pronounced Gurk, but that's not a great mnemonic. Anyway, so Leo is a jerk, and that stands for loss of electrons, oxidation is anode. Gain electrons, reduction, cathode. So as sodium comes here to this electrode, what's going to happen? Well, the Na plus is going to gain an electron, so plus an electron, and that's going to give us sodium, which is solid. So you literally have the cation, sodium, coming to the electrode, gaining an electron, and because it gains an electron, because sodium now becomes solid, it plates onto the electrode. It just literally becomes solid right onto the electrode. So this reaction, because it's a gain of electrons, that means sodium is being reduced, and that means that this is occurring at the cathode. So this half reaction, it's called a half reaction because it's only part of the reaction that is occurring. This half reaction is at the cathode. Let's look at the other side. Cl minus is being attracted to the positive electrode. When Cl minus goes to the positive electrode, it gives up its electron. All this based on opposites attract. So Cl minus goes to the other electrode. It gives up its electron, and so it's going to become chlorine, Cl2. So this is going to be actually 2Cl minus, and that's going to give 2 electrons, and that provides us Cl2, which we know is a gas. So in other words, because it provides Cl2, we know that at this electrode, chlorine will bubble out as a gas, and at this electrode, sodium will be plating onto the electrode. So when we do these reactions, these are half reactions, we want them to balance. We want the electrons to cancel. So we multiply through here by 2. Then we add the reactions, and then we end up with 2 Na plus 2 Cl minus, giving us 2 Na plus the chlorine gas, which is released. So now we have clear what our cathode and our anode, where they are, why this occurs, and it also provides interesting information about the idea of electrolysis. You can imagine what a wonderful invention this was in order to save industry money, because beforehand, if you needed to have a gold-plated jewelry, for example, someone would literally melt the gold and paint it onto the piece of metal. And so you would have millions of atomic layers of gold onto that substance. Well, could you imagine how much industry could save if they could take gold, which is Au2+, put gold into solution, and then create a negative electrode out of the metal or jewelry, and then let the gold ions just plate onto the jewelry to the point that it just looks gold. And then they sell that to you, and it lasts a few months. But you could imagine that by doing this, you can just put a minimal number of layers of atomic gold rather than millions of layers, and it would save a tremendous amount of money for them, of course. Nonetheless, let's move on to another slightly more complicated electrochemical cell. This is an electrochemical cell. Notice the highlights. To begin with, this is a zinc electrode. So it is made out of zinc, solid zinc. This is a copper electrode made out of solid copper. Here is a wire connecting the two. This is a voltmeter, meaning all it is, it doesn't drive, it doesn't give energy, it's not a battery. All it is it tells us how much voltage there is in the system. So it just provides a number. And then we have two containers with fluid. This container has zinc sulfate. This container has copper sulfate. Both of these are containers that contain water, so this is zinc sulfate aqueous, meaning it is an electrolyte, and so it will dissociate. So zinc sulfate really means to us that there is Zn2 plus and SO4 double minus floating around in solution. And here the same, there is copper 2 plus and sulfate double minus that are floating around in solution. This part of the electrochemical cell is called the salt bridge. In it, there are ions of salt. There's Na plus and Cl minus in the salt bridge. Thus, they can move from one container to another. This cell, which is called an electrochemical cell, is also called a galvanic cell. Now, you just have to take one thing for granted from me, and then after that, everything just sort of flows from that one point. And that is that if you were to hook up zinc to copper, it so happens that zinc has a tendency to give electrons compared to copper, and copper has a tendency to receive electrons compared to zinc. Therefore, just hooking the system up leads to electrons leaving the zinc, attracted to the copper, going through this system, and we'll discuss later how you could know this, but nonetheless, the electrons are passing away from the zinc, so we have electrons moving in this direction, electrons moving along here, electrons moving into this electrode. So this electrode will become negatively charged. Now, as a result of the fact that this electron is becoming negatively charged, and in the presence of copper sulfate, meaning Cu2 plus in solution, the Cu2 plus in solution will become attracted to the negatively charged electrode, and it will plate onto the electrode. It will become solid. So in other words, we have Cu2 plus electrons, 2 electrons, will give us Cu solid. So copper solid is being produced here because there's a gain of electrons. Therefore, copper is being reduced, and this is occurring at the cathode, given by Leo as a jerk. So we have copper, which is plating on to the electrode. But something's wrong with this picture. And that is that there was balance. It was a solution that had ions in it, but there was an equal amount of ions, cations and anions. So now that copper is plating onto the electrode, then the balance is disrupted. The overall charge of the container will start to become negative, because as the Cu2 plus plates on, then you're going to have a net SO4 double minus just hanging in solution. Well, you've never been to a chemistry lab and created a cup full of positive ions or a cup full of negative ions. It would always be balanced. It takes an extraordinary amount of energy in order to produce a container filled with one or the other charges. So this would not occur, certainly not spontaneously as it is happening here. Therefore, in order for this reaction to continue, in order to make sure that it's not going to just stop, you need a salt bridge. And the reason is because in here we have Na plus, in the bridge there's Na plus and there's Cl minus inside of the bridge. So as you start getting a net SO4 double minus here, the sodium in the salt bridge will start to race in to the solution on the right side. In other words, it helps to maintain neutrality. That's one of the objectives of this salt bridge, to maintain neutrality. Instead of having a cup full of negative ions, which wouldn't happen anyway, but in order for the reaction to continue, the sodium comes flying in here and rescues the situation. Now let's go on to the other side. The zinc is giving up electrons to the copper. Every time the zinc gives up electrons, releases electrons, the solid zinc in the electrode becomes Zn2+, because it gave up electrons. So it becomes Zn2+, and therefore it falls into solution. Well, once again, you can't have a container with a net positive charge in it created spontaneously. So as it falls into solution, creating a net Zn2+ in the solution, Cl- from the salt bridge comes down and rescues the situation and helps to bring back balance of charge or neutrality in the container. So the salt bridge does two things. One, it helps to maintain neutrality. And the second thing the salt bridge does is it completes the circuit. After all, watch the flow of negative charges. Zn giving up electrons, falling into solution. So we have negative charges here, flowing through the wire. Negative charge, 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 and then through the salt bridge. Negative charge, negative charge, negative charge, negative charge. And so the circuit is completed by the movement of charges because there's the presence of the salt bridge. Now you know why this is called an electrochemical cell. Here, the electrons carry the charge, and here the chemicals carry the charge. Electrochemical cell. It's also called a galvanic cell, and after all, this is the basis of modern-day batteries. A couple of more points. First, the half reaction of the zinc. So we looked at the copper half reaction. The half reaction of the zinc would be Zn solid going to Zn 2+ plus 2 electrons. This is a loss of electrons, therefore it's being oxidized. Loss of electrons, oxidation is the anode. So if you were to add the half reactions, you would get Cu2 plus Zn, giving us, because the electrons would cancel, that gives us copper solid plus Zn2 plus. So I have a question for you. Why is it that an electrolytic cell and an electrochemical cell are opposites? You may want to pause and consider your response. The reason is, in an electrolytic cell, we saw that we put a battery there. Then the battery pushed charges into the system, and then it broke apart the chemical. So an electrochemical cell requires energy, a battery, whereas a galvanic cell or electrochemical cell is a spontaneous reaction. This occurs spontaneously, therefore it actually is a battery. It does not require a battery in order for this reaction to occur. So that brings up the point of why is it that zinc was able to do this? Well, there are certain tables that would be provided to you that are called reduction potential tables. And what those tables show are reactions as reductions. So they will show a reaction like this as a gain of electrons. Then that reaction would have a value, a potential associated with it, and it's called the E0 value. This potential, this reduction potential value, the 0 represents it is at standard conditions. That is important for the following reason. Because it's standard conditions and that the concentrations are standardized, if you were to multiply your reaction through by any kind of coefficient, like we did before, where we multiplied through in order to get 2 electrons to balance our equation, you do not multiply the E0 value by a number because the concentrations are standardized. It is not like enthalpy, delta H, where when you multiply through by a particular stoichiometric coefficient, you must also multiply your delta H value by that number. But here, if you multiply through, you do not multiply the E0 value because the concentration is already standardized. So that's one important point. The other thing is that they're all written as reductions in these reduction potential tables. The way it works is that the E0 value, which is most positive, is the E0 value which is most likely to occur spontaneously. If you have a negative E0 value, you do not expect that that would occur spontaneously. So when you switch your reaction around, like zinc has been switched around, it's not written as a reduction, it's written as an oxidation. So when you switch the reaction around, you change the E0 value from positive to negative or from negative to positive. And then when you add 2 equations, then you just add your 2 E0 values to get the final E0 value of the reaction. Clearly, the final E0 value of a reaction like this would be positive because I've already mentioned that this is a spontaneous reaction. Welcome to Organic Chemistry for the MCAT exam. Organic chemistry is the chemistry of carbon-based compounds. Of the 4 basic sciences that you must know for the MCAT exam, organic chemistry is the subject that you need to memorize the least amount of information. The reason for this is that organic chemistry is based on certain basic rules. Once you've memorized those rules and you've understood those rules, then the rest is just play. First, let's learn some definitions. Composition. Composition refers to the molecular formula. Constitution refers to the order and kinds of bonds. Isomerism are different compounds with the same composition. For example, let's look at C4H8. C4H8. We can look at this particular compound. Now note, this is C4H8. 1, 2, 3, 4 carbons. Number of hydrogens, 3, and 3, 6. 2 more here, that's 8 hydrogens. So this is one way that we can look at a compound which has the molecular formula C4H8. Let's look at another compound. CH3, CH, we have a double bond here, CH3. So here we have 4 carbons, 1, 2, 3, 4 carbons, 3 hydrogens, 4, 5 hydrogens, another 3, 8 hydrogens. 2 different compounds, same molecular formula, same composition. Therefore, these are isomers. C4H8 can also be represented by certain geometric figures. For example, this looks just like a box, but the routine in organic chemistry is that every corner of a geometric figure represents a carbon. Then you just need to keep in mind from your general chemistry that carbon bonds 4 times. Carbon has 4 valence shell electrons available for bonding, and therefore it can bond 4 times. So to begin with, we have 1, 2, 3, 4 carbons. Then we have to put in our minds that because carbon is bonded 4 times, we have to add in our minds a hydrogen to make it such that each carbon is bonded 4 times. For example, at this corner here, carbon would be bonded to another carbon up there and another carbon over here. So then clearly this carbon must be bonded to 2 other hydrogens that are not noted. And in fact, that's the way it is in organic chemistry. Sometimes with geometric figures, the hydrogens will not be noted because they're implied. No other atoms are implied, only the hydrogens. So that if you see that carbon is bonded to 2 twice here, therefore there must be 2 hydrogens here, 2 hydrogens here, 2 hydrogens here, and 2 here. Therefore we have 1, 2, 3, 4 carbons, and we have 2, 4, 6, 8 hydrogens. So here is a third way of representing a compound, separate compounds with the same molecular formula or the same composition, C4H8. And here's another one. So here we have a triangle. Once again, 1, 2, 3 carbons must be in the triangle because each corner of the geometric figure. Then we have 3 carbons here and then the 4th one there. So we have our C4. Then in terms of H8, well, clearly there must be 2 hydrogens here at this corner because there's only 2 bonds for this carbon. 2 hydrogens here, so we have 4 hydrogens. 3 here, 7 hydrogens. Over here, the carbon is bonded once to another carbon, once to a carbon here, once to a carbon there. That's 3 times it's bonded. Therefore, there must be 1 hydrogen bonded to this carbon. So that makes C4H8. So once again, we've looked at 4 different ways, different compounds with the same composition, the same molecular formula. Therefore, these are all different types of isomers. Our next definition is that of conformation. Conformation is the shape due to free rotation about sigma bonds. You may remember from your general chemistry review that sigma bonds are single bonds. So let's look at this substance, this organic chemical, and it has 2 carbons. Therefore, its name starts with eth, since 1 carbon starts with meth, 2 with eth, 3 with prop, 4 with bute, 5 pent. So this starts with eth because there's 2 carbons, and it's an alkane because it's a simple hydrocarbon. and it's fully saturated, which meaning the carbon is bonded to the maximum number of different atoms as possible. In other words, there are no multiple bonds. So this is ethane. And in this compound ethane, what we're talking about is that the single bond between the carbons, there is a free rotation. There is turning. If you can imagine an axle between wheels, and the wheels can turn independently, that is conformation. So there's free rotation about this single bond. Now there's many ways to represent this free rotation. One is called a sawhorse projection. In a sawhorse projection, we have, say, our carbon here, and this carbon would be this one. I'll be representing over here. We'll have hydrogen up here. We'll have another hydrogen that looks like that. Another hydrogen coming out sort of in a triangle shape like this. Now, this hydrogen is going to be representing this one, this hydrogen, this one, this hydrogen over here. Then we go back. This is our bond between the carbons. We have another carbon here, a hydrogen here. Then we have a bond here to a hydrogen. Then we have this sort of triangle shape again, bonded to another hydrogen. Now, the routine here is that when we see a broken line in organic chemistry, it means this hydrogen is going into the plane. And this solid triangle means this hydrogen is coming out. Because though we write these sort of sticks and lines and so on, the truth is these are three-dimensional molecules. And that because carbon is bonded to 4 different atoms, we know that carbon must be in the center of a tetrahedron. And a tetrahedron is sort of shaped like a pyramid, in which, so you have the carbon here, you have a hydrogen above it, and then you have this triangle as a base, in which there's a hydrogen towards you, there's a hydrogen away from you but off to the side, and then another hydrogen away from you but off to the side. So if we have this tetrahedron represented here, where the line is in the plane, this broken line means that it's going away from you. This one is coming towards you. Then we have back here another CH3 group. CH3, a CH3 group, because C is just one carbon. That means it's a meth. And then it can't end in ane, because an alkane would be CH4. It would be fully saturated. This is CH3. Whenever you have an alkane minus 1 hydrogen, it ends in -il. So then we have a methyl group back here, CH3 group. And so we see that here as well, the hydrogen goes into the plane. This hydrogen comes out of the plane. And so we have our 3-dimensional image of the molecule. Now, when we look at this sawhorse projection of the ethane molecule, note that if I'm looking at the molecule from this angle, then for me, this hydrogen would be in front of this hydrogen. This hydrogen would be in front of this hydrogen, and this hydrogen would be in front of this one. Because this hydrogen is in front of that hydrogen, we say that it eclipses this hydrogen. So this is called an eclipsed conformation. There is yet again another way that we can look at this eclipsed conformation. And that is, if I were to be looking at it just like this, sort of head-on, like this, at this angle, what I would see is I would see this carbon, but I wouldn't see the carbon behind it. I would see this hydrogen, but I wouldn't see the hydrogen. And so we can come up with something called a Newman projection. And in the Newman projection, which is drawn like this, we would have the carbon in the middle, then we would have a hydrogen up here, we would have another hydrogen here, and we'd have another hydrogen there. So this is as if I'm looking at it from this particular angle. I can't see the hydrogen in behind, and I can't see the hydrogen behind that one. This is an eclipsed conformation. So This one is eclipsed. Now, the idea behind conformation is that there's free rotation about the sigma bond or the single bond. Therefore, let's keep this CH3 group the same, in its same conformation. Let's spin the one behind it around and see what would happen. Well, if we were to just spin this one around, well, we would have basically the same thing in front, which is broken line here, and then we have the solid triangle here of a hydrogen. Then we have the bond going in behind, as this one does. But now we're going to spin this. Okay, so then we're going to have carbon here, but then we're going to have one bond going up this way. We'll have a hydrogen which has been spun around, and then this will go this way, and then we'll have another one going from below. In other words, it's the same molecule as this one, same CH3 group as here, but this one has been spun around. This one has been twisted. So it's been twisted around, and so if we were to look at a Newman projection of this as it's been twisted around, well, if I'm looking at it from this angle, I will see hydrogens off to the sides, because the hydrogen that was just in behind, now by twisting this, I can actually see it off to the side. So the Newman projection here would be something like this. We'd have the one in front, the hydrogen in front. We'd have this hydrogen in front and this hydrogen in front. But this is the CH3 group in front. That hasn't changed from this. But the CH3 group in the behind has changed. So now we're going to see off to the sides, we're going to see some hydrogens. And this is called, because it's sort of twisted, it's turned off to the side, this is called a staggered conformation. So this is staggered. Now, in terms of room temperature, these molecules at room temperature, some might ask, well, what do you have? Which one do you have, preferentially or often or whatever? And most organic chemistry, more than 90% of organic chemistry for the MCAT exam, comes down to basic electronics, meaning opposites attract and like charges repel. 90% of organic chemistry will come down to those types of basic concepts. And so, based on that knowledge, that opposites attract and like charges repel, keeping in mind that around these hydrogens would be a shell of electrons. And because there are electrons in these shells, there will be a repulsion between these electrons. So there's electron shell repulsion. Because electrons are negatively charged, they're the same charges, so they want to be as far apart as possible. So these hydrogens, one is behind the other. Now they're a certain distance apart, but they're behind each other. They would like to be as maximally apart as possible. So if this twists, therefore, one would not be right behind the other. It'll be behind, but then at an angle. And therefore, it would be maximally apart. Therefore, the staggered conformation, as it twists, where you get the hydrogens off to the sides, this would be a situation that they would be maximally apart. Therefore, a staggered conformation is more stable than an eclipsed conformation. However, at room temperature, which they can always try to apply as a trick in the exam, do you get eclipsed conformation? Most definitely, because the definition of conformation is that there is free rotation about the single bond, or the sigma bond. And because there's free rotation, there's continual movement at room temperature along this bond. And so it is important to keep in mind that yes, you do get changes of all the different conformations between eclipsed and staggered, including eclipsed and staggered conformations. and you get all of these movements at room temperature. However, at any given moment, most molecules will be in a state which is staggered or similar to it, because most molecules will tend towards the more stable conformation, but all the different other conformations are still possible. Our next definition is that of configuration. Configuration refers to the permanent three-dimensional shape of a molecule. For example, let us look at this particular molecule. First, we'll name it. There are 1, 2, 3, 4 carbons, therefore it must start with bute. There is a double bond, therefore it is an alkene because it's the hydrocarbon with the double bond, therefore this must be butene. In order to be precise, we need to name the carbon where the double bond is attached to. So this would be the first carbon, because we can name any carbon in the link starting with the lowest number, which would be attached to the double bond. So here we have 1, 2, 3, 4 carbons. 1, 2, at the second carbon there is a double bond. Therefore this substance is called 2-butene. Now as we're looking at 2-butene, we see the triangular shape suggesting that this CH3 group or methyl group is projecting out of the plane. This hydrogen is out of the plane, and this methyl group is into the plane, and this hydrogen is into the plane. Each carbon would be at the center of a triangular shape. You may remember from general chemistry that that means there is an sp2 hybrid bond, meaning there is one sigma bond, and there is a p to p bond, which means there is one pi bond. So this is the molecule. Now note that this methyl group and this methyl group are on opposite sides of this double bond. When we have the main substituents meaning the attached group at a particular functional group. So when we have the main substituents, also called ligands, that are attached to a particular area, when they're on opposite sides of a double bond, we call this trans. So this is trans configuration. Now we're going to look at a substance which has the identical molecular formula. So here we have the same molecular formula. This is indeed 2-butene. Now this time the two methyl groups are on the same side, so they are the substituents. Why? Because once again hydrogen presents barely any importance. Just like when we look at geometric figures, we have to add the hydrogens. Sometimes they're not even written in. Well, so we're not looking at hydrogens as substituents or ligands, but these methyl groups certainly would be regarded as substituents. Both of them are on the same side of the double bond. They're not on opposite sides of the double bond. This configuration is called cis configuration. So to be precise, this compound is called cis-2-butene, while the other is called trans-2-butene. Now, in terms of stability, which of these molecules would be more stable? Well, once again, it comes down to electronics, and here's CH3, which you have to try to imagine in 3 dimensions. There is a ligand here. It's a group. There's a group here. And here, there's just little hydrogens. And of course, hydrogens are, well, it's the smallest atom on the periodic table. And so we have this small atom here, but we have this large group here, a large group here. And on the outer surface of these groups are electrons. And therefore, these electrons want to be as far apart as possible. So if you have 2 large groups here, obviously there's going to be some electron shell repulsion in the cis configuration. Then when we look at the trans configuration, where we have a large group here, the other large group is way on the other side of the molecule. And so they're maximally apart. Therefore, the trans configuration is more stable. Now, do you get a twisting and turning along this double bond so that you can go from this trans configuration to the cis configuration, because if you twisted this, brought the CH3 group down, then you would have the cis configuration. Does this occur at room temperature? Absolutely not, because to go from the trans to the cis, because there is a double bond, and the double bond restricts, in fact, it prevents free rotation. By having the double bond, it prevents free rotation about this bond, and therefore you cannot go from the trans form to the cis form without a chemical reaction. It's not something that occurs spontaneously. So trans 2-butene and cis 2-butene are independent and have different properties because of the fact that they do have the same composition, but the way that the molecule arrange is different, and therefore these are two isomers. The fun in organic chemistry can be found in the reaction mechanisms, and that is to look at the way different organic chemicals interact. And in order to do that, we need to continue with our definitions. Our next definition is that of a nucleophile. A nucleophile is nucleus-loving. That is literally what the word means. So a nucleophile is a substance which is attracted to the carbon nucleus. Of course, a nucleus is composed of protons and neutrons, and therefore a nucleus has a positive charge. So something that is a nucleophile must be in itself either negatively charged or partially negatively charged. There are electrophiles, which is obviously the opposite, because an electrophile would be electron-loving. It is something that is attracted to electrons, and therefore in itself it must be positively charged or partially positively charged. Then there are leaving groups, and leaving groups are substances that are bumped from the carbon nucleus. So that's the best definition or clearest definition that I could provide, but it would be even more clearer by looking at some examples. Now let's work out this particular reaction. First, we'll name the compounds. So here clearly we have sodium, and here, well, a common thing in organic chemistry is that when you see a compound that you're not familiar with, think about compounds that you do know that are very similar, and then it will help you to understand the properties and even the naming of these compounds quite often. For example, if you've never seen this compound before and you don't know how to name it, well, consider the following. If this compound was this compound, you would know what to say. This is sodium hydroxide. It's a very famous compound, of course, in both organic and inorganic chemistry. Sodium hydroxide. So now, if we were to look at this substance, we have sodium, and we have this, and oxide. So this is sodium hydroxide. This is sodium, and this is CH3. CH3 is the methyl group. So this is sodium methoxide. So this is how we name the compound, sodium methoxide. And over here, well, again, this is the methyl group, so you can call this methyl iodide, or you can call the compound iodomethane. Either way, it represents the same compound. So, in solution, what would happen if these two compounds were added together? Even if you had no organic chemistry experience, you should be able to work out the result of this reaction. You may want to take a moment and pause and work out the product, or we'll work on it together. So, if this is in solution, the first thing that you would consider is that thinking of what would happen with NaOH in solution. Well, we would get Na plus OH minus. It would dissolve in solution and produce Na plus and OH minus. Similarly, if you have sodium methoxide in solution, we would get Na plus and OCH3 minus. So that's how we start. We're going to begin our reaction mechanisms by identifying the compounds and then starting to work on the electronics, because that is the basis for the reaction. It's going to be opposites attract, like charges repel. It's the same old, same old. We're going to have different words for these types of things, but it's always going to be based on the same basic principles. So here, in terms of working out the electronics, all we have to do is look at the periodic table, which again will be provided to you on the MCAT exam. So you look on the periodic table knowing that iodine is bonded to carbon. Since carbon is bonded 4 times, so it's bonded 3 times to 3 different hydrogens and once to iodine. And so in the bond between carbon and iodine, how are the electrons shared? Well, you look at the periodic table, remembering that as from basic general chemistry, as you go to the right along the periodic table, you have an increase in electronegativity. And therefore, carbon is to the left, iodine is to the right on the periodic table, and therefore iodine has more electronegativity, meaning that when the atom is in a molecular environment, it has a greater pull on electrons. So iodine is going to be a little bit negatively charged because it's pulling electrons to itself. The symbol for a little bit negative is delta negative. Therefore, this carbon is going to be delta positive. This is very common arrangement in organic chemistry, that we get a delta positive carbon, because often, not always, but often carbon is bonded to atoms that are more electronegative. And this you would be able to determine based on the periodic table. So here we have delta negative and delta positive. Now, in order for you to make this reaction work out without any experience in organic chemistry, you need to know one more point. And that is the definition of a spectator ion. A spectator ion are ions that are found in the first and second group of the periodic table, with sodium and potassium and calcium and magnesium and so on. These are ions that are found in the first and second group of the periodic table that when they're involved in organic chemistry reactions, they're spectators, meaning that when the reaction is occurring, they're just going to observe. They don't get involved in the nitty-gritty. And so at the end of the reaction, when everything is finished, then that particular ion will associate with whatever might be left over, but it does not actively get involved in the reaction. So when we look at this, we have one spectator ion, which is the sodium, since it is in the first group of the periodic table. So once we know that sodium is a spectator ion, and we can just block that out of our minds for now, then when you observe the reaction, there's only one thing that can occur. After all, this is negatively charged. This has a partial positive charge. That has a partial negative charge. So the only thing in terms of opposites attracting would be this negative charge going to this partially positive charge. So then we have this negative charge, opposite attract, being attracted here to the positive charge. Why is this delta positive? Because the electrons have been pulled to the iodine, making the impact of the carbon nucleus more apparent, and therefore this negative charge is attracted to the carbon nucleus. Therefore, this substance is indeed a nucleophile. So that's our first definition was that of a nucleophile. So the first thing that happens in this reaction is that the nucleophile is going to attack the carbon, attracted to the carbon. And so in organic chemistry, the routine is that when you show an arrow, the arrow represents, when you're doing reactions, it represents the flow of electrons. In other words, really, technically, you don't show an arrow going from something positive to something negative. You show the electrons, the something negative or partially negative, going and attacking that which is positive. That's the routine. And so whenever you see an arrow like this, it means that we have an electron pair that is going to attack a particular atom. So this is the carbon nucleus. So here, this nucleophile comes in, it attacks the carbon. However, carbon can only be bonded 4 times. We know that because carbon has 4 valence shell electrons. It can have a maximum of being bonded to 4 different atoms, maximum. So if this oxygen were to come in here and to bond to this carbon, and so it would have 4 bonds, well, it would have 5, so it would have to kick something out. So it would break the weakest bond. Now, the carbon-hydrogen bonds are bonds that are pretty much equally shared, meaning the electronegativity difference between carbon and hydrogen is very low. But the electronegativity difference between carbon and iodine is so great that it is a polarized bond, where iodine is delta negative and carbon is delta positive. Because it's a polarized bond, because iodine is drawing electrons to itself, it means of the 4 bonds to carbon, the weakest bond would be that which is polarized between carbon and iodine. So when this comes in to bond with carbon, carbon must break a bond somewhere if it wants to bond with this new substance. And so indeed it could break a bond quite easily because it has a polarized bond. It has a bond in which iodine is dying to have as many electrons as possible. So as this comes in and knocks this carbon, this is going to leave. And that is our definition of a leaving.